All right. Good morning, everybody. It looks like everybody's seated, so we're going to get started with the first of our quick talks here. It's going to be a series of 15-minute talks given by students and faculty here at Moss Landing Marine Labs about different things going on, different projects, and interesting things we have here at the lab. So our first speaker today is Matt Hess. Matt is a student here at Moss Landing. He studies kelp, and he's going to tell you a lot about kelp. It's like his favorite thing. He's really excited to talk to you. All right, so give me a warm, let's give Matt a warm welcome here.
I'll figure it out.
All right.
Hi, everybody. Yeah, I love kelp. Welcome. I'm so excited to see so many seats filled. I thought with the first slot, y'all would still be asleep or checking out open house, but y'all are here to learn about kelp. And if you get bored, there are whales going off out there, so just look out there, look at the whales. I'll be up here talking about kelp. If I leave you with one thing today, one thing to remember, it's that kelp is beautiful. That's it. I don't want you to know anything other than that. And I'm going to learn how to use PowerPoint for the first time in my Moss Landing career. Here we go. 10 things you should know about kelp. I'm going to talk about kelp. I'm going to let my buddy Phil do actually probably most of the work. He's an underwater photographer here in Monterey. He did pretty much every photo on here. If it's not credited, it's one of Phil's photos. He's underwater, I think, more than a lot of the marine scientists here are, actually. It's kind of frustrating. So what is kelp? All right, let's start there. First thing you should know about kelp. What is it? Well, it's a taxonomic group. If you imagine the tree of life and how everything is related to each other, kelp is a branch of the tree of life. They all come from a single ancestor, right? So there's that. And so let's look at a couple of the things that are considered kelp, right? So that first one that you saw, that's called bull kelp. It forms a lot of the forest kind of up in the Pacific Northwest here. This one up here, this is what I want to point at. That one's called dragon kelp, Eularia, dragon kelp. That's something you see a lot in Alaska, the Aleutian Islands. Over here, I don't know what the common name for this one is. It's called, scientifically, it's called Aclonia. You see that a lot in Australia and South Africa. All right, so kelp's all over the world. That's a later slide, so don't worry about that. But it's got a lot of different forms. This one's cool. Golden kelp, Aurelia. Golden kelp is a new species. Well, it's not new anymore. It was new like a couple of years ago. But a couple of years ago when they came out and they said, we found a new type of kelp. And everybody's like, seriously? We're still discovering types of kelp? Like, what? There are new types of kelp being discovered around the world, all right? So it's kind of a taxonomic exploration of kelp around the world. A lot of people also consider kelp to be a big brown seaweed, right? Anything that's big and underwater and forming forests, you might just call that kelp. It's building ecosystems. So I put up this big brown seaweed, which by the tree of life, by taxonomy, is not technically kelp. But people call it kelp anyways because it is something that builds ecosystems, it builds forests. It is a big brown seaweed. I think that's a great definition. I'm fine. Everybody can be kelp. Everybody's welcome to the kelp family. Something cool about kelp is that it exists microscopically and it exists macroscopically. It gets really big. It's one of the biggest things okay, not whales, all right? I'm competing a lot with whales today. But it's one of the biggest plants underwater. It's one of the biggest seaweeds underwater. It can grow to be over 300 feet tall or deep, or however you want to consider it, right? It's massive. It's also tiny. It has a life stage, a completely different genetic individual that it cycles through in order to exist. That's just a couple of cells, a few cells wide. And it has to cycle through that and then become this massive organism again afterwards. But at that microscopic stage, it exhibits chemical warfare with other types of kelp. Right? Sexual chemical warfare. It does these pheromones, and other types of kelp will also send competitive pheromones that confuse kelp so that it doesn't find the other kelp and fertilize itself and exist. They fight on a chemical level. It's nuts down there. You might already know this one, but if you don't, kelp creates these beautiful ecosystems. This is why it's my favorite thing in the world. This is a shot from Monastery. It is, I think, the most beautiful underwater spot in the entire world. Things aggregate. They get pulled into kelp forests, and they live there for so many reasons I'm not going to get into right now. But it creates these beautiful ecosystems. And it's all over the world. Right? You might notice a massive belt missing in the middle. It does not like warm water, so the equator is not a really good spot to find kelp. But outside the equator, in the northern hemisphere and the southern hemisphere, where you get cooler waters like we have here in Monterey, there's going to be kelp. It's going to be there. For a plant, kelp actually moves really quickly. Right? It's one of the fastest growing plants on Earth. I think there's a species of bamboo somewhere that might grow a little bit faster, but it grows super fast. Some people say 2 feet a day. I think that's a little fast. That's being a little generous here, but it grows really fast. All these little fronds in a kelp forest, they only live to be 3 to 6 months old at the most. They just sit there and keep producing fronds, and it keeps turning over and over and over. It's moving fast for a plant. It comes and goes. It's physiologically advanced. Now, I'm going to move fast through this slide. This is what I study. So I could sit here all day and talk about this. I'm going to move really fast, though, right? On a cellular level, if you were to cut through one of the little ropey parts of the kelp, what they call the stipe, you'd see all these little openings. Those openings are plumbing. Those are tubes, right? If you look at it from the side, the tubes look like this. Here's a tube. It comes out, and it kind of widens and connects to another tube and goes that way. Like a tree, like a land plant, these things actually move carbohydrates and nitrogen and things that it needs and produces in one part of the kelp to the other parts that need it. They act like trees. It's incredible. But it evolved completely separately from the trees. Anybody know what that is in the background? Shout it out. What's that above the water? That's the aquarium. Kelp has human value, right? Like Monterey. A lot of people associate Monterey with the aquarium. Can you really put a number on kelp forests, though? Yeah, economists put numbers on everything. They value kelp forests at about $7.5 trillion globally, right? They provide about $500 billion annually of ecosystem services and tourism services and things like that. So they do have real economic value, especially when you consider from that original map that we saw how concentrated they are in those areas. Kelp is a buzzword. You hear it a lot, right? So I showed you that picture of the forest that is technically not taxonomically kelp, but people call it kelp because they like to include everything as kelp. That's because kelp is a buzzword. If you say something is kelp and it creates a kelp forest, it's a great way to get some funding. A lot of people are looking at kelp these days. It's really exciting, and a lot of work is being done because we still have a lot to learn. A lot to learn. I mean, kelp is behind seagrass when it comes to things. Seagrass! How much do we know about seagrass? Kelp is behind seagrass when it comes to like the amount of academic research that's happening. We still have a lot to learn. A lot of that is happening right here at Moss Landing Marine Labs. Thank you. Do we have time for questions? I did it! I'm at the 10-minute mark. We have 5 minutes for questions. I went fast because I was hoping that people would ask questions. So if you have questions, please. Otherwise, I'm going to talk about physiology.
Can I see that map again, the first map?
Yeah. You guys don't want me to talk about physiology. All right. There we go. Yeah. Actually, I saw a hand over here.
How nutritious is kelp just by itself?
Good question. So kelp has some interesting nutritional qualities. It's really rich in iodine, which we're kind of fine with iodine these days because they put in salt, and we put salt in everything. But in the times when that wasn't a thing, when it was an issue back in the early 20th century, kelp was a great rich source of iodine. It's actually really rich in proteins as well. And the cool thing about kelp is that the way that it produces its sugar, because it's so ancient, we can't actually digest them. It's just fiber. So it's technically a carb-free source of protein. That being said, how nutritious is it? It's nutritious. It's nutritious. How much kelp do I even eat on a daily basis? Not that much. I don't really see it becoming a staple. Does that answer your question?
Yeah.
Yeah. In the back?
Can you talk a little bit about the combination of like biotechnology and the uses of kelp in that arena?
Yeah, they're doing some cool stuff. There's a company out there called NotPla, like not plastic, NotPla. NotPla, I think, is getting some really good funding in the VC world. They're basically taking kelp derivatives, some of the stuff that's produced by kelp, and turning it into a plastic alternative. There's some folks out there that are taking kelp and turning it into clothing. They're spinning fibers out of it, which is really cool. Those are all new, really cool, neat, innovative things to do with kelp. We've been using kelp industrially for hundreds of years. In fact, during World War I, when a lot of our saltpeter came from Germany, and all of a sudden we weren't friends with Germany anymore, kelp was actually one of the main sources of phosphorus that we used to create the gunpowder for World War I. It's been used a lot as animal feed. It's been used a lot as fertilizer. So it's had a lot of resources that way. Those are the ones that come top of mind. Great question. Did that answer it?
Yeah.
Cool. Right here.
I got a physiology question for you.
Oh, yes! Okay.
How does photosynthesis in kelp differ between kelps and plants?
It's actually very similar. Photosynthesis is ancient, right? It's ancient. And so a lot of the mechanisms that kelp uses to photosynthesize, those pigments that capture light from the sun and turn it into carbohydrates, they're similar, but the difference is in the type of pigments that are used, right? So kelp is, there we go. Kelp is brown. Plants are green. And so the color, the types of pigments that you have in there that are capturing that light, that's going to be a little bit different. In fact, these do have green pigments in them, just like land plants do. They also have red in there. So what does red and green make? It makes brown, right? And red's a little bit more useful underwater. Blue light tends to attenuate pretty quickly. Green light tends to diminish as you get deeper. And so being able to capture a little bit more of that red light that percolates deeper in the water is a big advantage when it comes to producing energy. Does that answer your question?
Yeah, thank you.
So I'm walking on the beach and there's a giant mess of kelp. Is that kelp dead? And what can I do with it?
What is dead? No, that's a really good question. Kelp actually has a long life after it detaches from the bottom, right? It does a really great job. Like this blade will stay alive if I put it in a tank and I sit there and I can actually grow more kelp in a tank by sitting there and percolating it. One of the ways that kelp actually moves around the world is in these giant rafts. It comes detached from the bottom, floats up to the top, floats away to some other area where it actually will settle and create more kelp there, right? So technically, if you were to take it and rescue it, it might stay alive if you wanted to do that. That being said, if you leave it on the beach, it's gone. And that's actually a really cool phenomenon called eco-something subsidy. What is it? Cross-ecosystem subsidy? Is that right? Come on. Yeah, cross-system subsidy. There we go. It's this idea that kelp grows underwater, washes onto a beach. Kelp flies love kelp, and so they'll sit there, they'll start to eat the kelp. Well, birds love kelp flies, so birds will eat the kelp flies. Well, birds then move onto the dunes and poop, and that poop becomes fertilizer for the dune grass. And now we have dune grass, and all that just kind of percolates into the ecosystem that way. So what is dead? That's the question. What can you do with it? What I would recommend doing is, if it's on the beach for a little while, it's going to get super slimy. Take that slime and just rub it all over your skin. Another big use for kelp in industry is as nutraceuticals, right? That slime that kelp produces is actually gold to people who are producing like hand lotions and face lotions and things like that. So they're taking that gross stuff, putting it in a nice bottle and reselling it to you at a very high price. So the free stuff's on the beach. I think that's it. My information is somewhere, is there, down here at the bottom. Matthew Hess at sjsu.edu. Feel free to reach out if you have any other questions. Clearly, I like talking about this stuff, so I would love emails with questions. Last one question right here.
You talk about protein. Is that
Yep. Oh, 100%. Yeah, good source of protein. Also really great. One thing I didn't mention is that it's really high in umami flavors. Think of like manchego cheese, bacon, things like that have that umami flavor. Kelp has a lot of it too.
All right.
All right, I'm getting kicked off. Thank you, everybody.
All right. Is that our clicker? Or is it our clicker?
I don't care.
Okay, we're good. All righty, folks, we're gonna wait just a minute for anyone else to come in who wants to see the next talk. And then I will let you know who's talking and we will get started, okay? Thank you all for being a great audience so far.
Welcome to open house.
Woo! If we have enough chairs, you think?
I think so. I'm just gonna put it in their room.
I'll just try to.
I'm just trying to keep the walkways clear.
I'll try to. Five. And then it goes for how much? For me, I don't care. You can find it.
I mean, we should have it. Isn't it like a little like skinny one that?
We should get started.
Yes. All right. I will.
What?
Oh wait.
Yeah.
Oh, cool. Yay!
Alrighty, folks. So our next speaker is Olivia Doyne. Olivia is a student here at Moss Landing. She studies pismo-clams, and she's going to give you a talk about the pismo-clam species in the area and ongoing efforts to monitor the population and maybe do some restoration in the future.
All right, thank you, Doctor. Hi everyone. My name is Olivia. As you've heard, I am a student in Dr. Allison Haupt's lab at CSUMB, and I'm co-advised by Amanda Kahn here at Moss Landing Marine Labs. So today I'm going to tell you why clams rock, and I'll give you a little bit about their geological habitat as well. So first, you might be thinking to yourself, why clams? You might know them better for being in your clam chowder, eating them for dinner, but what do they do for the environment? So they are bioturbators, which means that they mix our sediment and allow for oxygen to infiltrate our sediments, and animals need oxygen to breathe, so they create habitat space for other animals. So more oxygen means more biodiversity, means a healthy ecosystem. They are also very ecologically important. They filter up to 60 liters of seawater per day, which equates to almost 22,000 liters per year from a single clam, which is pretty insane. And they are also an important food source for otters that you see in the bay, bat rays, and moon snails, and countless others. So, a little bit about their habitat. They live in a soft sediment habitat, which occupies almost 80% of the seafloor globally. And there's varying degrees of sediment sorting in our sediments. Sometimes you get some really coarse sand, sometimes you get some really fine sand you see in the mud flats, and sometimes they're mixed together as well. So infaunal invertebrates, or our guys without the backbones who live in the sand, have an optimal grain size. A little bit more about their beach. The beach slope means, a high beach slope means that there's larger grain sizes with higher wave exposure, which creates these steep beaches that you see. This metric also relates to how much habitat is available for species who inhabit the swash zone. And you may ask yourselves, what is the swash zone? So it's this area right here, in between the highest high tide and the lowest low tide. That's where our team works. So it's still considered the intertidal. So you might be asking yourselves, what happened to the clams and what's happening now? So the pismo clam fishery was booming in the early 1900s. Hundreds of thousands of pounds were being landed each year. So there's a very important commercial fishery here in Monterey Bay and across the California coastline. They were ranked third in mollusks in the state as well as an important fishery source. But unfortunately, they were over-harvested and their commercial fishery collapsed, which prompted the opening of the recreational fishery. And that is still open today. However, there is a size limit that we can take 5 inches for anywhere above Point Conception in Monterey and Santa Cruz County, and 4.5 inches south of San Luis Obispo. But unfortunately, this recreational fishery is de facto closed because we haven't found any legal-sized clams in the bay. And they're still being illegally taken in the bay. This was just from 2022, where hundreds of undersized clams were being taken in Santa Cruz County. And this was just from one day, and the largest clam was not even legal size. So, not much is known about pismos in Monterey Bay in general, as well. So Monterey Bay in particular lacks extensive population monitoring, abundance estimates, size estimates, or where they're distributed. Populations are recovering, but for the full restoration of their populations to occur, there must be a continued effort to keep an accurate record of their population sizes and identify physical drivers of their distribution, aka the sediments where they live. So this led me to my question: what is the ideal grain size, degree of sorting, or how mixed the sediments are, and beach slope for the pismo clam in Monterey Bay, the northernmost edge of their range and the most understudied? So I conducted a study across 11 sites. This is my ongoing thesis research, anywhere from Seacliff State Beach to Carmel River State Beach. And for our methodology, we adopted methods from Cal Poly San Luis Obispo. And we start off with a perpendicular transect. We start at the high tide watermark, the beginning of the swash zone, and we dig 30 meters into the surf. And we dig 1 shovel head deep as well and take our GPS points. So we shovel sand into the cart, run it out into the ocean, get a little wet. It's a really fun time, and we get a little bit silly with it. And we look for clams, and all that sand is washed out, and what's left over is our clams. If we don't have any, we go back to square one, and we start at a new section. If we do find some, we size them and then rebury them. So for their sediment sizing, we take our sediment samples from each section of our transect, from the start, middle, and end. We package our sand sample in the tinfoil you buy at your local grocery store. We dry it in an oven at 200 degrees Fahrenheit, record our initial sand mass, and then we separate it out by grain size. This is the Wentworth grain size class, so we can get an accurate measurement of their average grain size as well. We also go out usually around 2 in the morning to 5 in the morning over the summer, which gets very intense, but it's a great time, honestly. Here's us at 5 in the morning, and we are out there constantly throughout the year. And we also use what's called a differential global positioning system, which gets us an accuracy of our beach height up to 2 centimeters, which is really awesome. So we can get a really fine scale tune estimate of our beach slope. So here's a little bit of a day in our life. And we're just bringing it into the water, shoveling hundreds of thousands of pounds of sand. And then sometimes we jump in the water after, and it's a great time. And as you can imagine, sand is heavy, so we need so many volunteers out in the field, and we have almost 70 undergraduate volunteers help us out, which is a great effort. And you can follow us on TikTok at the Help Lab. Anywho, what have we found so far? North of the bay is where we find most of our clams. 4 out of the 11 sites had clams, which was from top to bottom: Manresa State Beach, Sunset State Beach, Moss Landing State Beach, and then here we have Del Monte, but only 1 clam. And we were like, what's going on here? So as you can see by our frequencies in Monterey Bay, the bigger the circle, the more clams are found there. So all of these clams are staying in this pocket in the northern half of Monterey Bay. And we were very confused by this. What's the difference between the north and the south? So in terms of our sedimentary results, here are all the beaches where we found clams, with Del Monte just 1. But then we have South Del Monte, which had a low grain size, which seemed like it was the most optimal, with a low grain size and a pretty flat beach slope, but no clams there. And we were wondering why that is. So you don't really have to know too much about a multiple logistic regression. Just know that the flatter the better is where we find clams. So if you get a flat beach, you'll find more clams there. The more mixed your sand is, so the more higher grain sizes you have and lower grain sizes you have, the better it is for our clams. And the smaller grain size is better. And that South Del Monte beach had a very small grain size, but no clams. So we were wondering what was the cause of that. As you can see here, our coefficient, we had a high predictability for South Del Monte with 0.83. That was the second highest predicted to have clams there. So in total, we thought that gentle beach slopes provided more space in the high intertidal for pismo clams. Smaller grain sizes provided optimal sand for burrowing, and heterogeneous sand or well-mixed sand may be useful for clams of different sizes to burrow and can promote recruitment as well. South Del Monte and Del Monte Beach had an ideal condition, but no clams, possibly due to beach grooming, human population counts, and as you can see in the upper part of Monterey Bay, there's an upwelling shadow, which means there's a long retention time for the water. So that water's just sitting there getting warm, possibly creating a better environment for these clams. So moving forward, we will continue to use sediment grain size and beach slope as indicators of beaches where clams will be found, but we still have to take into account their fluid environment as well. And we will be collecting and comparing seasonal data, and we will be comparing the limits of their historical distributions and adding more factors, both physical and biological, and we want to create more opportunities for undergrads as well. And we will also be making recommendations for the CDFW, possibly stricter in protocol where the clams are found, and we are near their their northern limits, and it takes them longer to mature and reproduce than their southern populations as well. And we got to fill that knowledge gap. So I'd like to thank my PI, Allison Haupt, all of our undergraduates who helped us as well, and I'll take any questions.
Is there any place you can dig for clams now?
You can dig anywhere you'd like for clams, but you can only take the sizes in Monterey Bay that are bigger than 5
Are there
inches.
5-inch clams anywhere?
Not in Monterey Bay, not that we found so far, but go down to Pismo Beach and you'll find a lot of more legal sized clams. They're coming back, yeah. Sure, did you have a question?
Yeah, what is their reproductive cycle, and why aren't they booming? Is it because of that?
Well, their reproductive cycle is bipartite, meaning that they have a larval stage that they send out into the ocean, into the pelagic zones, and they have to find their way back to the beaches. So a lot of that can be recruitment. Just like you saw with their upwelling shadow, all of that water is staying in the northern half of Monterey Bay, and it's having a hard time getting down to the southern end, so that could be a possible reason why there's a lot of recruitment in the northern half. So that's a great postulation. Yeah?
Is there any female and male here, or how long the reproduction happen?
Yes, there are female and males together. They broadcast spawn, so they send out their larvae, like I said, into the pelagic zones, and they reproduce that way. Yeah. And question?
Yeah, I was curious, for the distribution for why they're not at Del Monte, have you looked into the predator populations for them compared to those two locations? Are they pretty similar? Would it only be beach size, or could there be more predators at Del Monte than there are up north?
Right, that could be a top-down effect. We haven't been looking into predator counts or sea otter counts, but we are studying from the bottom up, so the physical to the biological, but it would be nice to study as well their predators creating less distribution down there as well, but that's a great hypothesis. Yeah. Is that it? All right, well, thank you, everyone.
There you go. All right. Give them a minute.
I don't have a nice video clip. I was like,
That's only giving the top minute time, so.
yeah. What time is the, battery life?
I believe that is 12:15 to 12:30, and then 12:30 to 1 is their food. I think we should probably. Is it just one person? Or
On the pan, I think it's a panel.
okay, so I think we'll put some chairs up there.
Yeah.
That's after my shift here, so not my problem.
Okay, yeah. I have a yearly meeting. I have my phone here.
I think I just started.
Okay, let's do it.
All right, everybody. So we're going to get started with our next talk. Our next speaker is Peter Jenkins. She is our director here at Moss Landing, and she's going to give a quick talk on mentoring and education in marine science. We're really excited for it. So let's give her a warm welcome, please.
Okay. Thanks for coming, everyone. Welcome. Oh, there's a bell. I got a bell. Excellent. So yeah, I'm the executive director here at Moss Landing Marine Labs. It's a super privilege to be in this role. And I wanted to talk a little bit about the next generation of marine scientists, why we need to have a next generation of marine scientists, and how Moss Landing Marine Labs gives them a really unique education to prepare them for those careers. So I'm going to start just really broadly, like taking a really big zoom out and talk about climate for a second. So our ocean is changing in lots of different ways. This is just one example of that, right? So there's, this is a temperature record. Oh, that pointer's not very good. So from the early 80s to this year, and you can see that on average, the sea surface temperatures are getting warmer. The climate in California is changing. We are getting warmer temperatures. We are getting more frequent droughts. We're getting decreased snowpack. Sea level is rising. Wildfires are increasing, and all of these things are causing change. And these changes are going to have impacts on a broad array of things, right? Not just on the ecosystems, but also on the people who live in those environments. So we have lots of examples. This is an example from a couple of years ago, which was a storm in Capitola. And as sea level rises, the impacts of storm surges increase. Wrong direction. We have here a picture of what was a really active kelp forest and is now a sea urchin barren. So the sea urchins have eaten up all of the kelp. There's a lot of really great information out there about that actually out in the courtyard. There's a group of people who are studying that. And then this is a picture of a harmful algal bloom, which is actually something that's happening in Southern California right now. And so all of these are changes that are happening in our ocean. And the science that's done here at Moss Landing Marine Labs does work to understand those changes and to figure out not just how the changes are happening, what the impacts are going to be, and how we can find ways to minimize those impacts and how we build resilience to those changes. So I'm just going to give you a couple of examples of the work that some of the scientists and students are involved in at Moss Landing. So this is a collaborative group of people. All of these dots are locations where people contribute to this. It's the CalHab map. So this is a group of people that monitor for those harmful algal blooms and can detect them early. So we have a group that goes down and takes water samples every week off of the pier in Monterey, and it's part of this California-wide system. There's a group studying ocean iron fertilization as one possible way that we could reduce the concentration of atmospheric carbon dioxide, which is a greenhouse gas. There is a group, if you go to the ichthyology lab and also out in the courtyard, that is the California Collaborative Fisheries Research, and they look at how our marine protected areas impact fisheries, how climate impacts fisheries. And then there's this Central and Northern California Ocean Observing System. And so that's a really important one. An ocean observing system is just a broad network of people making observations of the oceans. This is exactly what you think it is. But I think it's hard to underestimate the potential impacts of those and what would happen if we didn't have that kind of observation system. So this impacts how we prepare for changes in climate, but it also impacts how we look at fisheries and maintaining fisheries populations. It impacts how we do storm forecasting and extreme weather forecasting. It impacts national security because it impacts the way that ships travel through the ocean. So there's huge impacts and huge values in these large things. And so our students are involved in all of these projects. This is another really good example of a really collaborative project. So there was the Moss Landing battery fire that most of you heard about probably. And we were really fortunate that we had people here who already had data from the Elkhorn Slough. So we knew what the background concentrations of the different chemicals were in the soil already. And so we were able to jump right in, and this is Ivano Aieo, he's giving a talk later today, measuring the concentration of metals in those soils. But it's a very collaborative project that involves Moss Landing Marine Labs, the Elkhorn Slough Foundation, the Marine Pollution Group, California Fish and Wildlife, and scientists from UC Santa Cruz. So the students that come here get both the education they get here, but they also get these connections that can serve their careers really well. So they get to meet people from all of these other institutions. Okay, so the mission of Moss Landing Marine Labs is to provision the marine scientists of the future. How do we do that? There's a couple of really critical things that we do here that are unique. One is that all of our students work with faculty advisors and instructors that are really on the cutting edge of their own disciplines. So each one of the faculty members that you will meet here in the labs are nationally recognized for their contributions to the field. Our students, as master students, get hands-on student-led research experience with those alongside those scientists. So they get really excellent mentoring. We have a really broad range of research facilities that I'll talk about a little bit moving forward. That's really unique because of our location. And we give them unparalleled access to this amazing marine environment. So I'll talk a little bit about that. So our goal really is to provide the students with the skills and the training that they need in a variety of jobs. Some of our students will go on to get PhDs and go into academia. Some of them will work for government agencies, conservation and management. So these are the people that are being trained to then go out and solve the problems that are being created. Okay, so the most unique thing about our lab is our location right here at the heart of the Monterey Bay. And not only do we have access to the Monterey Bay, but we're also at the entrance to the Elkhorn Slough. So we have this really diverse ecosystem. And you just heard in the previous talk that the conditions in the northern part of the bay can be quite different than those in the southern part of the bay. We're right here at the head of the Monterey Bay Canyon. It's just a really, really dynamic environment. We have a boating program. If you go out on the parking lot, you'll see one of our vessels that's out there. But we have a variety of different boats. And what's unique about this is that we also train the students. So students that are going to use the boating program a lot in their research get trained in how to use the boats. They can get trained in how to trailer the boats and take them to their different field sites. So here you have some students sampling the water just in the slough. This is actually, you can see the lab back here in the background. So they get trained and then they go out and they do their own work. We also have a larger vessel, the John Martin. This is our largest vessel. It's a 57-foot vessel. It has an A-frame on the back that allows us to lower instrumentation. So here you see what we call a CTD that measures temperature, depth, and salinity as it goes down, and it also takes water samples. So students, they don't captain this boat. We have an official boat captain, but they do get trained as technicians, and they are student assistants, and they learn a lot of this boating expertise. We also have a really thriving research diving program. So this is training program that is not just scuba diving. You start out as like the basic scuba diver, and then it takes quite a lot of training to really become a scientific diver. And we do all of that on site. And so you have students who have research projects. They go out, they sign up, they get the truck, they get the boat, they take the boat down to their field site, they grab 3 of their friends, and they go diving on their site, and that is how they're doing their work. So it's a really unique hands-on training. We also have a really lovely lab facility, which you're going to see today as you walk around. So access to lots of instrumentations and really great labs. And we also have seawater access. So we have 2 pipes that stick out into the ocean. We pull in seawater, and that allows us to do science where we can bring seawater up here to the main building where we have an aquarium room that has a lot of different experiments. And we also have a large aquaculture facility where we can raise kelp and shellfish and all of these things. So that is a really, you can't see it, but it's a really important part of the lab and the resources that are available to the students. And then I also want to talk just about the uniqueness of the class structure that we have. So in most universities, you go to class and you have it on Monday and Wednesdays at 2 o'clock, right? These classes you have 1 class and it goes all day. And what that allows the instructors to do is to really think about how to integrate the hands-on experience with the lecture component of it. You can go ahead and just sit anywhere. So this is an example of our intertidal class last year. And they went diving, so the students themselves went diving. They built these sort of fake kelp holds at them, and then they put them out in different locations. They left them out there for, I think it was 5 or 6 weeks, and then they brought them back and looked at all of the critters that had collected there and sort of did an analysis of, does it matter where you place these? Where are these guys coming from? So it's a really hands-on experience, both in the classroom and in the lab. And so I'm just going to finish up here and give you some time for questions, but I just want you to know everything today has been completely organized by the students. They're incredibly independent people, and their research projects are really amazing. Anything that's donated today, whether it's the silent auction or the merch or all of that, goes directly to student scholarships. It's not for funding the research. It's just because life here is expensive. And one of the things that we're really trying to do and to be conscious of here at Moss Landing Marine Labs is to recognize that these changes in the ocean and the changes in the climate impact a really broad community of people. And it's really important that we create a program that gives people from all backgrounds access to that kind of education. And so one of the ways that we do that is by trying to provide more scholarship support. So I'm going to end there, and I am happy to take questions if you have any. Yeah.
So the students here are all graduate students from San Jose State?
They are all graduate students. About half of them are from San Jose State, and about half of them are from CSU Monterey Bay. Okay. So all of the faculty that are here on site are faculty from San Jose State, but we have a very close working relationship with CSU Monterey Bay, so the faculty from their marine science department can have graduate students that come through Moss Landing. So that's why, like Olivia from before, her advisor was from CSU Monterey. Yeah.
Can you join here if you are not a student?
If you're not a student, there are some labs that have volunteers and have opportunities for volunteers. But yeah, most of the work is done by the students and the faculty. Yeah.
I'm trying to understand your presentation. What kind of problems you try to solve? And I don't understand exactly the problem.
I'm sorry, I didn't probably hear
You talk about problem and issue that the research is trying to solve.
Yeah.
So I'm trying to understand what exactly the problems that we're trying to solve. Is it a cause for food that are going to be eliminated by just kind of changes in the oceans? Is it going to be affected the kind of human living next to the coast?
Yeah.
And if yes, that's gonna nature basically gonna impact human, and human gonna get shrunk. So what's wrong with that?
What's wrong with that? Well, that's a philosophical question, but I will say that I do think that the impacts are really varied, so it's hard to summarize in one talk. I encourage you to ask the students about that when you go to individual labs. But absolutely, the changes in the ocean and the changes in weather will impact society, both on the coastline and inland, right? So we think of the ocean as just being, it's right here, and so maybe it's just the coast, but because the ocean covers so much of the planet, it's really what controls the climate of the Earth. So it has a really, really broad impact. I'll take 2 more questions, and I think they're going to make it wrap up. Yeah.
So the data that you gathered, let's say from now or future, is only accessed by SJSU and CSU, or also with the government? Or another question is like 20, 25 years from now, are those data going to be accessed?
The data? Yeah. So that's a good question. So the data, it depends on the kind of data and who the group is, but as a sort of pretty broad rule, any data that is collected at a higher institute of higher education is funded either through the state or the government and is required to be publicly available. So yeah, all of the data that's collected is publicly available. The only people that keep data secret is the military. That's not us. We're not them.
How does the scope of what you do here differ from the Monterey Bay Aquarium research people?
Oh, yeah, so Monterey Bay Aquarium Research Institute is just they're our neighbors. And they are really incredible group of engineers in particular, so a lot of their work is focused on engineering, and they do a lot of really deep sea stuff. So there's definitely overlap, and there's lots of collaborations between the groups, but if I had to sort of summarize it broadly, I would say that we are a little bit more focused on the coastal and the ecosystem stuff, and they are a little bit more deep ocean impacts. But there's definitely connections, and we have a really nice working relationship with them. Yeah. Okay, thank you very
All
much.
right, sorry, I have to cut her off. Yeah, we got a lot of people on the talk. Thank you very much. All right, we're going to bring in some more people, and our next speaker is going to come up.
Will you heckle us if we're in the front?
Oh no, don't worry, I don't heckle anybody. Deep sea. But it's here.
I'll close it, it's fine. I got it.
Dang, a full house.
Oh, yeah.
This is like more than seminars. Don't worry about it. I can.
I'll give you 5 to it.
Awesome. All right. Are you still introducing me, or am I?
Alrighty, everybody. So, thank you all for coming. Our next speaker today is Ilaria. Ilaria is a student here at Moss Landing who's studying hydrothermal vents. She's going to give you a talk about hydrothermal vents and some of the research she does with working with them. All right. So let's give her a warm welcome, and then you can take it away.
I'm Ilaria Bardini. I'm in the invertebrate ecology lab here at MLML, advised under Dr. Amanda Kahn, and I study hydrothermal vents. So today I'm just going to be giving you kind of a broad overview about hydrothermal vents, what they are, and then a little bit about my research. So this is kind of a silly slide, but imagine a place with no sunlight, crushing pressure, and boiling water spewing from the sea floor. If you imagine something like this, then there you go. So this is a hydrothermal vent, and they're extremely amazing. And so I'm going to just talk a little bit more broadly about them and what they are. So they typically form at mid-ocean ridges. So water basically goes into these cracks and fissures in the ocean floor, and then it's warmed by this magma chamber here. And then it goes through this thing called a phase change. So the water separates into a less dense vapor phase and then a denser brine phase. And we're going to come back to the brine phase in a little bit. Does anyone have any guesses about how hot the water is when it comes out? Any guesses at all? You can just throw any numbers out there. 500 degrees. 800 degrees.
1200 C.
Okay. Any more else? Any else? You guys are pretty close. It's 400 degrees Celsius at the highest temperatures. Yeah, it's pretty hot. It's hard for anything to live down there, as you can imagine. So we've got two kinds of flow that ends up happening. We have this warm diffuse flow, and then we have this hot, very focused flow. So if you saw this vent before, that all this smoke you see spewing out, that's what we call focus flow, because it's the hottest, and it just keeps going. This diffuse flow is really cool because it's not necessarily exactly where the vent is, but it's more mild. And so what ends up happening is this fluid is being expelled out, and you would think that the area right around the vent would be impacted, but it contains all these heavy metals that end up actually traveling across the seafloor and building structures and adding to what we see in the deep sea bottom. So this is what we think the current distribution of hydrothermal vents are. And so what's interesting is that sometimes it's impossible to find these vents. So we also use like chemical profiles to kind of guess where we think vents are, but for a lot of them, we're not actually 100% sure that they're there, but it's just speculation. So life without light. As you can probably imagine, there's no sunlight down there. And so being able to get nutrients for animals to live is really, really hard. So what ends up happening is that we have chemosynthetic organisms that feed off of the hydrothermal vent fluid that's coming out. So these are called yeti crabs. They're pretty cool. And so what they do with all that hair on their arms is they actually grow bacteria from the hydrothermal vent fluid, and then they eat it. So that's pretty cool. So a lot of these organisms here, got some bacteria there. These are vent worms. We have spider crabs and mussels, and they're all being fed by this hydrothermal vent fluid in an otherwise food-limited environment. So another thing I want to talk about, and this kind of goes into the research that I do, is community succession, which is basically the way that organisms and communities occupy a space over time. So by the vents, you've got these certain kinds of organisms that form, but they in turn support other organisms. So as you get farther from the vent, you'll see different things. Think about like when a fire happens in the forest and the plants grow back. That's kind of what this is like. And so from my research, this is what I'm seeing so far. At the vent sites, you've got a lot of hydrothermal vent worms and sea spiders. Then you move on to, these are brittle stars and sea pigs, which are really, really cute. And then you get to octopuses, which are also very awesome. So deep sea exploration, as you can imagine, it's really hard to get down there. We can't scuba dive down there, that would just be too much. So these are actually some of the equipment that I've used for my research. So I went on a research cruise last summer and I was out there for 2 weeks. It's weird to not see any land for 2 weeks. It's super weird. But this is the RV Atlantis, and it's one of Woods Hole's Oceanographic Institute's boats. And this is ROV Jason. So ROV Jason has a bunch of cameras on it, a bunch of robotic arms, and that's what we sent down to explore these hydrothermal vents that I was looking at over the summer. So this kind of goes, now we're going really into what I study. So I explore brine-dominated hydrothermal vents. So remember how I was saying that there's two kinds of fluid that comes out of the vents themselves, that brine-dominated and that more vapor fluid vents? Well, I study brine-dominated vents. And so typically at vents, we've got more of that vapor fluid that's coming out. But at these locations, specifically along the cleft segment of the Juan de Fuca Ridge, which is off the coast of Washington, they have more brine-dominated fluid. And so this is saltier, this is higher in metals, and it creates, if you can imagine, an even more extreme environment for animals to form. And so because of this, we're expecting that we're going to see different kinds of megafaunal communities than we would see at other places. So my current working title, everything's changing in science, but brine-dominated hydrothermal vents, megafaunal community structure and dynamics of the cleft segment of the Juan de Fuca Ridge. So what are brine-dominated vents and why the Juan de Fuca Ridge? I kind of explained a little bit already, but this is just a little bit more detailed. Much saltier, lower gas concentrations, higher metal concentrations, and prolonged phase separation. So the brine storage is actually being stored within the ocean crust for much longer than other vents. And so actually over time, or centuries, this brine fluid is still being expelled, meaning that the organisms down there are still feeding off that vent fluid, which means that those communities are probably different as well. So with the cleft segment, which is off the coast of Washington, we have continuous stable venting and allowing more times for communities to grow and develop. So as I said before, it's off the coast of Washington. The cleft segment, I wish I had a pointer, is like the bottom part of that. So if you see those like three parts, that's what we're looking at. This was kind of the track that the ROV took. So I have 13 transects that I'm looking at, and then 7 fine scale chimney transects. And so basically these were really up close to each chimney, and this is a really great way to see like the percent cover that organisms are taking up on the chimney itself. These are glass sponges, and we were able to collect a lot of those. So currently, I'm dissolving their tissue, and they have this skeletal structure with spicules in it, and I'm trying to ID each spicule and figure out what the heck is even those sponges are, which is really cool. So this is how I look at my data that I have. So really, I have like hours and hours and hours of video footage that I'm parsing through and sorting through. So as you can imagine, that takes a really long time. So I'm using this video annotation software called Beagle. And what's really awesome about it is that you can make video annotations. So you can see it's a lot of brittle stars. So that's one brittle star, I don't know what that is. But these are a bunch of brittle stars and glass sponges that I'm seeing. And this is a really amazing piece of software that I'm using. Currently, I'm trying to figure out how to do machine learning. So you can teach this software to identify things for you. But currently, my most seen organisms are brittle stars and then unknown fleshy blob with purple streaks. So if I don't know what it is, how would I teach something else to know what it is? But that's currently where I'm at. Any questions? Yeah?
How did you choose this area of research, or how did you come into it?
That's a great question. I've always been interested in deep sea research. My last research that I did was beluga whales, so very different. But I got interested in this mainly because my advisor had this project available, and I was like, this is awesome, this is amazing, this is definitely something I want to do. Honestly, going into it, I never thought that I'd be able to do deep sea research because it's such a hard field to get into, but I'm really lucky.
Cool, thank you.
Yeah.
So you actually took samples from deep sea?
Yeah.
And you have it in the lab?
Yeah.
And then you do the research that you talk about metals and whatever, you have the combination of this idol. Is that correct?
Yes, so I have a bunch of biological samples. So mostly sponges, some snails. Yeah, mostly sponges and snails. Yeah.
So when you get it into the lab, is it changing or is it stay the same?
Yeah, we preserve them. So we preserve them for a lot of different things: RNA, DNA, taxonomy. So they're all in their different preservatives. So they're not degrading or anything. So we're still able to see them pretty much like they were when we pulled them up. Another question? Yeah.
What depths are these?
Oh, good question. These are about 2,000 meters below the surface. Yeah, 2,000 meters. Yeah. Anything else? Yeah.
Did you get to drive the robot thing?
I didn't get to drive the robot, but I got to drive the camera. So, I got to, yeah.
And how do you collect?
Yeah, so the ROV has these robotic arms. And so I could never do it, but we have a bunch of amazing engineers who are experts at this, that are in what's called the ROV van. So it's basically a box where all these monitors are set up. So this is kind of a view of what I was seeing when I was down there. And so the engineers are in there with us, and they are remotely operating these arms to collect these samples. So that's just a chimney piece. But if you can imagine it, the sponge samples that we're collecting are this big, and some of them are this tiny and very thin. And so to be able to do that is amazing. They're incredible. But yeah, we use robotic arms.
Is the van on Atlantis on the surface?
Oh, it's on the RV Atlantis. Yeah, so it's on the surface. It's for the RV Atlantis, it's on the boat, but it's not inside the boat, but it's like a metal box that's outside of it. Yeah. Oh, so many questions. We'll go here and then here.
I was just making an observation of this. This is such a otherworldly environment, and it's like we must be learning things that we'll wind up applying later on to exploring other planets.
Well, that's actually what a lot of this is about. So the thought is also that hydrothermal vents are possibly an idea or a window into the origin of life on Earth because of just how extreme they are, and also extraterrestrial exploration. Yeah. Who has other questions? Yes.
Is there anything special that needs to be done, when you're bringing a sample up to the surface, to preserve it?
We put them in boxes. there's boxes on the ROV itself, and so, or sorry, yeah, the ROV. No, we don't really do anything special. We just try our best to make sure we don't destroy the samples and put them in a box and slowly come up to the surface.
Extreme environmental change.
Yeah. Other questions? Yeah.
The pressure over there is very high under the of everything. When we get the samples outside, do they expand?
That's a great question, but I don't know. I'm not sure. That's a good question. I didn't observe any changes. Any other questions? I think I have time for one more question. Okay. So yeah, I'm just like floating around today, so if you have more questions about hydrothermal vents or want to ask me any questions about invertebrate ecology or just deep sea research in general, I'll be around. Thank you.
All right. Well, thank you, everybody. I'm going to open the door so we can let some people filter in. We can have some new stuff. We do have seating around. Thank you all. You've been a great audience so far.
Dive into the marine career.
Huh? Oh, actually, no, I'm 12 to 1.
Okay.
It's right here.
I need to bounce, but I wouldn't just have to get out here. But if you want to take over.
Oh, there's nobody.
What? Okay. Are you floating right now?
I'm taking over.
Great. Yeah, so we're just doing a quick introduction.
Great.
So, 5 and pretty much.
Okay.
A lot of chairs.
Yeah, would you be able to come back after my 15 minutes, or where are you going right now?
I'm going outside today.
Okay. I'll try to find something. Yeah. If you see Carrie.
I'll see if I can grab some of this.
Yeah. You see what time? Yeah, we'll serve you at 5.
I'm gonna be like distracted by that ocean,
It's just intimidating. So many distractions. I'm always looking up, and then I look at this quill. So fair. So I was admiring this too. Like just everything out. Thing they've done is so like look at this quill. It's crazy. It's crazy. If you go, obviously if you go after, I felt bad. Wow. Like it was a strap. No, we had a quilt mill. So we had a just like they have something. Yeah, like in their position, they come up to that's amazing. Yeah. All right. Hello, everyone. I'm here to introduce our next speaker. So from Stanford University at Hopkins Marine Station, we have Stephanie Cuddle. Sorry, thank you. More Than Science, my creative dive into marine careers. So give a warm welcome and big round of applause for Stephanie.
Hello, everybody, and good morning. It's a pleasure to be here with you. Today I'm going to be talking a little bit about my career, which is kind of taking an interdisciplinary lens to marine sciences. And then I'll kind of wrap up by sharing a little bit about what you all can do as community members to care and advocate for the ocean, or if you're interested in a marine career, kind of what paths look like for that. So as was mentioned, I'm a PhD student, a first-year PhD student at Stanford, primarily based on main campus, but my lab is at Hopkins Marine Station, just down in Monterey, so not too far away from here at all. So again, I'm gonna kind of talk about how I turned my childhood fascination with the ocean and my personal connection to the ocean into a career. And so this photo on the left here, I don't know if this works or not. Oh, I went back. Yeah, there we go. So this photo on the left here is just me and my cousins playing on a beach in the Gulf of Mexico in Destin, Florida, where both of our grandparents lived. And so as kids, we would meet there in the summers and spend a lot of time on that ocean and in that ocean and on that beach. And so that was kind of my first initial exposure to the ocean. But I'm originally from North Carolina, and I did my undergrad in North Carolina as well. And eventually, I went from here to holding sharks on boats and turning my passion for the ocean into a career. So as a young child, I was super fascinated with the ocean, with nature. I spent a lot of time outdoors. And so this is a photo of me and my sister and some of our classmates in the creek nearby our home. But as I mentioned before, when I would go to the beach where my grandparents lived, my grandmother would save these little containers for me, and I would collect little fish, and I would observe them and take notes on them, and I was just fascinated by the life in the ocean, right? But simultaneously, I was super interested by human communities and culture and society, and so I spent a lot of time doing things like studying colonial candle making, like this photo up in the top, and just being super fascinated by history as well. And so when I got to high school, I got involved in Model United Nations, which is exactly what it sounds like, a model of the United Nations. And it's very similar to debate, but essentially you get kind of met with complex issues in society and you're assigned a country and you get to represent that issue from the perspective of that country. And so you get to kind of research cultures and policies and perspectives. And I found that really interesting, but I was still super fixated on the ocean. So when I got to college, I was wondering, how can I combine all of these things that I love, and is there a way to even do that into a career that I'm passionate about, but that can also make a difference? And so I got scuba certified so that I could kind of understand the ocean, not just from the beach, but under the water as well. And then I did my undergraduate degree at the University of North Carolina at Chapel Hill, go Heels. And I majored in environmental science, and I got minors in marine science and environmental justice. And so this is a cool video. Let's see if it'll play here. Oops. Yeah, here we go. Where I got my first exposure to some really cool field research. So this is in the Galapagos, where I still do most of my work. And this is my dive buddy, Lavisa. I'm behind the camera. And we were diving with some sharks and just seeing what they were doing. But here she waves her hands, which spooks them a little bit. And I got some really cool footage of them swimming up to me. They're super friendly, a little shy, but not dangerous at all. And so I was really excited about field research. I love scuba diving. I loved interacting with these species. And so I see some familiar faces in here where I talked about my shark tagging project. So that's me on the boat doing some of the shark tagging, the marking recapture study. And and so I got to do this work in the Galapagos, and I also got to go to my undergraduate's marine lab, very similar to what you see here on the beach, and get exposure to this kind of field research. But I mentioned before that I got a minor in environmental justice, and that's kind of where I combined some of this interest with human communities and culture and the importance of understanding how these ecosystems and these environments impact us and impact human communities. So as I mentioned, I'm from North Carolina, and through my minor, I learned that the environmental justice movement actually got its start in Warren County, North Carolina, which is not too far from Chapel Hill. And this movement started because there was a company, a landfill company, that was taking these toxins from landfills, and they were planning to pave the roads through this community that was a predominantly Black community. And the company sort of assumed, well, no one's gonna know, no one's gonna care, we'll just sort of pave the roads and then we'll leave, and then whatever happens, happens, right? But the community and the reverend here who led this movement, Robert Bullock, said, absolutely not. This can cause a lot of diseases, this will harm us, and we're not gonna stand for this, we're gonna stick up for ourselves. And so there was massive amounts of movement and mobilization around this. And there was also a lot of youth that got involved in protesting this. And so this was kind of the birth of the environmental justice movement. And since then, and this is from the American Public Health Association, but environmental justice is essentially the idea that all people and communities, regardless of their background, their race, their ethnicity, their gender, their sex, have the right to live and thrive in a safe and healthy environment with equal environmental protections and meaningful involvement in these actions too. So in other words, they get a voice in what happens to them and how they're impacted and exposed to environmental hazards. And since then, since the Warren County kind of mobilization and birth of the environmental justice movement, there's been huge mobilization around other things that are impacting people's lives. So this is specifically some photos from protests around the Dakota Access Pipeline, which is essentially an oil pipeline that's was planned to go through the middle of the country. And it sort of had two hazards. One, it was going over this major source of fresh water that a lot of people in the middle of the country rely on for their drinking water. So if the pipeline had any sort of leaks, it would contaminate all of this, and several hundred million people would be impacted. But it also was going through sacred land to the indigenous communities that live in sort of the central plains part of the country. And so this is a movement that's ongoing and was kind of birthed in North Carolina. So that kind of connected me to understand and further understand that we are intrinsically linked to our natural environments, where we rely on it for our livelihoods, for our food, for recreation, for a variety of different sources. And so it's really important to understand how are we impacted by the environments that we live in, but also how do we impact those environments and kind of understanding how those dynamics work. So now I'm in my PhD at Stanford, and I did sort of figure out how I can combine this interest in culture and policy and the ocean. And so my research now kind of focuses on marine protected areas. And so this photo that you see here is kind of showing what marine protected area boundaries look like. So when you're in a marine protected area, there's sort of usually fishing restrictions, there's restrictions on what can happen in that marine protected area, whereas that's not the case in the unprotected area. In some places you can fish in the marine protected area and there's some restrictions, but it's meant to kind of preserve the life there, but also the livelihoods of people that rely on those ecosystems. So my research focuses on understanding how we can make these marine protected areas better for the species that live there, but also for the human communities that are relying on those ecosystems for their livelihoods and for feeding their families and for their jobs. And so a common problem in these unprotected areas is that when fishing activity goes on, oftentimes species like sharks and sea turtles are getting caught in fishing nets. But that's not the intention. And so I work to kind of understand how we can make maybe gear modifications or work with fishing communities to understand what are the best ways that we can mitigate this, but also not have negative impacts on the way that we need to live and survive in these ecosystems. So my key takeaways for you today are that the field of marine science is vast, and you can combine what you're passionate about and make a tangible difference, whether that's policy and marine sciences, whether that's economics, whether it's art in the ocean. There's a variety of ways to make this into a career. And so for me specifically, I'm hoping to kind of take these combined interests to inform policy and government decisions around this. But also, you don't have to have a specific career in marine sciences, although I'm biased and I think that everybody should be in marine sciences, to advocate for the ocean, the creatures within, and the people who rely on it, because we are all impacted by the ocean and what happens to it. And so it's important to be well-read and understand what's going on in this field, which all of you are here and learning about that, and so that's amazing. And also kind of reading and watching documentaries about this as well, and then voting and advocating for people that will protect you, protect the environment, and make sure that you're not being exposed to undue harm that can be caused as well. So with that, I will wrap up. Thank you all so much. Please feel free to reach out if you have any questions, and I'll also take questions here for a couple of minutes. And then I'm with the Black Ocean Stanford Scholars in the back patio, so if you have more questions after this, feel free to stop by and we can chat more. Yeah, thank you so much.
Pacific Island Marine Sanctuary. is there a role that you now have to do in order to try to protect that marine sanctuary, or
Yeah.
where do we go from here?
Yeah, this is a good question. So I will preface this by saying that I am not an advocate of lifting of marine protected areas and marine reserves. However, in this specific scenario, and I highly recommend reading about this as well, there was a lot of harm that was done to those local communities, and they were not consulted before this marine protected area was implemented. And so while I think that just lifting the marine protected area and trying to destroy all of the environmental protection that we're putting in place is negative, I do think that there's an avenue in which we can sort of redo this marine protected area, but with more input and insight from the community that relies specifically on that ecosystem. And so I personally don't work in that region. Most of my work is still in the Galapagos and off the coast of California. But from colleagues that work in that area, I think that there is a significant role that they can play in sort of uplifting the voices of community members that have a lot to say and a lot of thoughts on this region and what their needs are and what the ecosystem needs are. And so I think that this might be sort of a blessing in disguise of a way that we can kind of right some previous wrongs that have been done in the implementation of that marine reserve.
That at least gives me some hope. My
Yeah.
husband is a fisherman, and he said that we're so overfished.
Yeah.
All the places he used to go so overfished. So when he saw that, he's like, well, we're definitely probably already are overfished. And if one more area is opened up, and that was put under W Bush.
Right. Yeah, I mean, we need more people, we need more fishermen voices to kind of be advocating for this as well, because there exists a world in which we're all kind of putting input in these marine protected areas, kind of centering the voices of people that are out on the water every day, like fishermen, and understand these ecosystems more deeply than I ever can as a student that spends some time in the field, but most of my time behind a desk. So yeah, absolutely. Yeah.
What subject do you need to take to go into marine biology?
Yeah, this is a great question. So I recommend taking the things that you're most interested in. And I know that's kind of a vague answer, but if there's a specific area of science or a program, a summer camp, or a class at school that is interesting to you, whether it be biology or specifically like a sea turtle camp, I think that those are amazing opportunities. But I think something that has enabled me to be able to do so many different things is by taking a wide variety of classes. So a lot of history classes, but also a lot of marine science classes. Yeah, and I think also you can be doing some reading or some watching videos and things like that on your own too, to learn about these ecosystems. And you might be able to get more information out of that than you can in some of your classes that might be more broad. Yeah. I'll go here and then there, and then I don't know how I'm doing on time. Okay, we'll do one question. Yeah.
Is there a radical faction that just sees the fisheries, all the fisheries, NOAA and that are managing, and all the fisheries butting heads against just leaving it alone? Because I'm a vegan, so I say just leave it alone, because I don't eat fish. And that's radical, I suppose.
Yeah.
But that, I realize that the fisheries they're doomed to extinction. The fish go extinct.
Yeah, I mean, that's complex because I think while from my perspective, we live in and I was vegan for a long time, I was vegetarian for a little bit, and now I'm a pescatarian. So I've kind of had all of these different paths. And I think that we have to be cognizant of the fact that a lot of communities, especially island communities, rely on these ecosystems as their main source of protein. There's a lot of work going into sustainable seafood for making sure that youth in particular, and places where there's not a lot of access to nutrition, are getting the nutrition that they need through fish, because there are no other avenues for that. And so I think I caution against completely leaving these ecosystems alone. But I do think that we need management that works well for that ecosystem to not doom fisheries to extinction. And I think that there are a lot of scenarios where that is the case. There's there are island communities and local and indigenous communities that have been managing fisheries for thousands, hundreds of thousands of years, and those fisheries never went extinct. And so I think with insight from those communities, we can have the best of both worlds.
But their methods were primitive. I mean, they were throwing the net out on the reef. I mean, it was not industrial fishing.
Sure, yeah.
I think it's the biggest problem in
Absolutely.
fisheries and agriculture and everything.
Yeah, absolutely. We should definitely talk more. I know I'm out of time, so I'm going to wrap up, but I would love to, talk more after. Thank you so much.
Oh.
Just open the door for a minute.
Oh.
You want to open the door, Haley?
That's okay.
It's nice to see
Go ahead. Okay, everyone. Our next talk is from Madison here. She is a second year here at Moss Landing in the ichthyology lab. And today she's going to be talking about rockfish reproduction stress.
Okay. Thank you everybody for coming. Today I'm going to talk to you about rockfish reproduction and trying to understand the drivers that have driven historical recruitment of rockfish. Yeah. So just some key words that I'll be using today within reproduction of fish populations. There's kind of like two main ideas, and the first one is fecundity, which is the total larval release of fish, which if you stick around for the next talk, my friend Sam over here will be talking about fecundity of rockfish. But for today, I'm going to be talking about recruitment, which is the total number of those larvae that actually make it into the population. And during their recruitment phase, fish are actually planktivorous, and they kind of just float in the water column. So there's a lot of different factors that go into recruitment, and for my thesis, I have been investigating some of those factors. But first, let's talk about rockfish, because rockfish are super cool, and if you've ever eaten fish tacos or any type of fried fish, the chances of that was a rockfish is pretty high. So rockfish are really cool because they're super long-lived. There's some species that can live up to 100, 200 years. They're highly fecund, which means they have a lot of babies. Millions of babies are reproduced each year from rockfish. And they're really slow to mature, so that means that some fish species don't reach sexual maturity until they're around 20 to 30 years old. So that's really important for their conservation. They're also viviparous, which is pretty rare for fish species, and that means that they give live birth, kind of just like us, but they give birth to millions of baby fishes. They're also commercially and recreationally super important, so there's a lot of different studies that are done on them. And they also live in the California current, and that means they're subjected to a lot of variable ocean conditions. And those variable ocean conditions make their recruitment vary year to year. There's some years where there's a lot of recruitment, and then there's some years where there's almost no recruitment. And this is called a boom or bust system. And it has been studied that some of this recruitment is due to coastal upwelling, exposure to different types of source water, whether coming down from Alaska or up from Mexico, or different decadal cycles such as El Nino Southern Oscillation, the Pacific Decadal Oscillation, and other large basin movements of water. But so we're looking specifically at the California system, which is an eastern boundary current system, and you can see for most of the months in a year, it flows from north to south. And this flowing of wind and water from north to south causes seasonal upwelling. And that seasonal upwelling can cause lower temperatures of dissolved oxygen, pH, and higher productivity in the nearshore environment. And it can also, the physical act of upwelling can sometimes push pelagic larvae offshore. And seasonal upwelling just so happens to coincide with gopher rockfish reproduction and recruitment months. So it's really important to understand how this influences their recruitment. Their recruitment can also be influenced by large-scale climate patterns. So that's the Pacific Decadal Oscillation and the North Pacific Gyre Oscillation. I won't go into too much detail, but one operates on 20 to 30 years and one operates on 10 years, and basically it influences the stratification of the ocean. I'm not a physical oceanographer, so I'm not gonna go more of that. But the specific species of rockfish that I'm looking at is gopher rockfish. And I like to think of them kind of as the average Joe of rockfish. They're the most common nearshore species. They range from all the way up to Cape Mendocino down to Baja, but they're mostly common in Central California. They live at depths between 10 to 40 meters, so they're super near shore, and they have home ranges of only around 10 meters. So they don't move much. They reproduce during January to May, which is during the upwelling season, and their last stock assessment was done in 2019. And a stock assessment is kind of a tool that fisheries managers use to understand how many fish are in the population and how many can be caught both commercially and recreationally. And during their stock assessment, there was no environmental nor ecosystem considerations, and that's because there was a lack of relevant data at the population scale. And for future things, it said investigate possible drivers of biological parameters, particularly recruitment. So an objective of my thesis is to identify environmental drivers of rockfish recruitment to help with the next stock assessment. And for the methods I used for this portion, I just took the historical recruitment from the stock assessment from 1995 to 2018. And then I took large data sets from oceanographic parameters, one that resembled upwelling, one that resembled the North Pacific Gyre Oscillation, and one that resembled the Pacific Decadal Oscillation. And then I did a bunch of modeling to see which environmental driver predicted rockfish recruitment the best. And that's what this graph shows. So the black dots are rockfish recruitment, and the different lines are the different drivers of the recruitment. And when you run all of these things either individually or together, the best model that predicts rockfish recruitment is actually all three of them put together. So that's the upwelling, the Pacific Decadal Oscillation, and the North Pacific Gyre Oscillation. And you can see down in bold, the 0.56 means that it explained 50% or over 50% of the data variation, which is really strong correlation in science and a very cool finding. So because there's three different drivers, you can plot it in 3D space. And the yellow represents the highest years of rockfish recruitment. And those years were typically seen in lower upwelling, a positive decadal oscillation, and a positive North Pacific Gyre Oscillation. But what does this mean? So it basically means gopher rockfish recruit best during very relaxed upwelling and kind of warm phases in the ocean. So they don't really like to be moved offshore. They like when the physical conditions are kind of lower, and that's probably because their recruitment period is only 1 to 2 months. And yeah, another really important takeaway for this is that as rockfish go through stock assessment, and stock assessment is a very long process that costs a lot of money to pay scientists to do. So if you have tools like this one that can predict rockfish recruitment, it doesn't require a full stock assessment to be done, which is really important as there are talks of stock assessment being brought to the state level instead of the federal level. And tools like this can help state scientists manage our fisheries without having to do a full stock assessment. And yeah, that's pretty much all I had to say about that. Does anybody have any questions? we are a little bit behind time, so. Cool, awesome. Okay, everyone, next up, our talk is from Sam. He's also a student here at Moss Landing. He's a third year in the ichthyology lab and is going to be giving us a talk on rockfish reproduction.
Cool, thank you. Awesome. Ooh. Cool. Well, thanks guys for joining. Like I mentioned, or like I was introduced, my name's Sam Perello. I'm a third year master's student at Moss Landing Marine Labs. And I'm going to be giving you my talk about red hot rockfish reproduction. Now that title's a little bit of a misnomer. I'm not going to actually talk about the copulation, but kind of like what happens afterwards. So just some background, climate change is happening, right? We're putting out more greenhouse gases with higher concentrations that get sequestered into the ocean, higher carbon concentrations. This creates water that that's really hot, so it can't hold as much dissolved oxygen, and through a series of chemical processes become more acidic. If you don't believe me, you could read any of these IPCC reports, they'll fill you in, but they're pretty dense. But just generally, you know, climate change is happening, right? And impacts on marine species are pretty wide and well documented. This could be anywhere from range and migration shifts across ocean basins and across species to even decreased survivorship, and what I'm going to be talking about today, which is reproductive timing. So more about reproduction. Like I mentioned, higher temperature ocean can't hold as much dissolved oxygen. We've seen already that this has increased the energy cost for breathing for species. So that means they got to suck out all this oxygen out of the water. They're spending a lot of energy doing that. So they have less energy left over for survivorship or reproduction. We've also seen that in temperate environments, sensory function has been decreased for temperate fishes. So this is a temperate environment that we live right here. And we're seeing that with lower pH levels, these fish aren't able to sense their surroundings as well. And that leads to a lot of low survivorship. And then kind of a nuanced version of this is the combination of ocean acidification and hypoxia. This is a relatively understudied portion, but we're seeing in some preliminary results that species like Sebastius auriculatus, the brown rockfish, that eggs have been degraded. So just kind of some more background. If you've heard Massen's talk, you've heard this, but we have a really deep sea out here. And through natural processes, wind pushes water offshore. We get this really deep, cool water that has a lot of nutrients upwell to the surface. This creates phytoplankton blooms near the surface, gives us a really productive ecosystem that we have here. But these nearshore fishes are experiencing a lot of that conditions being brought up to them. So this is a map of California. So what we did was we kind of went out these last 2 years. On the left is 2023, which we had an El Nino, which is a natural decadal cycle. And then in 2024, we had a La Nina. El Nino is kind of the opposite of normal effects. We have less upwelling, a stronger countercurrent, generally higher temperatures. And in 2024, we transitioned into a La Nina. And like I said, these are natural cycles that happen over a decadal time series. And this is an intensification of normal events. So we have colder water, we have a lot more upwelling, and these maps that you're seeing, these heat maps, are temperature anomalies. So the red is really hot temperatures, and blue is really cool. And just generally you're seeing, 2024, we're seeing much less temperature anomalies, there's more nutrients, more productivity, more upwelling. And just to introduce the species I'm gonna be talking about today, I'm gonna be talking about rockfish. If you go out today afterwards and you get a fish sandwich, some fried fish, rock cod, rockfish, snapper, that's all rockfish species. We have over 70 species just off of California's coastline. If you want to read into these, you can. These slides didn't pace well, but we have gophers, rosies, and starries. And they'll have these little graphics up as we go along so you can follow along. So what did we end up doing, right? So we went out, we chartered fishing boats. We went out for both years, 2023 and 2024. We bought devices like CTDs, Niskin bottles to measure water quality. And we went out 12 times in 2023, 14 times in 2024. both years during the reproductive timing for these fish. These numbers that you're seeing on the bottom are QD values, which is an index I'm also not going to go into, but essentially red values are like higher temperatures, more El Nino, and that blue negative numbers are more La Nina. So we went out, we caught a bunch of fish, we sacrificed them, we got a bunch of variables I'm also not going to go super into right now, but essentially we cut up these fish, we dissected them, we got a lot of stuff. We also went through this very lengthy process of staging them. Rockfish put a lot of energy into reproduction. They go through this complex 7 stages of reproduction. Just know that as the stages get higher numbers, that's essentially later developed fish. We also did a bunch of fecundity analysis. We took subsamples. Just the little gophers will have anywhere between 150 to 600,000 babies in a year. So we don't want to count that many. So we'll take little subsamples and count that. What did we get from doing this, right? You know, some of these fish, these vermilions live over 70 years old. We killed these guys. Why did we do that? Well, we found out some very cool stuff. So the first thing I'm going to be talking about is reproductive investment, or a gonodosomatic index, which is GSI. So this is essentially how big their gonads are compared to the size of their body. So you see for all 3 species, and as I go through this presentation, 2023 is always going to be represented in red, that, remember, hot El Nino year. The blue is 2024, transitioning into that La Nina. And you can see all 3 species, significantly heavier gonads. They're putting a lot more energy into their reproduction. They have these big fat ovaries. But where does this energy come from? So rockfish do have mesenteric fat if they have extra energy, but they'll also put a lot of that extra energy into their livers. So you get big fatty livers. And so this index is called HSI, or hepatosomatic index. This is essentially how fat that liver is compared to the size of the body. So just like GSI, except for the liver weight. And you can see the exact opposite trend, except for stories, which are doing their own thing. But gophers and rosies, at least, we have really fat, heavy livers in 2023, and then kind of much smaller in 2024. Essentially what's going on here is I'm equating this to, when they're stressed out, they have high temperatures, they're sucking out all that oxygen out of the water. They're kind of trying to weather the storm. They're like, I'm not going to put all my energy into this reproduction, because why would I, when it's not going to be good for me? And then as soon as good conditions hit, they pull that energy from the liver, they put it towards their gonads, and so that's why we're seeing that inverse relationship. This is also another factor called weight-specific relative fecundity. So this is how many babies they're having average per the weight of the fish, so we can compare between species. So are they just having fat gonads, or are they actually having more babies? They are actually having more babies, significantly so between species. Another thing that rockfish do, they have exponentially more babies when they get bigger and fatter. It's not a linear relationship. Big fat moms will have 10 times, 20 times the amount of babies that young moms will. So this is a log transformed graph, so we can compare them linearly. But essentially, is it like, oh, are we just getting bigger fish in 2024? No, it does not matter the size of the fish. 2024 is having significantly more babies at the same size fish. So kind of cool to see. Same deal for rosies. Now, like I said, we took a bunch of water quality measurements. Do larger fish prefer colder temperatures? Kind of, but not significantly so. Do they prefer higher oxygen water? Kind of, but not significantly so. But this is what I really want to show you guys. So this is a really cool result. So this is just for gophers for right now. That El Nino year, 2023 on the top, La Nina 2024 on the bottom. Essentially what you're seeing is the proportion of fish we caught for each month and the stages of development that they are. So you can see in the top graph, that brown color is the really early stage of development. January, February, March, dominated by that early development. And then instantly in April, we're seeing, about 30% having really late stage, and then the rest are being spent. So that means that they've released their larvae already, and into May. Now, our 2024 data is kind of interesting. We have that same trend for the first 3 months, and then in April, we're still seeing 20 to 30% having stage 2, stage 3, that early developed larvae and eggs, and then that persisting even into May with stage 3s and there. So we're kind of seeing this delayed release of larvae in our La Nina year. And this is kind of concerning because rockfish fishing opened April 1st. So they're like, oh, their reproduction's done. well, not really. Over 50% of them still have eggs and larvae in their bodies when we're going out and fishing them. Rosie's kind of an interesting case. Top graph's kind of what you'd expect. As you go throughout the year, you get more and more later stages. Interesting thing in La Nina conditions. So Rosie's, given good conditions, could actually produce multiple broods throughout a year, which is insane. But as we're seeing, January, February, early stages, March, we're seeing dominated huge spikes in stage 3s and 4s, later stages, and then a dip back down in April, and then another spike in May. So essentially what I'm equating this to is we're having that first brood get produced, released in April, and then a second brood getting produced, and then releasing in June. So really cool to see that here. I'm kind of out of time, so here's some conclusions. Essentially, rockfish, do they like cold water? Do they like having oxygen? Yeah, they have higher gonads. They take energy from their livers. They have higher Fulton's K, which is a body condition value I didn't even go into. And this delayed larval release, which I thought was really cool. And then just generally, rockfish reproduction for 2024 and 2025 due to like PISCO dive surveys, great. Across the board, across species, high rockfish recruitment. I have a lot of people to thank my funding sources, my advisors. Yeah, I don't know if I have time for questions, but.
We have time for questions.
Okay, cool. You started walking up, I was nervous. But yeah, I'll take any questions you have at this time. Awesome. Thank you. What do you got?
Any theories on why the starry rockfish liver results were not?
Yeah, they were definitely ones we didn't collect as many as the gophers and rosies. So with infinite funding, I would love to go out and get more of them. But they were just a species we encountered less frequently. So I think if I had more individuals, that would be a more significant difference. Yeah. What you got?
So if we know that certain conditions can delay or bring upon the brooding and mating and breeding of these fish, then should we create rules and regulations and processes that say, oh, it's looking like this year the rockfish are maybe going to start having babies around this time. Should we hold off on rockfish season until after that?
Totally. I am one for adaptive management strategies. And yeah, I think given this, we could predict, we know when El Nino and La Nina years are coming. And I think we should be able to predict and set in rules for fishing in accordance with that. Now, something I didn't even go into here was all the species I was talking about here are demersal species. So they like to sit on the bottom, chill in the rocks. But there's a whole second clade of rockfish species that are like semi-pelagic. They swim through the water column, blues, olives, yellowtails, those guys. And actually, more preliminary results, we're seeing the exact opposite effect with them. So they're having earlier larval releases during that La Nina phase. So having something in place where we could say, okay, you could fish, but you can only fish maybe these species or deeper during this portion of the year for these years, and then maybe more like targeting these more pelagic species during years where it's a La Nina. I think that would be a better adaptive management strategy there. Cool. I think that's it. Thanks, guys. I have a bunch of other slides we're going to talk about. I'm just going to click through these. Okay.
He's going. You're not wearing your green t-shirt? Oh, I don't have one. I'm a guest. He's off the block today.
No, yo no puedo soportar todos estos cosas.
Like 10 minutes?
Okay, everyone. Next up, we have Jess. They are also a student here at Moss Landing. They're a third year in the phycology lab, and today they're going to be talking about scuba diving here.
Hi guys, I'm so happy y'all were able to join me today. Just to start off with a little bit more background, why should you even listen to me talk about scuba? I have worked for our dive program now since my first year. So I took the scientific diving class here with Diana Steller my first year, and then I started working for her the next year, and she trained me up to be a dive master, to be an instructor, and I've taken basically every dive class here that you possibly can. So I love it. I love being involved in it, and so I love to share that with you guys. So thanks for being here. So a little history about diving at Moss Landing. Does anyone know when Moss Landing was founded? Or how you can find out? Yeah.
1966.
Excellent. Yeah, great job. So yeah, founded in 1966, and 2 years later, the first dive class was taught in 68. I believe it was taught by a professor, but subsequent years were often taught by students that happened to already be certified. These pictures I have up here are some pictures from some of our blog posts about diving. So I think this is one of our old DSOs, Mike Foster. There's some kelp forest diving up there, some more experiments down there. But long story short, Moss Landing has a long history of diving and of training students up to do research underwater. We also have a diving safety officer. So the dive program looks a lot different than when it did in 1968. There used to be a lot less oversight. Now we have some amazing people that have been our leaders or our DSOs. One cool thing about these people is that they have almost entirely been phycologists. So I'm a phycologist, I study seaweed. I believe everyone but Ann Hurley on this list was a phycologist as well, which is really cool and speaks to the really cool things that you can do when you study seaweed underwater. The two people pictured up here on the left is John Heine. He was the most recent DSO before our current DSO, Diana Steller. These two have really pioneered our program to be what it is today. They've helped us with a lot of our sort of safety measures, becoming more involved in AAUS, et cetera. And yeah, they've really made our program what it is today. One of the coolest things I would say that our program does is it doesn't just train people in scientific diving, but it helps train divers to become leaders. So I'm a product of that. I was lucky enough to become an instructor during my time here with the support of the dive program. And that is something that our dive program definitely prioritizes. It's not just, putting people in the water willy-nilly. We want our divers to be able to be leaders, to be able to go out in the field, lead a research project safely, communicate effectively, et cetera, et cetera. So last year, myself and my coworker Logan, who's in the middle there, got our NAUI instructors. And we use these to help teach our scientific diving course in the fall and in the summer. Which segues me nicely into the biggest portion of this talk, which is dive classes. So we teach a number of dive classes here at Moss Landing. The introductory level is going to be the scientific diving class, so that AAUS, or American Academy of Underwater Sciences. We teach a 2-week intensive course in the summer, which requires a few more prerequisites, and then a semester-long course in the fall. Now we also have, once you've had that preliminary course, you can take the fun classes. So that's going to be subtitle ecology, which is, I'll go into more details, but it's been taught in a number of places. So it's been taught here locally in Stillwater Cove, which is in Carmel Bay, just south of here. It's also been taught down at Catalina Island. We also teach, well, I don't, but they also teach a course, Marine Ecology of the Gulf of California, which we all refer to as the Baja class. And so they go down and they spend 2 weeks on the Baja Peninsula, and they've taught that in a number of locations. Last year, they taught it on a really cool island called El Perdito in the Gulf side. There's also been taught a class that goes down to Chile. They haven't taught that in several years, but the amazing professors here do what they can to help us, learn about all kinds of different systems. So let's dive into each of them. The first one we call MS 105. That's our scientific diving class. This class is, again, a semester-long class, so we really take our time at the beginning, like teaching students how to be safe underwater before we add that science component. but we do like to give them a variety of different diving experiences. So this picture, you can't see the stacks, but we were diving in the Elkhorn Slough, which is just over there, which you have to dive on a high tide or else the visibility is like a foot in front of your face. But it can be really beautiful. There's sea lions, there's bat rays, little nudibranchs in the seagrass. Very fun. This, I believe, was taken at Hopkins Marine Station, which is Stanford's Marine Lab. That's our class from this fall. We take them there a lot because it's a great place to dive. But again, this semester is really just spent exposing students to how to be a cold water diver. A lot of them come from different places and they maybe only dove warm water before, so they've never worn 9 millimeters of neoprene on themselves before. It's very constrictive, and then you have to wear, you know, just as much weight to compensate, right? So I wear like 30 pounds on my weight belt. It's different. So we try to take our time getting them up to speed. The other course we teach for scientific diving is our summer intensive course. This tends to be for students that have been diving in California already. They have a few more tools under their belt, and we really try to expose them to science in particular and to different places around here. So we spend 1 week diving in Monterey Bay, and then we drive down to Big Sur, and we spend a week at UC Santa Cruz's reserve system, which is where this picture was taken. We get to dive in the bull kelp forests, which is cool. Bull kelp actually usually has more of a northern range, but we see it down south there. So that's me, Audrey, Sally, some of the students from last year. And we also got an amazing opportunity. You can't tell here, but this was at the Monterey Bay Aquarium. So we got to dive in the giant kelp tank. So that was really magical. And they're very particular about their kelp. They take such care of their kelp that they grow in that tank. So we made sure all of our divers knew how to be neutrally buoyant before we let them go there. But the summer intensive is a lot of fun. It's a lot of work, but it's really great for students that are coming in the fall and they know they want to do a subtitle thesis and they're like, I want to get trained, I'm ready to go. So that one's a lot of fun. The next course I wanted to share with you is our subtitle ecology course. This is what I was telling you guys about earlier. This is Stillwater Cove down in Pebble Beach. Look at that view, so good. So that we're teaching that course this semester actually, and I'm the TA for it. I gotta hurry up, okay. Yeah, so the students get to go diving weekly as long as the weather's good. They come up with independent projects, they ask questions, we run experiments, and they end up writing a paper about it the next week. But it's a lot of heavy lifting. We have been having to take out 3 inflatable boats into the field. We carry them down the stairs, across the beach, launch them, go diving, bring them back, carry them back up the stairs. So we're very strong by the end of the semester. And I was looking on our blog and I found this picture of subtitle class in 1978. And yeah, divers have too much stuff, long story short. But it's a great opportunity for students to become independent in the field. And here's some more pictures from us this semester. That other class I was telling you about was the Baja class. So we went out last year and conducted this class on Isla El Pardito, very small island in the Gulf. These classes are great because they expose us to different systems, right? Like we dive in the kelp forests mostly here, which are stunning, but it is really cool to be able to see things like corals and estuaries, and also meet new scientists and new folks down there. And we also build lifelong friendships. We brought a lot of gear down there, so we had to lift a lot more heavy stuff as well. And I think I'm almost out of time, but I just wanted to talk about the importance of field studies generally. It can be super confidence building for people to go out into the field and say like, yeah, I can get something done out here. And it can make people feel more connected to their work and more like they belong. So it's super important. That's one of the great things about Moss Landing is that we prioritize that. And so I feel super grateful to be a part of a program that wants that to happen. And again, yeah, it builds community. It helps people know how to exist in a community because you have a common goal. So yeah, if you can get involved, please do. And with that, I thank you. And I'll take any questions as well if you have. Yeah.
Is like the scientific dive course the requirement of like already having a school certification, or like how does that part work?
Yeah, for the fall one, we do ask that you have your open water and ideally 10 to 12 logged dives in cold water. If you don't, we sometimes are able to work things out, if you really want to do it, and like we can have conversations about that. But yeah, that's the goal. Yeah, yeah.
Are you still wearing your 9mm down in Baja?
Oh no. No, I think it was like 70-ish degrees. So I wore a 2-piece 5mm that was a little bit warm. I wish I had just worn a 1-piece 5mm, but yeah. Yeah. Okay, thank you guys so much.
I have a question about crossover.
Okay. Oh yeah, maybe. There's like some like cross-organizational like allowances, honestly. But does she like want to die?
I just wanted to ask about those summer
Oh, great. We recognize that as like a standard like certification.
It's honor.
That's so exciting. So she's gonna join Moss in the fall? Yeah. Oh, that's great. I'll probably be seeing that. Oh, of course. I'm at San Jose, yeah. But yeah, we all go together. Yeah. Yeah.
On the summer intensive course,
Yeah.
can you sign up just if you're not a student in the campus system?
Yes. Yeah, I forgot to say that. It's called Open University, but I think that's how people enroll.
Okay.
And there's like a course fee, but yeah, you don't have to be a graduate student or even a college student.
My son goes to UCSD, he's graduating this year.
Oh, cool.
We've built for a long time.
That's
so
awesome.
he has his master's.
I should have put our email up there. Do you want our email?
Yeah,
'Cause
if I
then
could.
he could, do you have something to write on?
Well, this, but I was
Yeah.
gonna return it in, but should I turn it back in?
You should, but maybe.
Oh, here we go.
Oh, yeah?
On that
Okay,
side? Yeah,
thank
that's
you.
great. Because I know he would love to do that.
Yeah.
The best thing we found about diving, the more we do, was if you had a purpose to be out, it
Yes.
was so much better.
So,
Even
yeah.
like the kelp restoration and all this stuff we did.
That's awesome.
So,
Yeah, no,
okay,
that would be he'd be the perfect candidate for our class, I feel like.
And it's a 2-week summer course.
It's 2 weeks summers. It's usually the first 2 weeks in August, but we
Okay.
haven't announced the dates yet. But if he just emails us and says like, hey, this is my name, these are my certifications, I would love to take your course.
Okay, MLML-diving-admin@sjsu.edu.
Exactly.
Okay, and your name is?
I'm Jess.
Jess, great, Jess.
It's
Thank
nice
you.
to meet
Yeah, I'm
you.
Derek. This is Danielle.
Hi, Danielle.
She's been wanting to get her.
You won't let me.
Dive?
Yeah.
No. Well, because I wanted to get it the full cert, but now they've lowered it to her age. She's 14. So
Oh,
now
okay.
she's ready.
That's awesome.
But I was worried about the weight.
That makes sense, yeah. Well,
We
you
do
should
it
totally
now.
get out there.
Yeah, no, she's ready.
Cool. Maybe
done
I'll
it a
see you
few
at
times
Breakwater
for
sometime.
class. Yes, absolutely.
That's great.
Thank you very much.
Yeah, nice to meet you guys. Did you guys have questions for me? Yeah, I haven't been diving with the whales. I think that would be a little scary, but I have seen sharks in the water. The one that I see the most is the super big shark. It looks like, if you were to draw a shark, that's what it would look like. And it was like 5 feet long. We also see leopard sharks a lot in shallow water. And then if you dive further south, like in like Catalina or Santa Barbara area, they see like 7-gill sharks a lot. So yeah, you can definitely see that. Yeah. But I haven't seen a great white, and I hope I never do.
If you see it, that is a nice encounter.
Yeah, I want to see it from the surface, not underwater. I want to see it on a boat bigger than exactly. Cool. Nice to meet you guys.
Thank you.
Yeah.
I was just like curious to know more about like the kids. And then I said, I will keep right he's a little part of that, and he's been like watching animals, and also raising animals. As far as those curious about what you guys do. So essentially, if like maybe some
I'm sure she's interested in that. Yeah, most of the work we do is involved for like thesis projects. A lot of the theses that dive are biology, so like there again are a lot of selection dives. I would say we also do like for the aquaculture, right? Like a lot of roaches, fish, that kind of thing. But yeah, it just depends on what research is and what the diversion is. So we have up to water. But majority of the diving we do here is in the complex, or right out there in the canyon head. Yep. But yeah, I mean, that if you could give me your, or I could give you my contact, we would love to, like,
like most of our stuff is like animal feeds, like cleaning, like stuff like that. So they get like experiences, like if they're interested in like we like they have to get a whole food for all of the animals and like that stuff. So people are interested in like lots of maternity field. So
should you give me your like contacts, or maybe I'll write on my phone.
I can even give you our ESS actually.
Oh, perfect. Who is that?
Nico Cowell, so Niko, Cowell, Kowell. Yes, his email should be, I think it's n.cowell@a.com.
Okay. Cool. And your name?
Emma?
Emma.
I'm new to the department.
That's so awesome. It's so nice to meet you, Emma. Thanks so much.
So interesting.
We're selling
Many years ago. And I finished exactly one. I don't consider myself.
Not every single.
We're trying to see if the next speaker is coming, so give us a few more minutes. And if not, there's definitely one at 12:15 about the battery fire.
All right, we'd love to do
So I just want to make sure that everyone knows that we've got a break until 12:15, and then that's when we'll have our battery fire talk.
What is this? Sedona. Oh, yeah.
Oh my gosh. I literally use it every day. I'm like, I have my own. Like, recharging. Yeah, basically, yeah.
Mostly I was just
Yeah, likewise. Yeah. I heard they used to do this, but I wanted to like. So when someone comes in, they put on. But it's all along the end. Yes, all three. That's what I was thinking. Like, like, okay, work on your days. Oh, no, but someone did ask me to look for that. He was like, can you go find my anchor? Yeah. Yeah, compared to that. Yeah, right.
You would say something there. Always
Yeah. That's great. That's cool. Yeah, they're actually. It was gonna sing things yeah, I know. Yeah, I know. I need to take a picture. Oh, where's mine? Another card catalog. Yeah, the next one. That was the last one. Last time I was here. Oh, you know why? Because I had
I don't know what to do without you.
No, you're good. Okay, we're good. So we're live, okay? Cool.
Amazing.
Thank you.
Yeah, thank you.
All right, welcome everyone. Oh, you're doing it.
Yeah, I'm moderating today.
There you go.
Hello, everyone. Thank you. We now have our next student speaker up. This is Dominic Kyle Yippold. I am lucky enough to call him my housemate, friend, and lab associate. He's in the biological oceanography lab, and he's going to be talking about microbial diversity and his thesis method. So give it up to Dominic.
So, as part of the biological oceanography lab, one of our main things is studying microbes. So a lot of pretty small microscopic organisms that the human eye can't see. Usually what they used to do is through microscopes and through cell counts, but my thesis revolves around using DNA to look at these microbes. So this is kind of like a very super simplified version of it. Think of it as like you're going into a grocery store. Everything has their own barcodes. You want to find out what's in the water at a certain temp, a certain season, but you don't want to see the item itself. And that's what microbes is kind of like. It's really hard to see and find them in the water. You kind of have to filter them out. But my research is I'm using just only a small fragment of their DNA. I don't need to see that specific microbe to figure out who's in there. So as you can see, it changes, not all of them are the same, but everyone has a specific barcode. And yeah, it's kind of, and then this is kind of like a bit more complex version of it. So we want to know who's in the water. Everyone sheds DNA in some form or another, and microbes are one of those. And so it's kind of like in different spots. My research looks into Elkhorn Slough and basically how it changes on a spatial and temporal scale. I'm collecting data for a year and I'm trying to figure out who's in the water. No one's done it before, no one really knows, and why should we? Because microbes are pretty much all at the base of most of our food webs. It's such a very important part of any ecosystem. So it's kind of, we sample the water, we filter it out. The filtering separates the microbes, depending on their size, you can kind of figure out what the mesh is, and it depends on who you're targeting. And then the fun stuff, we do extraction, you get their DNA, you do some library preparations, and this is where it's kind of cool because new technology has come around where it's each organism on Earth has some sort of everyone has ribosomes. So a lot of organisms have ribosomes, and each of them have very hypervariable regions that code for their species. You can kind of figure out who's there, and using some bioinformatics and some reference database, you can find out who's in the water without even seeing it. And it just kind of gets, you identify your species, you identify what you have in your shopping cart just with your barcodes, and then you do a little bit of data analysis. So my research is looking at how they change throughout a year, and also there's water samples and seeing how they change, not water samples, sorry, water parameters, seeing how they change pH, DO, nutrients, so a little bit there. So yeah, my research site, Elkhorn Slough, if you don't know, in 1947, this used to be a very freshwater dominant, ecosystem, but, we decided to dig through it and create Moss Landing Harbor, and now we have a very salty estuary that is continuing to erode throughout the years, and it's becoming saltier. You can see it's kind of circled in. So this is kind of just preliminary data. I'm using data from the Elkhorn Slough Research Reserve, Research Reserve, and they've been collecting data for the last 40 years, just typical water quality and data of different sites. There's 30 sites that they kind of monitor. I think I have 28 here. But as you can see, there are higher averages in some of the sites. So if you can see the sites on the far right, those are usually sites around Morrow, by the Salinas River, around agriculture, a lot of agriculture heavy places. You can see it kind of divides the way that the two places are different. some PCAs here and there, but this is just kind of like the work I've done so far. If you have questions, I can answer it after this. But, overall, like, why should you all care about these? Like, microbes, they're so small, why should I? They're very important in our ecosystem. They drive a lot of biogeochemical processes, photosynthesis, and then just goes up the food web. Yeah, so with a lot of the ways that the environment is changing, it's very important for us to kind of see how, the base of these food webs change and how diverse they are, or how not, that's not a proper word, but how, how they change throughout the cycle. And one of the things that I do like about this research is no one's done it before in Elkhorn Slough, and it will serve as a baseline, because once we figure out who's in the water column, we can kind of do future studies of how it's changed. Maybe, we have more agricultural inputs in the future. Maybe, we want to see like, oh, what did this look like before this happened? And this is a really nice thing. So as everyone knows, research costs money. And the one thing that I like to talk about to my other graduate students and other friends is there's a lot of initiatives out there that are funded already that do a lot of these monitoring efforts. And one of the nice thing is ESNR, or Elkhorn Slough National Estuarine Research Reserve, has been doing this for the last 40 years. So to save myself on cost and, because I'm a graduate student, I have to, hustle for money. One of the things I did was reach out to them and say, oh, you guys need volunteers. I like the data you're collecting. They're doing nutrients, they're doing pH, they're doing all these things. And they said, yeah, we need volunteers to help us collect. And I'm like, cool, can I collect some samples while I'm there too? So I'm collecting DNA samples, expanding what they're already been measuring. So I can, it's kind of like a two birds, one stone. You scratch my back, I scratch your back. We scratch backs basically. But it's nice because, it reduces a lot of the costs. Like I have to do chlorophyll analysis, I have to do nutrient analysis, but some other lab is already doing it for me, and I'm just kind of expanding on what they're doing. So there's like this, collaboration between us, and it's very efficient. Like I would recommend that for any potential graduate students or anyone that's doing research, is to just find out what's happening in your local ecosystem and seeing how your research can benefit, or you could benefit their research. And it helps time as well. But yeah, some funding sources. I've kept this talk very short because I wanted to get some questions and see how people, if there's any questions about this. But one of the major things that's happening now is I'll finish my data collection this May. So I will have a whole year of just microbes filtered out in the freezer. We're gonna go, we're gonna go sequence them and see who's in there, which is very exciting because one of the issues with estuary water is it's very turbid. A lot of the things that I'm doing isn't really done in estuaries because it's just so mucky that the filter's clogged. So I'm excited to see what kind of data we get from this. Yeah, so long cost sequencing. Here's some of the references I've referred. And does anyone have any questions? Yes.
I was just wondering from your research in general regarding estuaries, how do the microbes in the water affect the food chain and what kind of animals?
So with most estuaries, estuaries are very productive areas where there's a lot of organic matter, and so I feel like you get a very biodiverse estuary with the amount of microbes you have. And in turn, I haven't really done a lot of research of how they're impacting their ecosystem, but one of them is one of the things they are usually the base of that area. One species dominates a certain section of the estuary. I think it's mostly diatoms and dinoflagellates, at the lower reaches, and then the upper reaches is mostly cryptophytes during a certain time. And it changes throughout the seasons because there is freshwater turbidity. There's so many conditions that can change what a dominant phytoplankton species or dominant species have, they benefit from. Yes?
One is, I guess, about how many different microbes, a couple questions. One is about how many different microbes are
you're detecting and you're trying to find barcodes for, as an example. The second one is with the database that you're connecting, you're associated with, you're looking up how many barcodes don't yet have identified microbes associated with them, or you'd have your sample, but you wouldn't be able to find a matching.
Yeah, so those ones are like your database is your study is just good as your database. So most of the things that we're finding has to be something that's already been found in the past. I mean, not in the past, but that's in a reference database. So you can't really go into let's say your supermarket with a new barcode if there's nothing connected to it. So this database.
You'd have a sequence, but you wouldn't have anything that would match. So
Yeah.
You would extract.
Yeah, that's something I'm trying to see how I'm gonna go over that in the future, because the thing is, if it's not, it doesn't get identified if there's nothing in the database. It's a new species, but there's thousands of species of just the same thing, different versions of bacteria. I hope that answers your question, or
Yeah.
yeah. Sorry, I'm trying to.
I guess the different generations of DNA sequencing, right? What particular type do you use?
So for now, I think we're focusing on 18S and 16S. So the ribosome has very very hypervariable regions, and it's really up to the thing is you use different primers to to amplify these certain target areas, and we're trying to see if we can get everyone in a universal primer, because the thing is, it might be an issue if one primer is only selecting for dinoflagellates, or they have different, they have a higher affinity for different species. So that's what we're still trying to figure out, because it's something I'm, this summer I'm gonna hopefully address when I'm doing my research. Yes?
1 minute.
Oh, 1 minute, sorry.
Just letting you know, that was all the time we had for questions. So give Dominic a round of applause. Great talk. Thank you so much, Dominic.
Thank you.
All right. I am so excited to introduce our next speaker. She has been a huge help for this open house event. A lovely fellow SVO. We have Ava Bessiker, and she's going to be talking about Galapagos coral. So Ava, give a little bit about yourself and let us know about your research. Hi, everybody. Hi, my name is Ava. I'm a current second-year student here at the master's program at Moss Landing Marine Labs. I'm enrolled through CSUMB. I'm also a TA there. And yeah, I'm from North Carolina, so I did my undergrad in marine science in North Carolina State University. And I'm here to talk to you a little bit more about my master's research on Galapagos coral restoration. So you may be wondering, why corals? Most of you are probably familiar with coral reefs. Corals provide many ecosystem services. They're the most biodiverse life system in the ocean, and they hold the greatest economic value per unit area for humans. They enhance biodiversity. They support livelihoods of not just people on the coast, but also inward as well. They also protect coastlines from storms, tsunamis, and waves like that. Additionally, however, climate change threatens the extinction of many species. So corals are predicted to go extinct by 2050, and due to these climate-induced warming. And as you can see here, by 2050, temperatures are expected to increase at least 2 degrees Celsius. And now for us, that is not super dramatic, but for super sensitive species like corals, that is like boiling alive, essentially. So, oohs and ahs, yes, this is concerning. And one event that is triggered and causes an inductment or an enhancement is El Nino. And so during a normal year, El Nino only affects things in the Pacific Ocean. So during a normal El Nino year, these equatorial winds gather more on the western tropical Pacific here, around Australia and Indo-Pacific. But during an El Nino year, these winds cease on the equator, and so it causes warm water to go over off of South America and eastern tropical Pacific here.
Most notably here in this region that I talk about a little bit later, but keep that as an Easter egg. And so essentially, all of the corals that are found here in this circle, they're increased by temperatures of above 2 degrees Celsius or more, which is like boiling to them. So it can cause normal, healthy corals to bleach or expel their algae, their symbiotic algae that lives within them. And when they bleached, corals can rebound from bleaching, but essentially it means that there's a high % chance that they are going to die later on. So, an important trait that corals need to have is thermal tolerance. Thermal tolerance is additionally called temperature tolerance. And so, during the 2023 and 2024 El Nino event that occurred the last cycle, it caused mass global bleaching. You probably heard about bleaching in Florida and the Caribbean, but it also affects corals in the Eastern Tropical Pacific. So corals must adapt and show resilience to this stress. In some ways that they do that is through their physiology. So we found that genotype and through analyzing their genetics, some corals are genetically adapted to be more resilient to temperature stress. Additionally, those symbionts that I mentioned earlier, that's the symbiotic algae that lives within a coral. So coral are both animals that have plants living symbiotically with them. And those symbionts can determine their amount of thermal tolerance that they have. So for my research, I study in the Galapagos, and you may be wondering why the Galapagos. You've probably heard of Charles Darwin and the research that he did on finches, the marine iguanas that live there, megafauna like sharks and whale sharks, but there are corals there. And so the Galapagos has had a very extensive history of repeat bleaching. In 1983 and 1984, the El Nino event wiped out almost all the corals there, leaving just 10% remaining today. There have been La Nina events and preceding bleaching events that have wiped out nearly all the corals there. And so this is extremely important because corals found in the Galapagos represent populations adapted to extremely harsh conditions. So like I mentioned, that El Nino causes the corals to heat up, in addition to many La Nina events, and also there's many converging currents that occur there, currents that come from both the south, the west, the east, and bringing both warm and cold waters. Additionally, they also experience upwelling like we do here, bringing in lots of nutrients, but also ceasing their nutrients as well. So when you couple really long-lived species that also have very slow growth like corals with these extremely harsh conditions, it causes them to have a really long recovery over time. So if they're constantly being hit with these constantly changing conditions, heating events, cooling events, it causes them to have a long recovery over time. And so like I mentioned, the corals that are found in the Galapagos are adapted to these extreme conditions. So for my thesis, my research question is, what factors drive coral thermal tolerance and survival under heat stress in the Galapagos specifically? And I utilize a nursery to do that. So my study site is the Galapagos Reef Revival Nursery. It was founded in 2022. It was the very first coral nursery site of its size and kind in the Galapagos. It is composed of 3 main species, Passalopera, which is a branching coral, so ones that have lots of different branches coming off of it, and Porites and Pavona, which are massive corals. They kind of look like rocks. If you've ever seen a brain coral or a boulder coral, they kind of look like that. And yeah, so my 2 study sites are here. The nursery is located here in the green circle, and Bahia and Calera are outplanting sites from the nursery. And so what I mean by an outplanting site is the nursery looks like this. It is composed of coral trees and ropes here. And so these are what those branching corals that I previously mentioned grow on. And then here in the middle, we have an outplanting site. And so basically what we do is when these corals grow big enough, we cut them off of the ropes and we outplant them with underwater cement at these outplanting sites. And then we also have table nurseries here, and these are what those massive corals that I mentioned grow on, the ones that look like rocks. So last year, I was lucky enough to go back to the Galapagos. So I went in January and July of 2024, and I did a bunch of different field collections. This is here with me with a camera where I take photos of corals, and then additionally, that's what I'm doing here. So for my methods, I do what's called photogrammetry, and all photogrammetry is you take photos using a GoPro, so you walk in kind of a lawnmower pattern, you walk back and forth in the rows that the corals are outplanting in. And I want to have some sort of overlap, because then I'm able to take those 2D photos, input them into a computer software, and it stitches those photos together kind of like a quilt. And so basically it's finding those common pixels between the overlapping photos, and it's stitching them together to make one large 3D image. So this is kind of an example of the outplanting site. You can kind of see the dots in the middle. Those are all the different corals. So these points, the pixels, are also called point clouds. So points, pixels, synonymous, pixel cloud as well. And I take those point clouds, I input them into another software. From there, I'm able to measure coral growth. So I mentioned that I went in January and July of 2024, so a 6-month difference. So I take the 3D model of a coral from January, I take the July one, I put it on top of each other, and I'm able to measure the distance in growth from the January to the July time point that way. Additionally, I also do some genetic sampling. So we extract the coral fragments via scuba. So we go both on the nursery and the outplanting site, take some regular clippers, clip a fragment off. We bring it up to the surface and onto the land. And the fragments, you can kind of see my dirty fingernails, and you can kind of see how big the coral fragment is compared to my fingernail. And we also put it in these tubes on land. And then the team ships them all the way back here. So the corals had to go from Galapagos to mainland Ecuador, from Ecuador to Miami, and Miami finally back to here where we analyze them in the lab. So I do various genetic lab work tests on them. I extract the DNA. I also look at those symbionts that I mentioned to look at the level of thermotolerance. And lastly, I just wanted to mention that my research is also kind of like a living lab. So by utilizing a nursery site and utilizing the Galapagos, it is an amazing place to study these species in such a dynamic environment, in a location that is constantly changing with these harsh conditions and with a species with less than 10% remaining today. So it creates a great location to study these species. Additionally, it supports coral reef recovery and protection in the Galapagos. It's the first ever nursery that I mentioned, so it's a great place to see what works and what doesn't for corals in the Galapagos as well. And lastly, this is the first ever project that's being done on this nursery as well, so it creates a nice database for corals' survival, growth, and reproduction in the area as well. And that is all I wanted to leave some time for questions, so thank you very much.
What are the known consequences of having no more coral on the planet?
So I can really only talk for the Galapagos, but I would say overall on the planet, corals make up entire ecosystems. So fishes are composed of their many invertebrate species, and so that's a lot of livelihoods are based off of, especially fisheries. So if we take out the fish, or if we take out the coral, the fish have nowhere to live, and so they have nothing to feed on, they have no habitat, they have no refuge from predators, so if we take that away, that decreases the fish population. Things that eat fish, like us or other predators, that decreases their population. So it could create a whole chain event of decreases in species over time, not just biodiversity, but densities as well. Any other questions? Yeah.
How have the Galapagos Islands seen success with the research?
So since it is a very new project, we've also tried to get many kids involved as well. So we like to take kids out on tours of the nursery. Many of the kids that live in the Galapagos have never seen coral before. They've gone swimming, but they don't own goggles or they don't own snorkel gear. So it's been really honestly rewarding to like take kids out there and actually show them the work that we're doing and be like, this is what we're doing on your islands and this is how you can get involved in bringing back conservation too. Yeah.
Is there a way to volunteer with you guys at the Galapagos Islands?
Yeah, if you're actually going to visit and you wanted to reach out to the nursery, they're always taking volunteers. I think both education and then I think you can do a training too to go out with the corals as well. Yeah. Yeah, if you're interested, talk to me too. Yeah.
How is the nursery site chosen?
That's a really good question, yeah. So basically, I don't want to go back, it'll mess up the whole presentation, but it's in a really shallow site that's really close to where the boats go out. And it was chosen based off of the level of the substrate. So the substrate's pretty level. There are other corals that are there. So they're basically taking corals from a nearby site and implementing them in a nearby site that has also shown success. So part of my project is also determining which site is better. So one has more turbidity, the water's less clear, there's a lot more wave action. And so determining what actually works for the corals and what doesn't too.
Yeah, is there a study on the pH of the water? Is that changing, affecting it at all?
In the Galapagos or in the world? Not currently. So if you're interested in doing a research project, I haven't heard anything about anything in the Galapagos, but ocean acidification is definitely affecting corals. I would say it's probably mitigated less in the Galapagos as well because of all those different currents that are converging too.
All right, do you have any more questions today? No? All right, give Eva another round of applause.
And we'll give you some time to filter out. You got a lot of time to. Good. I'll just let you change it when you're ready. Okay. Cool. Actually, we'll just keep it up so that people know what they're doing. Yes, I believe they should show up right here, and you have a clicker right here.
Oh, thank you.
And so, are you, huh? Don't be nervous. I practiced since a year. No. Okay. So at around 7 to at the 8-minute mark, I'm gonna give you. I'm just gonna put up my hand. So that means you'll know that you have like 2 to 4 more minutes.
Yeah.
And I'm just okay.
It's probably gonna be like.
Okay, perfect. So, and then I'll allow like 3 to 5 minutes for questions. And so the next person we're gonna try. Fillers? Yeah.
I hope they put the right version.
The what?
Right?
The what?
The right version. Yeah, that's right.
Okay.
Because I sent it this morning again. I'm like, yeah, can you please change this?
And then I'm gonna end at 1:43 to have people have time to filter it out.
Oh, I see. Because otherwise they're all down, but.
All right, hi everyone. Thank you for coming to Open House. Next up, we have a talk from Vicki about emperor penguin research. So give it up for Vicki.
Thank you so much. Hi everyone, my name is Vicki Uwe, and I'm from the Vertebrate Ecology Lab. So from the Galapagos to Antarctic, now I'm going to talk a little bit about some of the research that we do on emperor penguins. So specifically, I'll talk about where you can find them and then what do they do in an annual cycle. So basically their life cycle, and then I will share briefly on the kind of research that we do on emperor penguins in the Vertebrate Ecology Lab. And finally, I will briefly touch upon my research on their foraging ecology. So emperor penguins are found along the coast of the Antarctic continent. And if we zoom into Antarctica, you'll see red dots along the coastline over there, and each of these dots represent an emperor penguin colony that we know of. So up till now, we know of about 66 emperor penguin colonies that are currently existing. But for today, all the research that I'll be talking about will be mainly focused on that region, which is the Ross Sea region, because that is predicted to be one of the last remaining strongholds as climate change continues. And also, for my research at least, it's mainly focused in that region, especially, actually, Cape Crozier, which is right here. So now before I get into the kind of research that we do over there, I want to bring attention to 2 specific life stages that are really important for them, which are mainly molting and breeding. And I'll do that by sharing with you, walking through their life cycle. And I'll start from the very beginning. So early in the year, emperor penguins, they will be feeding in the ocean. And then about mid-January to February, they will go on ice and molt for about a month. So molting means that they are shedding all of their old feathers and then growing new feathers simultaneously. So during this time when they molt, they don't actually feed because they can't get into the ocean because they have lost all of their waterproof feathers. So they have to fast throughout that time. But because, as you can imagine, if they're growing new feathers and you're not eating, they actually lose a lot of weight during that time. So they lose about 45% of their body mass. And because if you need to grow tissues which require protein, so they lose about 50% of their body proteins during that period. So after they molt, they're very hungry, they're starving, so they go back into the water, they start feeding, and then they will migrate to their breeding ground, which is again on ice. And so the breeding ground is where they will mate and breed. And because, as you can imagine, if you go so far in on ice, like you march 60 to about 100 miles in, yeah, so that is kind of far, and you can't frequently go back into the water to feed. So throughout the time when they're on ice, they are also fasting during that period as well. So in about June, the female will lay an egg, and then she will carefully transfer that egg to the male for the male to incubate. So at that point, the female will be fasting for about 1.5 months, and her duty is done. She's like, I've spent all my energy, I'm going back into the water and I'll be feeding. So that's what she will do. And then the male will have to continue to fast, and he will incubate the egg until it hatches, and he will then wait for the female to come back to take over that chick guarding duty before he is relieved to go into the ocean to feed. So towards the end of his breeding fast, he would have fasted for about like 4 months, and he'll lose about 40% of his body weight and only 16% of body proteins, because he's not really actively growing any tissue at that time. So after that, the male and female, they will take turns to go to the water and feed, and then come back and feed the chicks. They'll do that about 6 to 7 times. And towards the end of that chick rearing period, the chicks, they've grown larger now, so they require more food to grow. And so the parents will have to feed a lot more frequently, or more intensively, or forage on maybe better quality food to sustain that demand. And when the breeding season is complete, so the chicks are big enough now, they're good to go, and the parents will then return into the ocean, and they will feed, and then they will migrate to the molting ground, and the cycle continues. So because they fast for so long during this molting and breeding season, so that makes the time that they use to feed in the ocean like really, really important. But although we know relatively much about what they do on ice because we can observe them, so what they do in the ocean when they're out of sight remains more of a mystery, right? So we know that they feed out there, but there are a lot that we don't know about. So in our lab, we have a lot of projects that basically try to figure out this mystery. So basically, where do they go during this time? Like, what do they do, and what they eat during this period? And specifically, using GPS data and habitat modeling, Martin, which is a postdoc in our lab, he's looking at where do they go to eat after they molt, and are there any environmental features that are associated with these regions. And then Katie, which is another postdoc in our lab, is also looking at the same thing, but during the chick rearing season. So where do they go during the chick rearing season? And then are there any environmental features that are associated with those regions? And then our previous student, which is Parker, he looked at whether, when they're out there to feed, whether they have any individual foraging strategies, and if they do, are these foraging strategies associated with any intrinsic traits, such as like body size, for example. So once we know where they travel to during these periods, we can use accelerometry and also depth data to figure out where along those foraging tracks are they actually diving down and catching their prey. So are they actually feeding? And that is Taylor's project, another student in our lab. So now that we know where they go during this time, where do they actually dive and feed, we also want to know what do they eat exactly. Right, so for my project, I'm using DNA and stable isotope data to figure out what they eat during this late chick rearing period. So when the chicks need a lot of energy to grow, and also during the pre-molt period, which is the time where they have to store up a lot of energy so that they can molt for that 1 month period where they lose a lot of weight. And I want to see whether their diets also differ between males and females. And to determine their diet, previous studies have relied on collecting and visually analyzing their vomit. So basically what you do is you pump water into them and get them to vomit or regurgitate, and then you try to ID the food remains to try to figure out what kind of prey they consumed. And from those kind of studies, we know that they broadly feed on like fish, squid, and krill. But as you can imagine, this process is quite invasive, and you might really only be able to ID the prey species with like heart remains, because obviously they're digested, right?
So instead, my thesis is focused on using non-invasively collected feces or guano to try to figure out what they eat during the late chick rearing stage. Now, when emperor penguins return to feed their chicks during the late chick rearing period, they also poop. So we can collect those poop and see what you can find in them, right? And instead of trying to visually ID their prey items, I'm analyzing the DNA to see what kind of prey items are found in them, right? So all species have DNA, and some regions of the DNA, they're unique enough that they can act like a barcode. So like how you go to grocery stores and you scan the barcode and you can tell the item. So you can do the same. You can scan these DNA barcodes and tell what kind of species are found within the feces. And another goal of my study is to try to figure out what they eat before they molt using stable isotope analysis of their feathers. Why? So because these emperor penguins, they're feeding in the ocean during the pre-molt period, we can't really collect their poop. So how can I tell what they eat? Now, we can use something called stable isotopes. Imagine we're all made up of atoms, and for simplicity, let's just imagine that we're made up of dark and light shade of blue and red marbles. So when the penguin eats a prey, they'll break down the prey's marbles and build their tissues with the prey's marbles. So if we look at the marbles that make up the feather and compare them to the marbles of different prey items, we can use models to predict how much of each prey the penguin most likely consume. So what you do is you get the feather, you process them, you put them into tins and send them out for stable isotope analysis, and you get back these stable isotope values. And because different prey items, they have different kind of stable isotope values, so you can use models to kind of predict, how much of each prey they most likely ate. So this is, just an overview that I'll give about my research, and I won't talk much into that, but if you want to know more, you're more than welcome to ask me. But before I end, I just want to briefly talk about why all these research are really important. So they're really important because by knowing where they go during this time, what they do, and what they eat during this feeding period when it is really crucial for them to store up all these energy, we can better understand how they may be impacted by any sort of environmental changes. And we need to really better understand them so that we can come up with more effective conservation strategies to protect them for future generations to come. Thank you.
I saw the movie March of the Penguins, and if anyone hasn't seen it, they really should. It was spectacular about
I
their whole
agree.
lives.
Yeah, I agree.
Has anyone done the research to figure out why they go 60 to 100 miles away from food and then have to go back to feed? It just seems like a very illogical way to hard life.
So I guess they've evolved to do that, and they go back to the same breeding colony, or breeding area that they were from. And so that's what we found that they do. But we do have another expert, like the penguin expert over here. So she is my PI.
I will say that colony in March of the Penguins is kind of unique in how far it is. That 60 to 80 kilometers is.
She just said it was 60 to 80 on that.
Yeah, but
it differs.
I'm sure it is not. That is what it is. Different colonies, that is how far they may walk.
Mm-hmm.
But every colony is different in how close they are. But the reason they would do something like that and why they have these different distances is that they go there in April to lay their egg, and then in December the chicks fledge. And by December the ice is starting to melt and break. So they need to go somewhere where the ice is going to be good until December. And so when they arrive in April, that might mean they have to walk 60 kilometers. But by December, they may only have to walk 5 to 10. And that's because the ice is not stable year round. So when they arrive at a colony, they might have to walk. But they also, that's when they're fasting, like she talked about that. So they don't have to do that 60 to 80 all the time.
Yeah.
They have to do it a couple times. And then by the time their chicks are smaller, the ice is breaking out. And it's a much, like where I work sometimes, it's like a kilometer to the ice edge. So every colony is different, and it changes.
Yes.
How do you match the right poop to the right penguin?
To the right penguin, we don't. So you know that So what they do is they watch the penguin poop, so you know that it's from different penguins, and also you collect them far enough so that you don't want to repeat the same penguin, but you don't actually need to know which exactly that poop, which penguin that poop belongs to, because we know that it's different individuals, and we know that it definitely came from an individual, and so yeah, all you need to do is have enough replicates, and then you'll be able to figure out for the population, what they most likely consume during that period. Yes.
Can DNA tag feces to animals?
Yes, they can. So like
but that's really expensive, isn't it? Using DNA to know the DNA of an animal
and know the individual?
From its species.
Yes. Well, molecular work is kind of expensive, but relatively, it doesn't differ that much. So if I want to be able to figure out, you mean the species that the feces come from, or the individual?
Individual.
Yeah, you can tell individual. So I can use microsatellites, for example, to be able to ID specific individual. And differentiate and say, for example, is this sample and the other sample, are they from the same individual? I can tell by using microsatellites. And really, you only need to get primers. It's the same process, but it's
but you
just
have to
different
get a sample
primers.
from the individual to get the DNA.
No, you can use feces. You can use just their poop to do that.
But I mean, can you also tag the individual with its DNA?
Oh, yes, definitely.
I've got a question about bones, and if you can take DNA from a bone and you have another bone that's 10 miles away.
Oh, you want to match them? Yeah, sure. You can do both and then see if they're the same.
Where
to
can
tell.
I do that?
Sorry?
Where can I have that done?
Where can you have that done? Maybe you need to get a student or someone to help do that. But there are also, I believe, there are places where they offer those kind of services as well. There are companies that offer services like that. So you can get those done.
I'm trying to find out.
Yeah, but yeah, you got to pay for it, definitely. It's not going to be for free.
Well, or get it funded by someone.
Funded by someone, well, you got to get funding for that. I don't think anyone would just maybe if they're interested enough and they can collaborate and that can be part of their samples, if they're working on certain project like that and that can be part of their sample, they might want to do that. I don't know, but what kind of stuff are you talking?
Whale bones.
Whale bones. I'm not working on whales right now, but maybe someone.
Well, it's the DNA thing is what I've been looking for.
Yeah.
I don't know. Two sets of whale bones that are 10 miles apart.
Yeah, I don't know if anyone is working on whale DNA right now. Sarah, do you know anyone working on whale DNA right now? I don't know, or maybe Gita would know.
People are doing, I don't know, people do genetic modifications. I don't know about doing it with bones, but it is pretty common, if they tag a whale, they'll often take a biopsy because then you can know if you're studying the same animal again and again. So people do genetics on whales.
Yeah. You definitely can use one to do that, I know. But I don't know a specific person that I can refer you to, unfortunately.
Okay, well, it's kind of off yourself.
Yeah. So, I don't know a specific person.
We are out of time for questions. Let's give another round of applause to Vicky. So the protocol is around the 8-minute mark. I'm just going to hold up my hand and show you that you have about 2 more minutes until you should probably break for questions. Yeah, and then maybe at the 10-minute mark when you should be stopping, I'll just come much closer to you. Yeah. So.
Try to keep it nice and.
Yeah. And I'll be able to cut people off for questions, so you don't have to worry about that.
Okay, perfect. We'll have a little slide at the end.
Cool. Let's go out.
Oops, sorry.
You all set?
Yep. Okay. Just good to go.
Well, thank you everybody for continuing to hear our talks today. Next we have Ryan Rogers talking about the biodiversity of undersea meadows. So let's give a round of applause.
Hi everybody, I am really excited today to talk to you guys about seagrass meadows and how they're not kelp forests. As much as I love kelp forests and the work that they do, all the time I'm talking to people and I tell them I study seagrasses and I love them for all the great things they do. And they're like, oh yeah, I know seagrass. It's like out there at, Santa Cruz. I'm like, no, that's kelp. So hopefully by the end of this presentation, you guys will know the difference between seagrass and kelp. And a little bit about myself. I am a second-year PhD student. I'm actually not here at Moss Landing, but I'm here for outreach with a community partner. I work at Hopkins Marine Station of Stanford in Monterey. So I am based on there, I live down there, my advisor's there, and we do research on intertidal ecology. So I'm going to tell you guys a little bit about our seagrass work. So we know, sorry, sad, but contemporary biodiversity loss threatens a lot of the ecosystem services that we like to, associate with healthy ecosystems. And then when we couple our loss of biodiversity with our rapid increase in human population, we can see that we are sort of stressing our natural systems. And so this map here is a map of sites that have been listed as important in quotes. I don't know how they decided important, but they decided aggregate importance to local country level optimization. And you can see that a lot of the sites are on land, but even more of them are in the water. And I'm here to talk to you guys about some of those water sites, which include our intertidal seagrasses. So first, big difference here, algae on the left, seagrasses on the right. I don't have any hate against kelp and algae, but they have comparatively simple body structures to our plants that we like to think of and value. So algae have a single blade, they have a stipe, and they have a holdfast. These are pretty simple structures. They're unicellular organisms that grow clonally, and we're familiar with the macroscopic forms, so the really big guys, the large algae that make these beautiful stands of kelp forests. Macrocystis is the giant kelp, and Nereocystis, the bulk kelp, is another one. Our seagrasses, are different in that they have more specialized structures or more akin to terrestrial plants like their ancestors. So you see that they have roots, they've got rhizomes, they've got shoots, they've got flowers, they've got seeds. So they're much more like your terrestrial plant than your algae are. And algae are actually eukaryotic protists. So they're a bit more closely related to animals even than they are to land plants. They just are superficially similar because they have the same sort of conditions. They evolved this sort of photosynthetic lifestyle. And so this part becomes really important because when we value these ecosystem types, one of the main reasons we value kelp forests is because they are really productive, right? So there's a lot of growth of kelp, there's a lot of fish and animals that live in the kelp forest, and there's a lot of sequestration of carbon into the deep ocean. But the kelp are actually not attached to their sediment, right? They, if you see here, are just attached to the rocks, right? So they're not actually taking that carbon into their bodies and shunting it into the rock, right? They are producing more kelp, and then that kelp gets ripped off and shunted into the deep ocean, and that is the kelp carbon pump. Seagrasses do it a little differently, and they actually directly put it into the sediment, where it's most biologically inert form, per se. And so what's really cool is that seagrasses can actually store way more carbon in the soil than kelp forests ever will, because of that simple difference in their biology, right? The fact that they have these advanced structures, these rhizomes and these roots descendant of their terrestrial origins. So we're going to talk a little bit about why we should care about seagrasses. They're widely distributed foundation species and ecosystem engineers that support diverse biological communities across temperate and tropical latitudes. So another ding against kelp. Again, I feel like I hate kelp. I love kelp. Kelp is primarily temperate, right? And so it exists only in our temperate latitudes. Seagrasses are really diverse. There are 15 species globally, and they cover tropical and temperate latitudes, right? So whereas our kelp is just temperate, we've got seagrasses in New Zealand, we've got seagrasses in California, we've got seagrasses in Hawaii, right? They're everywhere. We know of a few, these are a few species here. Zostera marina, anyone know the common name for that one? Just shout it out.
Eelgrass?
Eelgrass, exactly. So that's our eelgrass. It's common, it's the one species that grows intertidally here in California. The other one is the Philospatix, the scalary, the surf grass. It's more common on the intertidal and the tide pools. And then I have two comparative species from Palau, our Inhalus acaroides, the tape grass, and Thalassia, the turtle grass. I don't really love the common names because for me, they don't really have that much value, like turtle grass, which one, you know? Like, but it's the Thalassia because it's the preferred food of sea turtles. So, why does biodiversity matter? Any guesses? You just like shout some stuff out.
It's the entire basis of the food chain.
Yeah, food chains, those are important.
Creates more resistance to change. In the
Yeah, that's the big one, right? The insurance hypothesis. This idea that if we have a store of diverse methods of living and methods of movement and response, that will be buffered from these variations in conditions. And so I have it here pretty simply defined as the variety of life on Earth encompassing all living organisms. And we value biodiversity because it is directly underpinning a lot of these things that we value out of our ecosystems. So provisioning like food, fresh water, fiber, biomass, all these things. We think about farming kelp, right, as like a potential fuel source in the future. Cultural, right, for some habitats are just culturally important, like the kelp forest has a lot of cultural relevance here in California, right, more so than the seagrass beds do. But if you go to the Indo-Pacific there, you'll actually see that there is this sort of inverse relationship, and there's a lot of cultural importance on seagrass meadows and the fish that they support. Seagrass ecosystems are some of the most productive vegetative habitats on Earth, not to brag. And if we look, this is this map here of the carbon storage. We can actually see, one, kelp nowhere to be seen, right, because they're not actually storing that carbon into the sediment, right? But two, that they are third to some other terrestrial plants, salt marshes and mangroves in our nearshore coastal habitats for storing carbon and not just turning it into biomass, but actually getting it into the soils, right? So look at how little of the seagrass is put in the biomass compared to the soil, right? And the biomass is still considered biologically active carbon, so it's still directly there in that cycle, and so it's not actually removed from the carbon cycle. So if we're interested in removing carbon from the atmosphere, those bottom three habitats that are going to get it directly into the soil are sort of our way to go. And we see that 83% of global carbon actually cycles through our nearshore coastal ecosystems. And those systems only cover about 2% of the Earth's surface, right? So they have an outsized role in our world's carbon cycling, supporting global fisheries provision, and ameliorating stressful conditions near shore. So they're super cool. About 50% of the sediment carbon in the world is stored in these nearshore sediments associated with submerged aquatic vegetation, so seagrasses, marshes, and mangroves. And so that's yet another reason that these critical habitats are, despite being overlooked, crucial to our climate adaptation policies. And there's a lot of complex interactions that support seagrass growth. I just look at sort of how variations in the biologic community, this box in the middle, it says biodiversity underneath, how that affects our ecosystem goods, services, and properties, and then feedbacks, right, between our management, abiotic conditions, and large global changes. So, just to give you guys some examples of what this looks like in practice, there's a few levels of biodiversity. We can obviously think about counting individuals, right? So 1 seal, 2 seals, 3 seals. We can think about counting species, right? So fur seals versus harbor seals versus sea lions. We can think about guilds, right? So if I only cared about pinnipeds, then it wouldn't matter to me if they were sea lions or harbor seals, right? They're all pinnipeds. I'm just counting pinnipeds, and now I'm going to count mollusks, right? So there are different levels, and obviously you can see that at different levels of specificity, you might have different actual metrics of biodiversity. So, oh God. Oh yeah, so, oh wait. Oh gosh. I think I was here, right? What are some levels? Yeah, okay. Okay, yeah, so the smallest level that I like focus on is abibiotic organisms, or things that grow on the blades of seagrass themselves. And so these can be about 30% of the above ground biomass in the meadow. They can be about 50% of the primary production in the seagrass bed, and they can be upwards of 38% of the nitrogen fixation capacity in a seagrass bed. And they're really diverse. So we've got the rhodophyta, the red algae, the phaeophyta, the browns, our greens, and some animals, the anthozoans. You know what that anthozoan is? An anemone, yeah, good job, he's on fire. And so here's some that I took from some blades at Elkhorn Slough, put them under a microscope, blew them up. And you guys are kind of like, what's in these pictures? But this one at the top here is a bryozoan, so colonial animal, right? So animal, green algae there at the top, the chlorophyta. and you see question marks, right? Like, because I don't know what I'm doing, so I don't actually know what some of this stuff is, and we'll have to use molecular tools to really get at some of that distinction. But seagrass-associated community members like the megafauna are diverse, and they vary in size from small amphipods to larger macrofauna like these crabs that we associate with our eelgrass beds up north. Sea otters here at Elkhorn Slough love the eelgrass. They love to hang out in it. It has all their favorite prey. Then we get lots of fish and mega grazers like sea turtles and dugongs in the tropics, right? So these large organisms that are associated with seagrasses, they eat the rhizomes and the roots of these productive vegetative habitats. Again, the specialized structures, right? The roots, right? Kelp do not have roots, right? So things are not eating the roots of kelp, right? They're just eating the holdfast, the stipes, the urchins are, right? But for these things, they are eating the roots, the rhizomes, and that is directly allowing that carbon to get into the soil. So how do I measure? How do we measure seagrass biodiversity? So I try to get, okay. So we can use a combination of visual ID, we can do scans and samples, so you saw some of those earlier in the presentation. We use molecular ID, I think we were talking a little bit earlier about it with the penguins, but there's environmental DNA, or the idea that living organisms leave parts of themselves in the environment where they are, and that we can take that, blend it up, give it an enzyme, and then magnify regions of the genome for the animals that were there. And then I use this third type, functional groups. So I remember talking about diversity types and how I could count individuals, I could count species, I could count populations. I like to count functional groups because these are broad, they're not species specific, but they still have relative meaning for ecological thinking, I guess. So I think about calcifiers as a group of algae. So again, this isn't species, right? This is just something that it does. So there's the functional aspect. Nitrifiers, there's some common names for the CCA is really common, is a common calcifier. Diatoms, cyanobacteria, green algae that photosynthesize and they directly compete with the plant. And our nitrifying brown algae, which come from this one unique group of brown algae. And so what I could do is I can maybe take a site like Elkhorn Slough with all of this eelgrass in the LiDAR survey here, and I could pick points along Elkhorn Slough, sample a shoot of eelgrass, and get a composition for what is on that seagrass at that point. And then what I can do is I can make comparisons from, let's say, we know that tidal flow is really restricted in the upper parts of the slough compared to our tidally flushed lower parts of the slough, then we can see how do those changes in abiotic conditions lead to variations in the biological community. And then are they meaningful for functional outcomes for the groups of organisms? And so here's a comparative work. We can also put out indirect sampling, so video and camera traps. And so these are 6 of my sites from Palau, where we put out these camera traps after we do our surveys, and we capture about 52 minutes of data about sort of how the seagrass beds are, what sort of animals are there. And these are normally videos, but they didn't embed in the coming over to this presentation format. But there's really cool animals that'll pop up all over this video. So we had some interns over the summer their job was to sort of sit through all of our hours and hours of footage and mark down every fish to the species that they could, or like shark that they saw, or ray that they saw. And they saw a lot of really cool stuff. So even like in our short little period of time that we're out there, because we only lived this out there for about 52 minutes, an hour or so. So incorporating species traits into biodiversity monitoring supports comparisons across ecosystem types. So I was mentioning how I can make comparisons at the site level. I can also make comparisons at the biogeographic realm level, although I would probably need a lot of samples and sites, right? But one of the ideas behind a lot of the work that I'm interested in is, do high species diversity sort of systems like tropical seagrass beds have different functional responses to stressors? Right? And one of the ways we would test that is by coming up with our assessments of functional diversity and community composition for something like our monospecific seagrass bed here at Elkhorn Slough. And then we would compare it to something like our fieldwork in Palau, these tropical multi-species seagrass beds, and using our species-blind categorization system, functional traits, right? Remember, I don't actually care what species of algae it is, I just care that it is a nitrifier, or that it is producing a calcium carbonate skeleton, or that it is photosynthetic. And those things allow me to characterize these 2 different seagrass beds in a similar way and make comparisons without, being like, that species is not here. There's no, throw your hands up. Cool, so to wrap up, Earth's biodiversity is changing rapidly. These changes, oh gosh. These changes can have significant consequences on ecosystems and the services that they provide, and measuring and monitoring biodiversity will be key to assessing ecosystem condition now and into the future. So that's all I've got for you guys today, but I heard there's a question period, or maybe, yeah?
We have time for one question.
Okay.
It's really basic, but why, I didn't really understand, you talked about kelp, sorry to mention kelp.
No, it's okay. I'll go back.
Well, why do grasses grow instead of kelp? I mean, like, why did that ecosystem develop, do you know?
Well, so I don't want to say this and like disparage them, but I like to think that if you're a plant and like things were going well for you on land, that you'd be living on land, right? So I think these guys were some of the weaker individuals in the intertidal habitats where they existed and got really resistant to saltwater conditions and started to live in the water. So like all of our seagrasses have terrestrial ancestors, right? So that suggests that if we go back some time, the thing we would recognize as a seagrass would be growing on land somewhere. Probably like an intertidal place that's periodically flooded, right? Because they get that sort of like conditioning. But the idea is that maybe they were being outcompeted by another grass or plant in their habitat and got pushed into this subtidal, eventually intertidal into subtidal sort of habitat range that we see them in now. But because of their terrestrial roots, they have these really cool superpowers that the kelp do not, right? So kelp are of the sea, by the sea. Seagrasses, like all life, came out of the ocean, did a little land thing, and then decided, I'm gonna go back, and went back into the sea.
Fossil records in that?
Plants are really, really hard to see in the fossil record. That's like the con there. Sorry, yeah. So, yeah.
All right, thank you very much. One more. Next up,
Hey, thanks so much. Yeah, of course.
we have Bruno.
Press forward right here. So we have Bruno Matoli presenting about you, me, and fish pee.
Yeah. Hi, everyone. Thank you so much for being here. Thanks for supporting Moss Landing Marine Labs here at Open House. I hope you're having a great time. I'm glad the sun came out. So I'm here to talk to you about fish pee. So let's get the one question out of the way. Wait a minute, fish pee? So the answer to that is sort of. So they might not pee the way that you and I pee, but rather fish are constantly releasing waste from their gills. So saltwater fish specifically, because they live in salt water, have to conserve water within their bodies. Water tends to flow across membranes to areas of higher concentration. So the higher salt in the ocean makes all the water within the fish body want to leave and go into the ocean. So in order to prevent that, to minimize that, they actually don't store urine in their bladder and rather excrete ammonium through their gills right over there. So the majority of the excretion is actually through the gills. Only about 5% of waste leaves through the urine. And that waste, when we talk about waste in this case, we're looking at mostly the nitrogen, which comes in the form of ammonium and urea. But why do we care? Why do we want to talk about fish pee? It turns out that nitrogen, the building block that comprises ammonia and urea, is one of the most important nutrients in the ocean for building life. And giant kelp is a really important foundation species. We've heard a lot about that today, I'm sure. But it needs a constant supply of nitrogen to grow. And that can come either from nitrate or ammonium. Nitrate is the preferred resource though, and nitrate comes from upwelling, which you may have also heard about today in the talks. And so nutrient-rich water comes up from the deep. That nitrate is brought up, that is not usually common in the surface, and the kelp can absorb that. However, the kelp grows all year round, and its growth rates are much higher than what we would expect to see if we just looked at how much nitrate is being brought up from the ocean. So because of that, people are theorizing that the pee from animals, and in this case specifically fish, could be a sort of fertilizer for kelp and for algae. And so this all kind of started with Richard Bray. I call him the first pioneer of fish pee. And back in the 80s, he was working with a team out of CSU Long Beach, and they went to Santa Catalina Island, just like 40, 45 minutes outside of LA. And what they did was a handful of things. First, they measured ammonium in the ocean just to see what the levels would look like within crevices that were occupied by these fish called blacksmith, or crevices that didn't have fish in it. Then they captured those blackfish, and in the lab, they put them in incubation chambers and tried to measure how much ammonium and how much pee they were releasing into the water and get a rate for that. And then finally, they combined the fish and the kelp in tanks and experiments to see if that would affect the kelp growth or if there was some sort of relationship between the two. So what did they find? First things first, bigger the fish, the more they're gonna pee. And that's a pretty standard relationship in physiology across all animals. So wasn't really too shocking, but of course in science we love to confirm things with numbers. Secondly, they had kelp and fish in certain tanks, and when they compared that to tanks with just fish in them, there was less ammonium in the tanks with the kelp. So what that means is that the fish were producing ammonium, and in the tank with just the fish, the ammonium was going up. But if the kelp was in the tank as well, the ammonium stayed kind of constant, or even decreased sometimes. And that wasn't quite realistic because the level of ammonium was a lot higher than what you would expect to see in nature due to the small size of the tank. But thankfully they thought of that. And they took tanks with just kelp, and they went ahead and manually added ammonium to match the levels that you would see in the wild. And they did that with one control tank with no algae and one tank with algae. And as you can see from this graph, the ammonium level on the y-axis and time on the bottom axis, no algae in the tank, pretty consistent amount of ammonium, right? But once you have an algae in the tank, that level of ammonium goes down. So we can presume that the ammonium is being used by the kelp as a nutrient. And then finally, the shelters where they extracted water samples from that had blackfish in them had higher ammonium levels than the shelters that didn't have fish in them. So what we can glean from that is that there's a pretty significant input from these fish that we can notice. And that brings us to pioneer number 2, our very own June Shrestha. She was an ichthyology student here from 2016, I think, to about 2020. And her thesis project involved measuring the excretion rates for a huge range of kelp forest fishes. So for local fish, she went out diving and fishing down at Stillwater Cove and at Point Lobos, 2 very popular spots for research here at Moss Landing. And then she also got to go down to Catalina Island where Richard Bray did his work and measure some excretion rates by some more southern specific species like that California sheephead up there. And when I say many fish, I mean many, many, many fish. We're looking at a total of about 460 individuals. And they measured fish across all these different families, and the goal was to try to get a sense of how much ammonium was being put into the ocean at a time. So she took that data about the pee rate and combined them with fish counts provided by monitoring organizations such as PISCO and kind of extrapolated. We know that this fish pees this much when it's this size, and then we've seen this many fish of this size at this time. Multiply those together and you get an idea of how much pee is being put out there. And the sites are a little biased as there are a lot of sites within MPAs, but if you look at the sites that are outside of the MPAs, you do maybe see a little bit that in MPAs there's a lot more pee going on. And that could be because you have more fish in the MPAs. And so that brings us to me. I saw June's research and I said, wow, that was pretty cool. But like we all know, the oceans are changing. And unfortunately they're getting warmer, they're getting more acidic. And so if this is actually an important input, I wanted to know, how is that going to change as the ocean changes? Is this something that we can maybe rely on to help buffer? And so I collected these juvenile blue rockfish. They live on the bottom, kind of near the kelp holdfast, and they're really abundant, and they were easy enough to catch. I got to give credit to my advisor, because he was an animal out there. I have 15 different tanks at 5 different temperatures, and I'm tracking how much they're growing, eating, and consuming oxygen across all the different temperatures. In terms of the excretion, here's a little picture of my setup. I've got individual tanks for each fish, plus 1 background tank, so that I can measure if there's any ammonium changes going on from microbes. And I have them in there for 30 minutes. I take 3 water samples, 1 before I put the fish in, 1 after the fish has been in there for 15 minutes, and then a 30-minute sample. And from that, I can establish a rate of change of ammonium in the tank, and that'll tell me how much is being put in by the fish. So here's a little bit of a close-up. You can see my little water sample tubes, and we got fish in each tank lined with a plastic bag to ensure that there's no contamination from previous samples. And finally, I get to run my samples on this lovely beast of a machine. It's called a Latchet Flow Injection Analyzer. I put my samples here, These are all the necessary reagents that mix in with my samples and help change the color of the sample to look for a certain analyte or chemical. In my case, I'm looking for ammonium, urea, and phosphate. That all runs through this bird's nest of wires through a light sensor and then back out into the waste container, and I get a sweet little number that tells me how much ammonium is in the water. And that's pretty much what I'm doing. I don't have any results for you yet, but I'm hoping to update you next year. Any questions? I got a question in the back, yes.
Have you taken into consideration the effects of current in the different areas that maybe disperse the ammonia?
Oh, definitely. I have not personally taken that into account, but that's definitely something that needs to be considered. Not only because the currents cause the concentrations to change very quickly, but ammonium is actually the most readily available form of nitrogen. So in order to ingest it and use it as energy, it requires a lot less chemical manipulation versus nitrate. The bonds between the nitrogen and the oxygen are a lot stronger, and the individual needs to consume more energy in order to use that. So when ammonium is out in the water, it's not out for very long because things are using it. So it gives us an idea of how much is being produced, but like most models, it's hard to really get a realistic number for any specific place in time. Yes?
What's your feeding timing protocol? Because is there a relationship between feeding timing and the amount of fish excretion?
Absolutely. And as soon as they eat, because they're always kind of excreting, those processes are just happening at all times. They excrete a lot more as soon as they eat, and then it drops off after about 8 hours to kind of like a baseline. The inspiration for this study was June's thesis, where she pulled the fish out of the water and was trying to get kind of a natural view, not something where we just fed them what's the most they can pee or what's their baseline of peeing. So because of that, I went for kind of like a middle approach where the day before I feed them a little bit extra, but I feed them late in the evening so that we can capture kind of like a realistic snapshot of how much they would be peeing at any given time. But it's hard to control for like exactly matching what June did because of that. So that's what I do, just to try to get like a realistic medium, but that is affected by the type of question that you're asking. Yeah.
Is there a correlation between depth and oxygen level, and that taking into account the flow of nitrates and what was the other one, ammonia?
Yeah, ammonium and urea. That oxygen definitely changes with depth. Colder water can hold on to more oxygen than warmer water. However, a lot of the oxygen in the ocean is caused by production from photosynthesis. So you get a lot more oxygen up near the top, and as you go down, there's a lot less oxygen.
Follow-up.
Yep.
So in the more shallower regions where the most warming is happening, does that mean that there's more readily available oxygen, which kind of leads to more like prone to algal blooms and stuff like that?
Yes. Well, algal blooms aren't really, because algal blooms, they take in CO2 and they produce oxygen.
Right.
And I think also, maybe confused, I'm not sure. The oxygen is produced by photosynthesis, and it escapes a lot more in the surface. So there's kind of like that relationship. But I'm sorry, I'm not really seeing the connection between like the oxygen and the nitrogen. No, it's okay. I want to answer this.
Thinking, okay, so basically there's more oxygen at the surface, like you're saying. So I'm wondering, so like there's more warming going on there, and as a result of that, would there be less fish there to pee into that water?
Oh, I see.
Because of the loss of their habitat, because it just is too darn hot.
Yeah, that's very, very real. And one thing that happens as the ocean warms near the surface is you see species kind of getting deeper and deeper. And so if that were to happen, this whole idea of it benefiting the kelp, as you're saying, is no longer, because the kelp can only grow to so deep.
At a certain depth.
Yep. So I think that's what you mean.
Yeah.
Cool, hope that answered your question. Looks like I'm out of time, but thank you everybody for joining.
Okay, you get 1 minute?
Yeah.
And yeah.
Yeah, second year, you get to the open house coordinator, I think theology. I actually in the fish lab, but I don't know anything about Oh, Coastal Ecology Lab.
Yeah, it's a little more, so we're gonna check it.
I think it ends at 5, but like the sunlight.
It's good, I'm in a group chat.
I know. This is just
Yeah, it's not like 20 minutes or having conversations. It's just like, I do this.
Hi y'all. So we're gonna get started with our next talk. Our next speaker here is Haley Bregoff. She is a second year here at Moss Landing. She's in the Coastal Ecology Lab over at CSU-MB and at the Ichthyology Lab here at Moss Landing. And this lovely person was also our lead coordinator for this whole event at Open House. So please give her a strong round of applause.
Hi everyone. As Sebastian said, I'm Haley Bregoff, and I'm going to be talking to you about kelp forests of Monterey Bay because, of course, look at our location. How can I not? So, in case you're not familiar with the area, that yellow star is where you currently are. You are at Moss Landing Marine Laboratories in Moss Landing, California. I have a map here describing the distribution of kelp forests along the Central California coastline. So for kelp to grow, we need cold, nutrient-rich waters in a shallow depth for sunlight to reach the kelp, because if you remember, kelps are plants, surprisingly enough. And this map is depicting our most common and dominant kelp, which is Macrocystis pyrifera, which is our giant kelp species. So as you can see here, I'm just going to highlight that this box here is Monterey, and this is where my research personally happens and where most of the research in the area comes from. So there are two dominant canopy kelp forming species in Central California. On the left, we have giant kelp, and on the right, we have bull kelp. So giant kelp actually dominates here in Central California because it's a perennial kelp. What that means is it's here year-round. So as an evergreen tree has those bright green pines all year, giant kelp is standing tall and proud all year long, where bull kelp is an annual kelp. So once a year it detaches to reproduce, so it doesn't do as well here in Monterey Bay because giant kelp just outcompetes it. So there's very few bull kelp forests here. The main one that I am familiar with is at Stillwater Cove in Carmel, and that's because that side of the peninsula is higher exposure, which bull kelp thrives because of that high intensity wave action, which allows it to have the opportunity to compete for space, which forms these beautiful bull kelp forests. So what are kelp forests? Here we have an individual. So first was that survey shot, and here we have an individual kelp. So the holdfast is the anchor of this kelp and is really the foundation of all kelp forests, and there's lots of organisms that lives in the cracks and crevices of the plants themselves. We have the stipes, which are essentially the stem of kelps, and that allows for the blades to reach the surface where there's sunlight so they can photosynthesize. Here is an up-close image of the blades and pnematocysts of the kelp. The blades are synonymous to leaves, so this is where we collect all the sunlight for the kelp to grow and gain that energy. The pnematocysts are like balloons that allow this very large, heavy blade to reach the surface and collect that sunlight to produce energy to reproduce. In case you're not familiar with the subtitle view of kelp forests, here are some photos of divers. So not only is kelp really important to all the organisms, but it's a great playground for you and me to go diving and just get a glimpse of life. Subtitle, I have a video here. If it'll play for me. Let's see here. That's okay. It was just a subtitle video of how it is to swim through a kelp, but I can go back here. Essentially what you have are these long kelps that grow these forests, and it's very dark, as you can see on the photo of the left. There's usually limited sunlight, and that's because the kelp is blocking all of that sunlight, trying to outcompete everything. But there is a level in the canopy that is from the benthic, and then there's usually lots of fish that are swimming around in the mid-level. But what I'm really here to talk about is not why kelp forests are so important to me, but why kelp forests are so important to local communities and all the organisms that call kelp forests home. So there are ample benefits of kelp forests. Not only does it remove carbon pollution, it also protects our coastline by absorbing some of that wave action, as kelps like to grow in that intertidal area where we have lots of that wave power that's absorbed, which leads to, like I said, coastal erosion of our coastlines like we see in Santa Cruz and like we're seeing in Pacific Grove. So it's also farmed for food. Giant kelp is one of the fastest growing organisms. It can grow, I had the number, but it's gone, so I don't want to say anything, but it grows up to 60 feet and it's constantly growing. And not only is it important, like I said, for food and for organisms to eat, but it's also an incredible habitat that hosts large levels of diversity. So this is what I'm most excited about, the biodiversity in Monterey Bay. So all of the organisms that are here are the various invertebrates that call kelp forest home, from sponges to tube worms, anemones, snails. We have a red abalone, egg yolk jelly, and urchin. So there's a plethora of phylums, or these taxonomic groups, that are really important to kelp forests. And I can't forget all of the friends with backbones, even though I'm biased and prefer the invertebrates. We have a variety of rockfishes and marine mammals like harbor seals, sea lions, and otters that really rely on this habitat. So what I'm really here to talk about is my interest, like I said, and that's the biodiversity in kelp forest. And so how do we understand this kelp forest community when it's subtidal, and obviously we're very limited? Our solution to that is scuba-based visual surveys, which allow us to get a census of the community structure of these kelp forests. So essentially what is done is, you can see this diver has that bright orange tape reel in its hand. That's a transect tape. And what we do is we'll tie off a transect tape to one of these stipes, and we'll go in a direction, and we're taking notes on every invertebrate, every fish, and we're counting the number of stipes that these kelp have, because it not only gives us a metric of healthy kelp, the size of kelp, but it also gives us an idea of what organisms call that kelp forest home. And I know that the photos I've shown you have been really beautiful and gorgeous, but most of the time in Monterey, this is how kelp forests look. It's low visibility. Any of the divers here understand that when you get into the water, it's not like those perfect picturesque moments like I showed earlier. This is how a lot of the diving looks. So here's just another photo of, you can see that with that low visibility, counting things that are benthic might be a little bit easier because you're able to get up close. But fish that are far away, that are swimming, it's really hard to get a taxonomic identification on those organisms. So what I aim to do with my master's thesis is improving kelp forest monitoring. And you might ask, how am I planning to do that? So, but we can see here that most of the year, all of our data, or so if this pie chart is our data of what we have on kelp forest communities, most of our data comes from the summer. Great conditions, all of the photos I'm showing you crystal clear, that's how diving is. Fall, you start to lose some of that visibility, the surge gets high, it's really hard to go diving regularly. Spring and winter, very green, very turbid, really hard to see. So how are we handling these lack of visibility? So divers are limited by visibility, air supply, and thus their taxonomic identification skills. And they're very intensive. So there has to be some type of solution for us to get more data about what lives in a kelp forest at different times of the year. And that's when I included environmental DNA into my master's thesis. And I know a lot of you are sitting here thinking, I came to a talk about kelp, what is environmental DNA? But environmental DNA is short fragments of DNA that are floating freely in the water column. So when I do DNA extraction, it's not from an organism like you or me. I'm essentially taking DNA from a rock or a drop of water. That's the best way to think of it. So how do we do this? eDNA allows us to detect multiple species from DNA extractions by using these primers or molecular tools. Each of these fingerprints you see go to a different group. Like you and I are distinguishable by our fingerprints. Each of us have genetic fingerprints that we are able to distinguish one another from each other. And we're able to do that with these really large groups. So we can see in the blue would be our 12S genetic region, which allows us to target fish. On our red is our CO1, which is cytochrome oxidase 1, a very, very famous gene region used that captures a wide range diversity of organisms. That red fingerprint, you would be able to detect it using that primer. And with 18S and 16S, you're able to detect plankton and bacterial organisms just by using those primers. And so all of these different groups of organisms can be detected from that one water sample by aligning the different primers to look at what group we're interested in. So by pairing all of these universal primers, we are able to capture multiple groups of organisms without even needing to visually see them in person. And if you remember how most of the year looks in Monterey Bay, that's an incredible improvement. We're able to get more taxonomic identification with a higher success rate and accuracy without the increased intense surveys of going out multiple times during a month to try to understand how this kelp forest community is changing. So by pairing molecular tools with our contemporary and traditional kelp forest monitoring, we're hoping that we can improve not only just baseline kelp forest ecosystem community understanding, so overall improving kelp forest ecosystem, kelp forest ecology, we can also understand policy and implementation and recovery of kelp, as if you're unaware, we've lost a huge population of kelp along the California coastline. And I'm running low on time, so this is me and my master's thesis. I went very, very high level because I wanted to highlight mostly kelp and just give a little bit of what drove me to my master's thesis and why I think it's so important and why it's so, why I'm so passionate about it. So if you want to learn more, you can email me, but I'm happy to take any questions if you have any, and I have all my photo credits and whatnot. Any questions? Oh, yeah.
This is slightly tangential, but
Yeah.
I'm just thinking about the holdfasts and their properties.
Yeah.
Seem like whatever it is that's making them so effective might be useful in building homes or structures that would withstand hurricanes or something.
Yeah.
Are you aware of any research about the properties of holdfast?
Yeah, there is actually ample research. Here at Moss Landing, Mike Graham is one of the leading researchers on giant kelp in California, and there's lots of research down at Hopkins Marine Station where I do my research. They do ecophysiology, so understanding how the physics of kelp movement impacts their growth and all the species that live in it. But you're totally right. A larger holdfast allows for more habitats for organisms that are, those small invertebrates crawling through the cracks. So larger kelp, healthier kelp equals more stability and resilience when there are large environmental stressors that come through. Yeah. Yeah.
Every time you go to the Monterey Bay Aquarium, they tell you that the kelp grows super fast.
Yeah.
What is it that enables them to do that? And the related question is, given how fast they grow, are the sea urchins decimating the kelp? So if you remove all the sea urchins, do you think you'll be able to get the kelp back?
Yeah, that's actually a great question. I'm not too familiar with the physiology because I do more of an ecosystem community-based level, so I don't have all of the information for you, but I'm sure somebody in the phycology lab who studies the reproduction could answer that in a heartbeat. But regarding to the urchins, so there's actually a project going on there was a project going on called Tanker's Reef where they were doing urchin removals to try to see if there was having an impact when you remove the urchins, if the kelps are coming back. And they found it's complicated because urchins are a natural part of the ecosystem. We need herbivores to graze that drift kelp. So those big blades break off and they'll start to smother things below. So having herbivores are good because it keeps it in balance, but you're right, removing those very barren, nothing but, urchins and no kelp, that would totally give kelp a chance to grow because they would be able to settle before the urchins graze them down before they reach adulthood. But yeah, I think it's about having a delicate balance of ensuring the native species are there, but in a more relative distribution, because we did see a huge boom and we're still not really sure what happened there with them. So lots of research is still being done about the relationship of kelp recovery and urchin recruitment. Yeah.
Do you know that, let's see, there's urchins making the perennial kelp go away, is that sort of like making it equivalent like a disturbance that then potentially the annual kelp could have a better shot at recovering or something?
Yeah. So bull kelp is also completely decimated because of the urchin booms. Like Northern California, there's a master's thesis that's dedicated to replanting kelp because they're really having a hard time coming back in Northern California because of that annual reproduction. But essentially, yeah, there has been a lot more of like reds showing up. Like when I go diving, that lower understory is just booming with so much red algae that could be the space for these larger kelp. But those smaller reds are just able to recruit really fast and grow and take up that space. So when you have like bull kelp, which reproduces that special time of the year when it's annual, and there's just that lack of space, it's really difficult here when Monterey Bay is so protected and so biodiverse that so many other organisms can thrive. And then, yeah, any other question? Oh, yeah.
Using the DNA fragments to detect the presence of different species, that's great, but that doesn't really tell you how many other species are there. Right?
You're totally right. So eDNA has become this huge biomonitoring tool for biodiversity, and what we really have a gap in knowledge is the overlap between what is it detecting that we're seeing in our visual surveys or what our visual surveys are completely missing. So I'm not using it to quantify. This is purely, I want to understand how is the community changing in seasons that we haven't even been able to get in the water to know what's there. So I am very interested in the quantitative aspect, but eDNA, the technology just is not there yet, but I'm very hopeful that we will progress and have, there's lots of RNA developments to understand the life cycle or like what stage that the organism was when it was shed, and there is quantitative methods, but for my aim of capturing as many taxa as possible, quantitative PCR would not be appropriate, so like give me an abundance.
Thank you, everyone. That concludes all of our speed talks for today. We have a trivia coming up here in a few minutes. And we have a student Q&A panel later today starting at 3:30. Thanks, y'all. They're actually right across the hall.
Is there another lecture about to happen?
That's right. We went to your whale fest. Did you make it there? You were just about ready to go on. You were waiting for the other band to finish. They went over.
And I know where she was.
Very awesome.
Hi, how are you? Another whale fest.
We were there this year too.
I keep forgetting.
So, yeah.
So she was asking me about
No, not. They're changing things since COVID. They've changed things around. They want
I
more
see,
water.
so you guys met Sharks.
Yeah,
they were
Bridge earlier.
Yeah.
I don't know.
That sees you.
I think he's-
Right,
So he kind of collabs with these.
I don't know if he's out there.
right, that's what she
Well, he
was
does
telling
think
me.
short setting on, but he's back with his parents.
Go ahead, goodbye Sebastian, thank you.
Is, I think it's in June, like June 6th. Remember that Sherry Finzer movie?
Right, the flute player, yeah.
So he's having an in his own studio house concert. And it says they're gonna provide like snacks and drinks too.
Oh.
Did you hear about that?
No, I've not heard.
It's like June 6th, something like that.
Okay, we have the Mary Ann and June
Congrats
6th.
on the, Mary Alice, congrats again on your latest Whale
Oh,
Fest.
thank you so much.
It seems very popular now.
It sure is.
Yeah.
I had no idea they had 4 boats.
Yeah, isn't this beautiful?
Can you,
too?
what is that, is that?
No,
Can you read what that
let's
was?
see. Yeah,
And
that's,
then it kind of disappeared, so I.
oh, this is the John Martin. Do you know
Okay.
Marlene? She's the widow of John.
I met Marlene
Did
Martin.
you? Yeah, she volunteers on my shift at the aquarium also.
Oh, really? Would
She's
you
a-
tell her hello?
Oh, I could do that.
An email, and for some reason she didn't respond, so I'm wondering if I have her email correct.
I see, yeah, I mean, I could send it to you or whatever,
Sure,
but.
that'd be great,
yeah,
because
Marlene.
she gave it to me, I wrote it down, I mean, she gave it to
That's
me.
a nice help, yeah.
You know, she was standing there writing on not such a great piece of paper.
Yeah, right.
So maybe I will.
You have a camera with you right now? You can get a photo of this, where is it? Oh, it's.
You guys wanna do some
down here.
trivia? Okay, we're gonna do some trivia. Can you guys give me like 2 minutes? Yeah? You guys can also sit front row.
I can't participate
Could be
in
more
trivia.
just us hanging out, huh?
I'm biased, I would know the questions.
Oh, do you know the questions? These are not reserved, these are not reserved.
Let's see,
You can take
ML.
off, I think we're good.
Martin, number 4. She's at AOL.com.
Oh, there's a romantic. She's quite a bit older.
Okay.
Yeah, I helped her edit questions, so.
Oh, did you actually?
Yeah.
She should still be there today. We're gonna go look for her. She was here at 11. I know that.
Okay.
So he's involved with that.
And then the Sheila B.
Oh, yeah, right, yeah, I just learned about the Sheila Bee. They had it here last year.
Yeah, so the Sheila B was scheduled to come to the Wales, but couldn't.
Really?
Because, it was so windy.
Oh, I
And even the boats couldn't go out. On that day.
I remember that, because we were gonna have the Sheila be there, and
So
then, yeah.
Sebastian, do you still have these, 4 boats?
Getting some programs going.
No, we have the Sheila Bee, and we have the, John Martin.
Oh, okay.
Oh, right, that's up
So
there.
is the point served, does the point served, did that go to the aquarium?
There was a big one, yeah, that was our big research boat, so this was way back in the day, this was before me, but it got sold apparently to some group in Florida. Which is kind of unfortunate, because they sold it for like 800,000, which is like, okay, that's a lot of money, but also it was bringing in like 200,000 or 300,000 a year for us in terms of profit, so it's like, that doesn't really make sense to me, but.
Wow.
We'll have to look up Michael C. June
That
6th.
is, listen, so do you, the author of this quilt is not here. Do you happen to know?
Sorry, what was that? The author of this, the Quilt Guild, I believe, is the one that made it.
Yeah, oh, right there.
And they're actually just literally like right out this hallway in the room right back here. And they have some of their quilts up actually right now too.
Yeah, that's right, that is,
Oh, Red Trumpet.
They make some beautiful things, yeah.
Oh my gosh. So how do you feel?
I'm doing okay. I'm renovating my brother's old condo. He lives in, do you know Ivy Park?
Yes.
Yeah, so my brother's in Ivy Park now, where my mother lives. She's my mother's.
How are y'all doing today? Is it really just gonna be us today? I thought there'd be more people.
So I'm renovating his old condo.
We'll have a good time.
That's been my whole world.
I can't take full credit for this trivia, by the way. I was leading the team that developed this, but it was really one of our members that like took the lead on creating this.
Okay.
Windsor,
For a year and a half.
Yeah.
So we don't like that, so all the hiking up.
Sure, yeah, totally, I'll take it. I was responsible for them, so I did approve this, I guess, so.
Oh, I know.
Let me see.
Have you been on the catamaran tour, the Elkhorn Slough?
We've been,
Yeah.
Yeah, one of the boats is like.
So before,
Like 3 or 4.
lasted, well, lasted.
That's
so is that,
We're not really.
so let's see.
Well, we got
I guess for the kayak, you can really dip into other places. But no, and plus the narration, the experts on the boat.
You know, that would be a good idea.
Yeah.
Hey, Nick, I'm thinking for June 8th.
I guess we only have 2 teams here. Yeah.
Yeah, and they call it LCAT for
Oh,
electric
the electric cat that goes on Elkhorn Slough?
Yeah.
I've been wanting to do that.
It's really fun. Very
I've
fun.
been wanting to do it for years.
Yeah.
Yeah, since kayaking, I've wanted to go on that electric boat for a long
Did you
time.
get?
And why June 8th? Is that what I got? For your birthday?
Okay, I got 1 clipboard. I was gonna force people to like somehow try and write. Okay, give me 1 second.
By the way, the quick room is open.
Okay.
Isn't that the most?
Oh, it's really neat.
I know, I'm like trying to come up with a plan. Last time I did trivia, my team was bat raised, so that's like, it's pretty good. With that, we did pretty well.
Where'd you do trivia?
Other brother, his friend, they're good. Yeah.
The last time I tried to go to their trivia, the power.
Oh no.
So I was like, oh, yes, I'm
Yeah, I tried to go, I was like, oh yeah, come this is Wednesday, and I went, and they was like, you don't have that, I was like, okay.
You're like, well, I'm gonna
I
try.
was like, well, I went last week, but guess it's not every week.
I thought it was every week.
I thought that, I clearly thought
Just
so
happened
too.
to go on the 1 week that they came. Did you grow up around here?
I grew up in Chicago.
Oh, nice. Did you go to CS?
No, I moved here to work at the
Oh,
aquarium.
wow, nice.
Yeah, I went to grad school in Seattle. I've never, I mean, I've always loved the West Coast, but I lived in California.
How do you like it?
I like it, yeah, it's been a year since I moved here, so. Yeah, it's been nice. The weather is really good here. If it was, if I lived like more inland, I think I would hate it, but no, I like it different. Yeah, I loved Seattle weather, that was great, but.
Hello. Sebastian's like, do you forget? Yeah, they're working for us in trivia. Well, should be here for like 20 minutes and then I have to go over parking. What's the prize? I like the trivia so fun. Yeah. I grew up in Sacramento area. I went to undergrad in Colorado. Graduated in the pandemic, so I moved back here and got an internship with the ecology, so.
Yeah, I graduated spring of 24.
That's good time. Really good.
Yeah, I was like, oh, there's always grad school.
Yeah, I took a break.
Just
grab
Big team.
What did you say?
Are you saying that because I'm gonna
wear it?
No, because it's his team. Oh. Interesting, okay. And because you know what I mean. Okay. I'm really for the story, it's really awesome. So, you know, my friend Quinn, my friend. Yes. So, I went to go text him this morning for the streets, and he's got like tape of little water with me. And I opened the wrong text. That's what I know before I sent it. But you know what I realized? a month ago, I sent him a message, hey, you gonna go over to honors meeting? Never does. Wednesday, I sent ex-boyfriend to him an online Sudoku game. Like one of those links they play with me on no, not Sudoku, on call out. I sent my ex-boyfriend.
Okay. Pencils, paper. Oh, you guys are ready? Oh, everyone's got one, huh? You guys, come on, sit up closer. There's like none of us over here, so.
Stay for like 10 minutes and I have to go to parking.
That's fine. That's fine. You can sit. Here, paper and pencil. Paper and pencil. You guys coming in? You guys want to do some trivia? Come on.
Yeah, sure.
Get over here. We're gonna do some trivia right now.
Yeah, sure.
Can we share some pencils? Is that all right?
Yeah, you got one.
Oh, you guys got one? Oh,
So it's not cheating that I'm whispering in your ear? I want a trivia.
Also, this will be maybe like chemical or something.
Like 1962.
Yes, people do. There's literally a chemical.
Where'd the cheese open?
Yeah, exactly.
There's the animals there.
We're in the fun little.
Oh, yeah.
Okay, we'll share to maybe.
Yeah, right now.
I say that having named one.
Okay, we're gonna go ahead and get started here. There, I hope will be some more people trickling in. Everyone's got a pencil and a paper. Do you guys all have a team name? Are we playing individuals here or teams? What's going on? So, this trivia, you guys have to follow the rules. That means this trivia is self-scored. So I need honesty from everyone in the room. No writing A, and then I give you the answer and you scribble it out and you say B. All right? Ailish, I'm looking at you.
Okay.
Here we go.
I won't cheat, I promise.
Okay. I'm going to tell you guys the answers right after we do the questions, all right? And each round has 7 questions, all right? Cool? Sound good? All right. Let's go. Round 1, local flora and fauna. Which one of these animals is a sea otter? And which one might be a different kind of otter? Don't be embarrassed if you got this one wrong. But B is our sea otter. That was too easy for y'all. You guys want to do some trivia?
Sure.
Yeah, you do. Come on. Who doesn't want to do trivia? Everyone loves trivia.
My citation is
Okay.
Are you?
Question number 2, no peeking. Rule number 2, no peeking in each other's answers. Question number 2, what is the general name of the type of plant-like plankton that generates around half of all Earth's oxygen? Something plankton. Blank plankton. Did anyone get phytoplankton? Oh! Trivia? You guys want to do some trivia? Come on. You guys want to do some trivia? I know. I'm sorry. I asked. I didn't mean to ask. All right. I got one pencil, so y'all are here. Okay. You guys kind of missed the first 2. Don't worry about it. We'll keep rolling. Okay. This one's a good one. How old was the oldest aged rockfish? How old is the oldest rockfish we know? 57 years? 139? 205 or 261? It's a pretty exact answer. That's kind of crazy. What are you thinking it is?
C.
What do you think?
D.
What do you think?
C.
Oh!
That was
205 years old?
that was
Y'all, this is crazy. I did not know this.
wild.
I didn't mention this to everyone. I mentioned this before a lot of y'all walked in, but I actually did not make these trivia slides. I am part of the committee that made these, but it was someone else's duty. So I'm actually learning just as much as y'all right now. I did not know these. So, okay. Let's keep it rolling. Question 4, true or false? Corals live in Monterey Bay. Corals, do they live in Monterey Bay? I'm thinking like Finding Nemo. Something like that. I'm seeing some head shakes. I'm seeing some head nods. Everyone put an answer already? Oh, Bruno's getting in on the action here. Bruno, you want a trivia thing? Oh. If you guys don't know, me and Bruno go to trivia every week together.
I gotta get my practice in for next week.
Okay, who said true? Raise your hands.
Oh, yes, no.
Who said false? Raise your hands. Oh! There is actually quite a few cold water corals that exist throughout the world, including in Monterey Bay.
I told you.
Deep and cold. Okay. Question 5. Which one of these algae is giant kelp? Which one of these algae is giant kelp? If I may provide a hint, one is giant kelp and one is bowl kelp. Which one's which? Which one's giant kelp? And the hint is the other one is bowl kelp. Bowl kelp? Like a bowl, like B-U-L-L. How many of you guys said A? How many said B? Sorry, it's A. That was a good guess, though. They look pretty similar, huh?
I'm thinking because one was soft and another was harder.
You playing with us?
Yeah.
Oh. You can pass it off to Carrie. Carrie will play. Okay. Oh, you don't want her answers?
She said that she's not giving me her answers.
Oh. Here, there's a paper right here.
Up in the front. I can only stay till 3:15.
That's fine. That's actually when this ends, I think. So. Okay. Okay. Question 6. Question. This is supposed to be question 6, no?
No, fine.
No. On your paper? On your paper, though, it's 6? Or, okay, we're just gonna. This is for funsies. No one put this down because I think this is a double. How many teeth does the animal have? They have teeth? They have exactly 5 teeth. They have exactly 12. They have 40 to 60. Who thinks A? No one write this down. Doesn't count. Who thinks A? Who thinks B? Who thinks C? D? This is a good one. I did not know this. I don't know enough about inverts. Good job, Bruno. Okay, that didn't count. That was just practice. Number 6. Which two of these animals are most closely related? So, which two are most closely related? Is it A and B? Is it A and C? Or is it B and C? Huh? A sea star, a sea anemone, and a person like you and me. So, one last time, which two are most closely related? A and B, A and C, B and C. Did anyone say A and C?
Oh, we were close.
You guys are close, you had the right idea, I heard it. I heard it, I was like, oh, you had the right idea. You caught on to our trick question. Okay. Ooh, this is a good one. How many hearts does an octopus have? How many hearts does an octopus have? Does everyone have an answer? Did you guess?
Yeah. I have a question.
Yeah.
What is this place like during work is?
Not this fun. Not this fun. It is a lot quieter. There is usually a class going on right in here. We set up like 4 different tables. We got some computers out. We're doing some coding, doing some stats.
Oh dang, in this classroom.
In this classroom.
Were you teacher or student?
I'm a student, but I'll take it as a compliment. How many hearts does an octopus have?
3.
Oh! Did anyone else say 3? Oh! Hey-oh. All right, y'all. We have finished round 1 Okay. Have you guys all self-graded so far? You guys have which ones are right, which ones are wrong? You guys have a total for the round? Yeah? Okay. Round 2. Thanks, y'all.
Thank you.
Wait, round 2, sorry. What is the subject? Boom. Oh, oh Lord. Moss Landing history. Question, hi you guys want to play some trivia?
Yes.
Did you grab a sheet?
Yes, I found one on the chair.
There might be
We're
some
good.
answers already on that, but that's okay.
There's not. We're
There's
good.
not, okay. Okay. Question 1. Moss Landing Marine Labs had to be rebuilt after many of our facilities were destroyed in the 6.9 magnitude Loma Prieta earthquake. What year was the earthquake? You guys remember the year of the last big earthquake? I know a lot of us weren't alive then, so I've heard about it. As old as I may look. You guys have a guess? Everyone got a guess? Can someone throw out a guess that they had? No.
89.
What is it?
89.
Did anyone else say 89? Only one other person? Oh! I'm disappointed in y'all back there. Didn't, right? Question 2. John H. Martin, notable oceanographer and former Moss Landing Marine Labs director, is known for his research on which element as a fertilizer in the ocean. And then hint, the element is a common metal.
Common metal.
Again, I did not make these trivia slides. I would never have overlaid the text like that. Just saying.
Probably if I'm being honest. I don't know what that is.
Anyone know some metals? Anyone got a guess? Someone shout one out, yeah?
Iron.
Oh! Walks right in, just crushes it. Do you want a sheet?
Yeah, sure.
Walks in, just crushes it.
Thank you.
You guys say iron? I know you guys said iron. Did you guys say iron?
Which round is this?
This is round 2. Round 2, question 2. You want a sheet?
Yes, please.
Mm-hmm. Round 2, question 3 now. Okay. We're self-grading after every question, by the way. So you write a check or an X. Question 3, round 2. Which Monterey County native and acclaimed novelist wrote The Log of the Sea of Cortez, which one class used as their textbook? You probably do know this name. It's from Monterey. Maybe think like Cannery Row. Anyone got a name they want to throw out there? Oh! Have you guys heard of John Steinbeck?
Yeah.
There we go. And fun fact, the boat that he used to I don't know what you call it, boat down, motor down, drive down?
What?
Boat person, can you tell?
Steam down.
Steam down, thank you. To Baja, California, just got refurbished and actually just left to recreate the original trip. So they are either on their way or in Baja, California already. So, anyways, moving on. Question 4. In which decade did construction begin on Moss Landing's power plants? All right, y'all, I'm not gonna lie, I did not know this one. I do not know this one. Does anyone have a decade? Decade. If you get the decade right, we'll count it. Yeah?
1950s.
Nope.
60.
Nope. Nope. No. No. No.
80.
Tens? No.
1800s.
No.
1900s.
There we go. You were really close. That was fair. That was fair. I think no one got that. We'll put a big X on everyone. It's not hurting any of y'all. 1940. All right. Moss Landing is home to a society honoring which very famous Elizabethan era author and playwright? Woo! Couldn't even tell you what that means. Elizabethan? What does Elizabethan mean?
It means from the era of
Queen Elizabeth.
There you go.
Thank you. Okay, okay.
Nobody totally cut you off, sorry.
famous author and playwright from that era. Do you have an answer, Bruno? I saw your hand shoot up.
I do.
Does everyone have an answer written down? Yeah? Okay. Shout it out, Bruno. What do you think?
William Shakespeare.
Wow. Wow.
On your way out, you can check out the Shakespeare Society of America. And if you're nice to the guy, he'll let you try on some authentic costumes. I'm not even kidding.
This is in Moss Landing?
It's in Moss Landing for some reason. The story was that when the old Globe Museum burned down in LA, I think this was in, not this recent LA fire, but another LA fire, they just didn't know what to do with all of the old relics, and they decided they weren't going to rebuild anything in LA, and one guy bought this property in Moss Landing and brought it all up here. It's been here ever since. Like most things at Moss Landing.
Very nice. Okay. Question 6. What degree do Moss Landing Marine Lab students graduate with, and in what subjects? So what degree and what subjects? Degrees might be like a high school degree, a bachelor's degree, that kind of thing. And then subjects might be like, I don't know, psychology, sociology.
One day, I will be able
Invertebrate
to,
psychology.
one day we'll be able to do that.
Invertebrate psychology. I got a question if I could talk to whales earlier today. Of course. I called them up earlier. That's why they're going crazy outside right now.
Somebody was like, can you tell someone to call it? I know a guy.
Okay. Who said Masters of Science in Marine Science?
Yeah, we did.
Yeah? Good job, everyone. Good job. Moss Landing is only a graduate school for masters thus far. PhDs might be coming.
Loading in.
Question 7, round 2, end of round 2, right here. Moss Landing Marine Lab's logo draws inspiration from this famous Japanese wood block print. What is the title of this piece of artwork?
That's crazy.
There's a Lego set of it.
Yes, there is.
Ooh.
Yeah.
Sorry. Is it still going?
That's pretty, I was like,
Two.
just keep the names. Or maybe I can.
Okay, does anyone have a guess? Oh, we got some answers then? Hold on, I'm getting back.
We'll see.
Wait, where'd my thing go? Oh, there it is. Okay. Okay, who's got a guess?
Great Wave of Kazugawa.
So close.
Did I mess up on the name?
Yes, but it's so close. Did anyone else get something like that? No. Oh my gosh. Hold on, come on, click over for me. Would you click? Let's go. Literally, I'm counting that. You get that. That counts. That counts. Does anyone have a problem with that? No, because I'm in charge right now.
Thank you.
All right. All right, that was the end of round 2. Okay. You guys have a total of points so far? You guys have been doing yes and no for everyone? Got a total so far? Okay, nice. Good job. All right, round 3, pop culture. This is you. This is you right here. You ready? You got this. You're gonna carry this forward. You guys ready for pop culture?
I guess so.
What address does Marlin find his son Nemo in the film Finding Nemo?
9?
Shucks. What's the full address? If you think about like what Dory's saying over and over and over again. Yeah, exactly. You heard it at least like 20 times in one watch, so.
Last time I watched this was like probably 4 or something.
Did you like it?
That's all right.
Has anyone got an answer here? Anyone feel confident enough to shout it out? Yeah? That is so close.
Sherman, 42 Wallaby Way, Sydney.
There we go. P. Sherman 42 Wallaby Way, Sydney. C. Sherman 42. I can't even say that. That's hard. P. Sherman 42 Wallaby Way, Sydney. Okay. Who got that? Bruno got that. Anyone else got that? There we go. There we go. Okay, question 2. What kind of shark was portrayed as the antagonist in the 1975 film Jaws. Sorry, that says Jaws.
That's his name.
What type of shark was it that was the main shark in Jaws? I feel like everyone's got an answer here, right? Did anyone say seven-gilled shark? No, because that's wrong. Everyone said great white, right? Good job. All right. Question 3. This is round 3, question 3. Fill in the blank. The 2004 disaster film The Day After Tomorrow wrongly claims that the collapse of the blank blank would set off an immediate catastrophic ice age. Oh, I corrected this. Oh, did I not resave it?
Are the spaces correct?
Yes.
They are.
Yes. Anyone got some ideas? Anyone want to shout out an idea they might have?
Ozone layer.
Close, but no.
It's the jet stream, right?
I'm gonna take jet stream too because I feel like that's the same thing. It's the Gulf Stream, yeah. Yeah, but I'll take that. I think that totally is fair. You guys get that? No? No? Sure. Question 4, round 3, question 4. What slinky pair of cartoon villains are named after the terms for cargo waste or debris found on the ocean surface? Wow, I did not know this.
Ursula's, right?
What is this? What movie is this from? Little Mermaid? Okay, okay.
I can't read your writing.
Oil and smell.
No. Oh? You know what it is? Do you guys have an answer? Do you guys want to put some? Yeah?
No, I was reading stuff.
You locked in on answers, everyone?
Yeah.
Okay. Do you wanna go?
Flotsam and Jetsam?
I think that's exactly what it is.
Oh, yeah.
So then, flotsam is the term for cargo waste, and jetsam is the term for debris found on the ocean surface, I suppose. I did not know this. This is new to me. It could be the other way around, I couldn't tell you. Question 5. All right, question 5. This is a fun one. In the 1995 movie Waterworld, the sea level has risen 25,000 feet from the melting of the polar ice caps. What would be the closest amount of sea level rise caused by the melt of the polar ice caps? Sorry, it's all getting jumbled right now. If the polar ice caps actually melted, scientists have gone out there and estimated how much the sea level would actually rise. How much do you all think that would be? A, 25 feet, B, 2,500 feet, C, 25,000 feet, or D, Waterworld got it correct.
Kevin Costner.
Who thinks A, 25 feet? Who thinks B, 2,500 feet? C, 250 feet? And D, Waterworld had it right. Kevin Costner. You got it right.
It's a great movie.
You got it right. Well, unfortunately, you did not get it right.
I knew it was
250 feet. Y'all, 250 feet's a lot. That's a lot of sea level rise. That's half of Florida. That might be more than half of Florida. I don't know. Okay. Question 6. Question 6 here. Round 3. Question 6. Who lives in pineapple under the sea? Da 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 da. Who lives in a pineapple under the sea?
SpongeBob.
SquarePants. Everyone get that right? I hope so. This is like one of my favorite shows growing up. And the movie, actually. I love the movie too, actually.
I wonder if anybody called Mickey Mouse.
Okay. Question 7. Question 7. What city is not included in the beach song Surfing USA? Hold on. All right, everyone got an answer? You want to take it away? What do you think?
Yeah, I'm guessing Manhattan.
Okay. Anyone else put Manhattan? Yeah, we got a couple other Manhattans. Oh, I see a shaking the head. What'd you put?
No, yeah.
Oh, you did? Did anyone put anything else? Oh, did anyone put anything else?
I'm judging. I'm judging.
Oh my gosh, you guys. I would have guessed this wrong too. San Diego. That's crazy. Yeah, where do they mention Manhattan? I guess they do.
Manhattan Beach, there's one in Southern California.
They don't mention Manhattan.
That's a trick question. Let's go to the person who made these slides and take it up with them. Okay, you guys, that's the end of round 3. That's the end of round 3. Do you guys all have points thus far? Yeah? Okay. We trust in everyone to grade themselves properly. Round 4, oceanography. So, not gonna lie to y'all. The person that made this was a physical oceanographer. These are going to be rough. We're gonna get through it together, okay? Okay. What is the biggest source of sediment in Monterey Bay? Runoff from rivers, crumbling sand from sea cliffs, sand swept up the canyon from offshore. What is the biggest source of sediment in Monterey Bay? Runoff, crumbling sand from the sea cliffs, or sand swept up from the canyons from offshore, upwell? Who thinks A? Who thinks B?
No, we put A. Oh, G thinks B.
Who thinks C? It is A. Runoff from the rivers. Biggest source of sediment in the Monterey Bay. Question 2. Which of the following is not a characteristic of hydrothermal vents? Is not a characteristic of hydrothermal vents. Low iron, rich biodiversity, high temperatures, absence of light, which is not a part of hydrothermal vents. Do you guys know where we find hydrothermal vents? Up near the surface or deep? So maybe not deep. Are they hot or are they cold? Who knows? They actually are both technically. There's different varieties. They are, okay, so that leaves only a couple answers here. A or B, A or B. Who said A? Who said B? It's A. They are typically high in iron, though. They also can be low temperatures though, by the way. They're not always hot. Anyways, question 3. As light descends through the water column, different colors are gradually filtered out. What color of light is lost first? So you think about a rainbow. What color in that rainbow is lost first as you get deeper and deeper in the water? You guys know this? Yeah? I actually heard a 4th or 5th year graduate student earlier today giving a talk say this wrong. So, oh yeah. Not gonna name names out here, but they should like revoke that degree. What did y'all say? Red. It's red. Good job, you guys. He said red was the deepest one, like the last one he said. I was like, that's wrong. That's wrong. That's wrong. Okay. Question 4. What is the name of the climate oscillation that occurs roughly every 4 years, bringing more precipitation than usual to California? Hint. There's a hint up there.
We couldn't do pop culture. We couldn't do this.
Everyone good? Did anyone say La Nina? Or El Nino?
Yeah, slash a boy.
Okay, question 5. Only 3 more questions to go. Where can you find the oldest water in the ocean? So where's the oldest water in the ocean? If you're thinking about like the thermohaline circulation, water goes deep down into the ocean, it gets trapped there for thousands of years and then pops back up somewhere near the surface. Is that surface area near the Antarctica, near Antarctica, near Atlantic, North Atlantic, North Pacific Ocean, or anywhere along the equator? You guys better get this right in the back, because I know we talked about this in Chemosh. Yeah. Yeah. Okay. Does anyone have a guess? Yeah.
North Pacific?
Does anyone have a different guess? A? B? Oh, we got everything.
I said D.
Oh, D? Anywhere in the equator? Okay. North Pacific Ocean. The water right out here, you guys, is like thousands of years old. It's the oldest water on the planet. Goes deep down, stays down there for 1,000 years, pops right back up, right out here.
Where's the new water coming from?
The new waters, so it's going down near Antarctica where it gets really cold, and near Greenland where it gets really cold. So it gets really dense, it gets really salty. It's like super salty water coming up the coast from the Gulf, goes up to Greenland, gets really cold, and then gets really heavy. It sinks down to the bottom.
Never see it again.
We see it right out here, 1,000 years later. Because if you think about like the water at the surface constantly like evaporating, and then precipitation and other things, right? This one is literally like, it goes down, it hasn't been touched. And it's actually really cool. There's not much mixing going on either between like the different like densities of water. So the like chemical properties that water had when it subducted down into the bottom of the ocean, it stays like that for 1,000 years, and it doesn't change. Like the salinity, the density, the coldness, it all is pretty much like trapped in there and comes back up in the North Pacific. So I'm surprised I remember all that. That was like one class of chemical oceanography last year, so I think I nailed it though, right? Yeah, let's go. Question 6, you guys, 2 more questions. What type of rock makes up most of the ocean floor? A, limestone, B, basalt, C, granite, D, feldspar? Feldspar. Feldspar. Thank you. Did I say basalt right?
You said feldspar.
I don't know what I'm saying here.
Limestone, basalt, granite, and
That's salt? Okay. Not a physical oceanographer here. Anyone got an answer? Are they a guess or an answer? Same, I would have guessed it. Did anyone say B, basalt? Did you? All right, last question here. We're only 2 minutes over. We're doing pretty good. What % of our Earth is covered by water? Round to the nearest multiple of 10. 10, 20, 30, 40, 50, 60, 70, 80, 90, 100% water. Who said 100? Yeah, good job. Anyone say 70? Okay, ayo. Okay. Put that final check mark. Put that final check mark. You guys want to total up how many points you got? Total up your points. Does everyone have a total for their points thus far? We'll give a minute. 1 minute for grading. 1 minute for changing answers. We'll scribble out on it. You got an eraser on there? Oh, I got 100! He's so smart. Someone give that man his master's. Okay. Did you guys get your totals yet? Did anyone get, I don't know, 21 or over? What is the total? 28, right? 407, so. Did anyone get above 21 points? Okay, how about above 14 points?
14.
Oh, ayo. Oh. Anyone get above 16 points? Anyone get above 18 points? You got 18? Okay, and you guys got?
19, 20.
Oh! 3rd place, 2nd place, 1st place. Except you don't get a prize, Carrie, because you work here. So. 2nd place and 1st place. Everyone get a round of applause. Great job, you guys. Great job. Good job, y'all.
Go for it. No.
Okay. Okay. Let's go get our prizes. How does that sound? And if anyone's interested, we have a student panel going on here in 10 minutes. So, thank you. Thanks for that. Thanks. Oh yeah, you guys can drop off any pens, pencils, everything just on the table right there. Thank you, thank you. Yeah, thank you. Rude. Matthias, you can get a present.
What the hell? Thank you.
Thanks y'all, great job.
That's our answers.
We didn't. Yeah.
We get a prize or not?
Yeah, come on. First place, second place, first place, second place.
We're second place. Come on.
Right.
If I get distracted, is it a free shirt?
Yeah, we just do like name, lab, and maybe like a sentence or two about our thesis.
Yeah.
How'd the trivia go?
It was good, but there was only like, I don't know how many of us were there.
I should have played two.
It's okay.
How many?
I think there's 6 of y'all. I think there's 2 from every year actually. Oh wait, well, let's just do it from the front. Ooh, can we almost do like, not a full half circle, but almost like a little like each other. Is your Elena?
Maybe half,
Huh?
maybe half.
Oh.
They're studying. Just that. A little better.
Oh, good. So we have a spotlight.
It was
Love
so
that.
dark, Sarah.
How are you guys doing?
I'm so tired, dude.
I'm ready to sit too. Take a seat.
And I did.
Is it gonna be? I hope it's not like the one from like CSUMB where there's like the same amount of people in the panel. No, there's like 6 of y'all. Okay. Yeah.
Are you not okay?
No, it was. There's already quite a few of us. So I'm the moderator. I'm just gonna like call out, like pick on people and pick questions. Oh, I probably should have preemptively like gotten some questions together. Just in case.
Yeah.
What is the most challenging thing about? So how did you guys find Los Landing? No. So how did you guys decide to go to graduate school? Ooh. I saw an ad on the newspaper. Oh Lord.
Are you on this panel?
I'm a wee bit nervous, yeah.
Why are you nervous?
Me too, because I don't want to talk.
It's fine. I think as long as you're like, hopefully like people come in. It'd be like really weird if there's more people than in the panel. There's gonna be quite. Last year there was quite a few, wasn't there?
There's a lot of like CSUMB students. All the volunteers just came in here.
Oh, true. I'm gonna go round up some volunteers.
I mean, it's possible.
I'll be right back. All right, cool.
Just sit here awkward.
Okay, cool. That sounds good. Oh
Why does it
man,
have to be awkward, Sarah?
I didn't realize the view is so good from here.
What's up?
I didn't know the view was so good from here.
It is really nice.
Like I didn't even notice. Like, oh wow, I've been like, sitting this way for like
Yeah.
the last like 2 years, and I realized, wow, it's pretty nice.
Yeah.
So when I'm doing my defense, if I start panicking, I'll just look out.
Well, what I learned in like middle school about like public speaking, if you want to like look at people's like ears, like sometimes it looks like you're looking at
That's what
their
I usually do. I like look over
Yeah.
people's heads.
And you look at their ears, so they looks like you're making eye contact, but you just make looking at their ears. Yeah.
I can't tell.
Yeah, right.
I like
That's
looking
I
at
learned it. I
people's
learned it in
foreheads,
middle
like,
school.
I like looking right here.
Wait, what?
Like if you're like
Talking
someone who
about
gets
looking
like,
at people but not making eye contact.
Oh.
So like looking at like their ear.
if you're
or,
like
their
anxious
ear.
and you can't make eye contact, like if you look at their ears, it's like a way. Like I learned it in middle school.
Interesting.
Or you can view everything else besides them.
Yeah.
Well, the thing is, it makes the conversation more personal. I feel
I
like.
was gonna say, I generally just like blank out and don't look at people's faces if I have like a thought, and then I'll come back to them later.
Oh, no, I'm like super shy. Like, so I need to like. Oh no, like even until like no, it wasn't
There's
until
like,
like.
there's not.
No, I'm super.
I swear, like when our
Me too,
cohort first started, like you were the one being like, yeah, let's all go out today.
Yeah, but that's like after like 2 years. Yeah, no, like I had to learn this. It was
My cohort said the same thing about me, and I was like, no, I'm introverted. They're like, no, you're not. You're so outgoing. And I was like, I was trying to make a support network.
Well, I'm
You
like,
don't realize that I knew I
I feel like you can still be outgoing and be an introvert. Just go hand in hand.
I mean, I'm like really
Maybe that's
awkward
how you are.
at the beginning. Like whenever I meet someone for the first time, I'm like I'll like walk away. I'm like, hello, bye.
least amount of social interaction
Yeah.
possible.
I feel like through diffusion, I like just start talking to people. Like it's like, oh yeah, I've seen you a bunch of times. Hello.
Just pretty happy you're high, I'm down.
Yeah.
I'm in Dale, and it's just us two. So awkward.
I feel like that's what happened with you guys. Like we never had a conversation. Now we're just like, oh hey Mason, what's up?
Y'all were in a class together for an entire semester.
I know. I feel like it's awkward with people until like you get like, I don't know.
right?
It's easier to talk to someone when they're like in the class with you, like that semester, you know, but then after the class ends,
Thinking
it's like
about
just awkward to talk to them. Yeah.
Thinking about it, I don't think I've ever like had an acquaintance, like hello, my name is this. Like I think we just like start talking to you as like everyone here.
You've never had like a solid introduction?
Yeah, like with everyone. I feel like it's just that's how I feel awkward about it. I'm just like, yeah, I don't want to like go. It's like, hello, my name is this. And I'm just like, hey, there. Eventually, you become friends with that person.
It's true.
And that's how I like to do it. I'm like, what's your name, Adrian? Something fun about you.
Well, the good thing is all of us have the same major, so.
So true. Isn't that kind of crazy? We're all doing like such different things.
Are you on this panel?
I am on this panel.
I didn't know, we had one seat.
I think it's almost 3:30.
We're waiting for our special because he's moderating.
Okay, when he comes back. Yeah.
He's trying to get more CSUB students.
Never had that experience.
Like that sort of stuff.
There are minorities.
It's kind of fun.
Okay. We are in the minority, the labs. No, because I said there are 3 bio people here.
Oh yeah.
And then I was like, actually, there are only 2 ecological wing people here, and it's us 2. We're in the
This is the time. This is our time.
Jellyfish are so there's an ecology question.
Perfect. All right.
I tried to recruit more. Hi, everybody. So we got a student panel that's going to start here right now. Thanks y'all for coming. We have 6 brave panelists up here that are going to talk a little bit about their experience here at Moss Landing, their experiences before, their hopes and dreams to come. So yeah, we'll start this off by everyone just introducing themselves, you know, what year, what lab, what they're studying, and then we can open it up to the group for discussion and any questions that you all might have. So yeah, Dominic, if you want to take it away.
My name is Dominic Ypel, and I am a second year at the Biological Oceanography Lab. My main study is microbial diversity using DNA, so I like it here.
I'm Sarah. I'm in the Invertebrate Ecology Lab, also a second year, and my thesis focuses on the relationship between leatherback sea turtles and jellyfish.
I'm Kelsey. I'm a third year in the Ichthyology Lab, co-advised at CSUMB, and my thesis is regarding juvenile white sharks in Monterey Bay and the environmental influences that are correlating with them here.
Hi, I'm Hannah. I am a third year and I'm part of the Biological Oceanography Lab. I study vertical algae blooms and run a bunch of benchtop temperature experiments.
My name is Heidi. I'm second year. I'm in the Geological Oceanography Lab, and I study erosion at Bay Beach in Santa Cruz. Hi everyone, my name is Emma, and I'm a first year, and I'm in Cheryl Logan's lab at CSU Monterey, and here I study the thermal tolerance of coral and coral rheology in the block.
Nice, thanks y'all. So if I may, can we just start with what brought you all here to Moss Landing? Why did you guys choose Moss Landing over potentially other graduate programs? Yeah, why are you guys here? Why are you guys here in front of us right now?
Well, so I graduated in 2020, got my bachelor's in biological sciences. I was like, oh, you know, I was just kind of like working for like about 2 years in LA, just doing, you know, what most people do, like biologists, like lake tech, going around sampling lakes. And then I realized I do like research as I dove into it more, but I felt like I didn't have the proper training or the knowledge of certain things. So I looked into graduate programs, and Moss Landing was one of the ones that I really liked because it was like a very specific thing that I've done before and wanted to pursue. And it was very local. I really wanted to stay in California and be in a new area that's not like, you know, LA or NorCal. So I guess, Yeah, no, it really drove, like, I liked the area. It's really nice. There's a lot of cool, like, labs in the area too that you can collaborate with. And I really just, like, found it, and my advisor was really chill, and she was really nice to talk to about it, and the rest is kind of history.
So I've always known that I wanted to go to grad school. It's just a matter of where I was deciding to go. So I did my undergrad up in Humboldt, and I decided I wanted to come down to Central California and kind of see how different things are here and study by Monterey Bay. And I actually was originally applying to CSUMB's grad program. But then I found a link to the MLML website on their website, so then I ended up applying here instead.
I was undeclared when I went into my bachelor's and then decided to go the bio route as at Fresno State in the Central Valley. So they didn't really have many ecological opportunities in general, let alone marine. And I started leaning this way my junior year of college, and so I knew I needed to go to grad school to kind of compensate for the lack of knowledge and skills that I didn't get when I was in undergrad. And I looked all around and finally settled on applying to Moss Landing because it includes opportunities for practical skill building, education, and autonomy and research as well. So that's how I ended up looking here.
I originally didn't know I wanted to go to grad school, so I'm from the East Coast, and I went to Colby College, a small liberal arts college, where I got to drive a pontoon boat one summer and take water samples, and so I was like, what's cooler than just being on a boat in the sun and taking samples? And I learned that's grad school, so I needed the experience to drive that boat more so and like understand what's in the water, so that's why I decided to do grad school. And I went to see if the West Coast was the best coast.
Yeah, so I also didn't know that I wanted to go to grad school. It was actually a super last minute plan. And I originally majored in environmental science and I minored in geology. And so this was the only geology program that I had applied to. And the other master's programs that I had applied to was conservation and restoration. And so when I interviewed with my advisor, he, on top of geology, he also asked me if I wanted to do conservation and restoration. And the fact that I was able to do best of both worlds kind of just drew me in.
I studied biology in Southern California at a small liberal arts school down there. Originally, I'm from the Santa Cruz area, and so I graduated in 2019. 5 years of just kind of working in different areas in education, marine science education, and environmental consulting. And then decided I wanted to go back to school and wanted to stay in the area. And what drew me to Moss Landing was like my advisor, Cheryl Logan at CSUMB. I was really excited to work with her on her coral project, and she seemed like a great mentor. And the community at Moss Landing was something that seemed very tight-knit and exciting to me. And I really liked that the classes were kind of like full days, and the activities and classes structure is like very hands-on. That's kind of also why I was interested in coming here. And yeah.
Nice. Yeah, that's actually really fun to know. Like, I know a lot of you obviously pretty well, but it's really cool to hear like the backstory about like how y'all came here. It sounds like either like a mix of location or degree or like area focus or collaborators or something like that. That's really cool. So anyways, we can open it up to the group now. If any of you guys have any questions for our student panelists here.
What does it cost?
It's grad school. Usually, like, if you find the program, like, the way it works is usually you ask around, like cold ask like a bunch of PIs, say, hey, you have a project, I like your project, do you have funding? Some of them usually have funding, but if you want something very specific that there's no funding, it's kind of like, you can become a student, but you kind of have to hustle and apply for a lot of grants and funding if you want to do specifically what you want to research. But most of the times, like, we just kind of email professors that have something that we like to do, and we can kind of like, figure out what projects are available.
Like, as far as like tuition costs and stuff like that, it kind of depends. So, probably about half of the grad students here are through CSUMB, and the other half are through SJSU. And I'm not sure if their tuition costs are exactly the same.
So the degrees are not offered through Los Angeles Marine Labs, you just learn here? Is that how it works?
Yeah, so it's,
Classes, research classes.
But then you're getting a degree from SJSU or CSUMB?
This is technically a satellite campus of SJSU with the home campus, but with a memorandum of understanding that was established a year ago. We used to be a consortium of, I think it was 9 different campuses in the CSU, and since our students were only coming from CSUMB and SJSU, they created a memorandum of understanding between the two, where SJSU would be the home campus, but students from CSUMB would still be here as full-time students, and that any SJSU students here could have access to facilities and other benefits from CSUMB.
So you would get a degree if you go CSUMB or any other CSU?
Right now, it's just CSUMB and SJSU, so your degree will be from whichever home institution you have. So for Emma and myself, that'll be CSUMB, and for everyone else up here, that'll be SJSU.
So it'll be the same at Hopkins? They do the same deal like that?
They're only Stanford.
They're only Stanford, and that's it.
They're a satellite campus though, so it's kind of like the same thing where their home campus is in Stanford, but ours is like in SJSU for some of us at CSUMB.
Thank you for explaining that. I never do that.
Yeah.
How did you all narrow down what you wanted to study?
Luck. Yeah, I was lucky enough to do some undergrad research in Florida where I did hands-on shark and ray field research and incorporate that into my undergrad thesis. And then when I applied here, my advisor wasn't even faculty at CSUMB at the time, so it was really lucky that he was incoming faculty when I applied. I kind of just applied with the baseline, like I'd like to work with bigger picture ecological things, and I know that an easy way to do that is with fish. And then my advisor contacted me, likely given my application by one of the advisors here, and was like, here's someone who has worked with sharks and rays, and he works with white sharks, and he contacted me and we hit it off, and that was that.
Yeah, I'm kind of in a similar boat. I went to Florida as well, but I looked at phytoplankton, so I was studying red tides. You see all the dead fish on the beach, and everyone coughing, that's what I was studying, and I got to do a little research there during my undergrad, and that sparked my interest in studying harmful algae blooms. So I also did luck out because someone loosely mentioned down in Florida, you should talk to this woman named Holly Bowers, and fast forward, we hit it off, and I love her as an advisor now.
Is she married to Mark Bowers?
She has a partner, but it's not Matt.
Okay. So I had a marine professor in college named Dr. Bowers, so I was like, wait,
no, not that.
So I have a question for you then. Would you then know about this, why we have this blooms of the Sargasso seas, like in Florida, for example, and it's coming from
The Caribbean?
in the Caribbean? Do you know?
So there are a lot of different types of blooms. That's more of a macroalgae bloom. So I don't specifically study that, but I know blooms are becoming, more prevalent, and their distribution's increasing because of increasing temperatures and human-induced activities like nutrient runoff. So around here, we have a lot of agricultural land, and so when it rains, that runoff and nutrients from the farms, the fertilizers, seep into our watersheds, and so that helps stimulate a bloom. So we're basically feeding it. We're amplifying that bloom. So here, I'm more familiar with the landscape here. I'm not sure about the sargassum issue in the Caribbean and Florida, but I have heard of it.
It seems to be a lot
Very
worse.
deep.
Yeah.
On the beach. Not very nice.
Were any of you considering or like choosing between doing a master's or a PhD first? And if so, why did you choose doing the master's first?
I was thinking about a master's or PhD route, And I decided that I wasn't positive that I wanted to spend all of the time required for a PhD. And I didn't want to go into a program without being sure that that's like, that the outcome of that and the degree that I would get from that, that would lead me to like down the kind of professor route is what, I wasn't sure that that was what I wanted. And so I wanted to just spend a little bit more time in research and get a master's degree with the possibility of like going to other fields besides academia. So that's why.
Also, like a lot of jobs don't really like the amount of skill that you have as a master's is pretty comparable to that of a PhD. And I think the nice thing about like the reason I picked a master's over a PhD, it's obviously time, but it's also like I feel like a master's gets you a lot of the skills a PhD does too in a shorter amount of time. But, yeah, I think I just repeated myself. But like the way I think about it is if you're, because the thing is I wasn't super confident. I wanted to do some like you know, do what I'm doing now when I went into my master's, and I was like, oh man, I don't want to like have this regret like a few years down my PhD. But now I'm like, okay, I'm really liking my master's. I do want to continue this, so I'll probably apply for a PhD. And the nice thing is I've done a lot of the classes that a lot of the PhDs usually do, so the time there would be a lot shorter coming in with a master's.
How many students are there roughly in the program?
I think it's around 100 right now.
Okay.
Small community.
Everyone knows each other.
Yeah,
It's small, but it's also growing too. The
Yeah.
cohorts have been getting larger and larger every year. So I think this last year there was like
25.
25.
The thing is, it's only like one hallway, as you notice, so we walk by each other like probably like if you're there that day, you walk by everyone.
Multiple times a day, yeah.
So you gotta know everyone.
And not everyone who's enrolled is here all the time. Once people kind of finish their education, like their formal classes and their on-site research, they're free to write from wherever. So we have many students who are like all over the US, even different parts of the world, who are writing their actual theses before depending.
How many teachers are there?
How many tenure track?
9?
10?
9 or 10?
10?
Yeah. Probably?
9?
What about affiliate? Like, or they do?
There are 9 or 10 tenure track, and then there are some affiliate and postdoc people who come in to help support with other classes. So there are only like, what, 5 or 6 classes offered a semester?
More.
More?
More. I think most of them teach every semester, right? At least one class or a seminar.
But some do combos.
True. But still, maybe like, yeah, okay, true. Maybe like 5 to 7, something
Yeah,
like that,
5 or 7 classes a semester.
They're very intense too, so like it requires a lot of like time because it's like 9 to 4, usually the full-time classes.
And most people just take 2 classes, like undergrad, usually you're like, oh, 4 classes, and that transition from undergrad mindset to here. I remember when I applied, and then I got in, and my advisor was like, so you take 2 classes, and I was like, I can do 4. You can't do 4. That's typically your first year, you take 2 classes, core classes.
Yeah.
Is everybody doing possible PhD route after the master's, or?
Not everybody. People take a lot of different paths. There are a lot of people who do continue into a PhD. Like I know for me that's my plan. So a lot of people do end up doing that, and it's usually the people who want to kind of stay in like the academia route. So like for me personally, and for a lot of people I know, we want to be professors ourselves one day. So that's like one path that a lot of people do take, but a lot of people do end up stopping at the master's and going into other fields, like continuing research or going into like policy, stuff like that.
Piggybacking on that, do you know what kinds of jobs besides the professors in academia that you might be interested in, or are there people recruiting you, or how does that work out for you?
So a lot of us want, like a lot of the people want to work at NOAA, but there are things.
Difficult right now.
It might be a little difficult in the future, but
There's some like NGOs and like local organizations and stuff who end up hiring a lot of like Moss Landing graduates. So it's probably the people who don't end up in academia go and work for those kinds of organizations.
I know there are people that go work at consulting firms after too, so that's not common, but people do it. Yeah.
Fellowships, internships as well, which are shorter term, but can usually get people into like the policy side of things as well. We have a lot of people who are either in the program and doing those things or just finish and then do that as well.
Can I even throw the private sector in there too?
Yeah.
A lot of people go like, we have an aquaculture facility, a lot of people like streamline that into something else too, so.
Like water quality too, like urban water runout, like anything that involves.
I know Carol also works for the USDA.
Yeah.
So
It's more of like how your
There's a little bit
skills
of everything.
transfer.
Yeah, there's a little bit of everything, sprinkled everywhere. Yeah.
Aside from like today's event, how often do you open to the public and talks and seminars and speeches?
one day a year. It's this event, but we do offer tours for groups. It's usually like some like schools will bring like a class and we'll give them a little like guided tour of the facility. But yeah, other than that, we're close to the public for the whole rest of the year. That's why we make this such a big event and we try to get all these people to come because we want to share our research with everybody.
Do you still have weekly seminars that are open to the public, or they're all on Zoom now?
They're hybrid, they're half and half. So they're not typically open to the public. You can reach out. We do have them weekly. This semester it's on Wednesdays at 4. So if you are interested in coming, you could potentially reach out to the front desk and see if you could get kind of an accommodation for coming in to see the seminar. But we do also have the Zoom links.
Are they on Zoom too?
Yeah.
We live stream them on YouTube as well. We have a Moss Landing YouTube, and so you can probably go on there and find like old recordings of seminars, and they live stream them when they're happening.
Oh, great, thank you.
You can find them on the Moss Landing website too.
Okay.
This might be a lot to ask, but I think I would like to hear each of the students' thing that they're most excited about from their research. Just talk about it a little bit. Like what's getting you really jazzed?
Ooh, I have something I can talk
Yeah.
about.
Yeah, go for it.
So I became a parent to 6 beautiful little baby jellyfish. It was actually yesterday, because so I've been running kind of a trial experiment. So for my thesis, I'm looking into the environmental drivers of jellyfish reproduction. And so they have these little polyps that are super tiny, and they will settle onto like rocks. And then when certain environmental conditions are favorable, it'll cause them to start producing these little larvae that will then kind of pop off and grow into the typical like jellyfish that we know and love. So I have had a couple of polyps for the past couple months that I've been caring for, and I finally got them to produce larvae. So I have little baby purple jellies.
Do you do that in a little petri dish or?
Yeah, I actually have, they are currently in a little plastic Tupperware container, and there's a hole cut in the side with some mesh over it so that I can have water flowing through, but they don't get, so they don't flow out of the container, and they're sitting on like a little glass dish in there.
Oh yeah, I'm pretty jazzed about like the methods I'm doing. So I'm doing something called metabar coding. So it's kind of like a new way to kind of like use environmental DNA to find out what's in a water sample. And it's just using like, just DNA to find out like the base of a food web. And it's really cool because one of the things I'm studying is microbes, which are like, super small, but the thing is they produce a lot of our oxygen. They do a lot of things for our like, trophic food web. They pretty much the start of the energy, like where energy starts. I think so. I'm sorry to everyone else. But it kind of, if you know how this change, like, if you know how like the base of the food pyramid changes, I guess, it's really easy to figure out how it's going to affect everything up the trophic level. And it's like kind of cool too, because it's like technology that's been around for 10 years, but it's like becoming more advanced as like, you know, a lot more molecular tools, you know, get more advanced, I guess. And I'm just really excited because since it's so new, I'm like seeing where the technology is going to go in the future. And I really do want to like be part of that because not a lot of people like know what I'm doing. So I'm like excited.
Should vet something.
Yeah, hopefully.
I'm really stoked that my research is the first of its kind at the spatiotemporal scale that it is. So at the spatial scale and the time scale that it is. There's been a lot of research on juvenile white sharks in Southern California. Not so much up here. There's like one paper that documents a big scale range shift from Southern California to here across the entire coast, but nothing within the bay, nothing using satellite data, which is what I'm doing, or the way that I deployed the acoustic resources that I did. So I'm really stoked about that and the results and what it kind of is showing and trending for this area as a new ecosystem as well.
So you use acoustics to find where the sharks are, or?
So we have a limited network out here. We don't do one where we don't have enough set up to pinpoint where they are in location. We do more presence absence of whether they're in a certain area or not.
And how do you do that? By drones or?
No, so we deployed some subsurface moorings that sat in a certain place in the water column, and then we tagged sharks, and when they were within range of that, they would send a ping and say, I'm here for this amount of time, and they would go off every minute or so. And so for every minute they were there, we got a beep, and then I would record that as a unique shark or a number of detections for that day.
Well, it seems like in Florida right now, there's so many sharks coming close to the where people swim, that they're deploying drones, cameras to alert people. That's how bad it's gotten.
We use drones as well, and actually there's been a lot of research that shows that the sharks were already in the areas that people were swimming a lot, and they really don't want anything to do with us. Any case of biting is just a mistaken identity. So we're all using the same water no matter what, whether we know it or not.
I'm really excited to, you know, I do a lot of molecular work for my research, and there are 4 quantitative qPCR, which stands for quantitative polymerase chain reaction assays that haven't been published yet, and I'm using them to look at a harmful algae species, the abundance of Pseudonychia. So Pseudonychia looks like it's a diatom, and it looks like needles in the water, basically, under the microscope. And it produces a neurotoxin called domoic acid. So you've probably heard about this, of, you know, whales beaching themselves in Southern California, or birds dying, fish dying. So that's what's been responsible for, you know, all those events lately. So I'm looking at that, and I'm really excited to use molecular techniques, which haven't really been done for community composition for Pseudonychia.
So how would they find that in a beached whale? Would it be in their gut or in their skin, or?
Yeah, that's a great question. I am not on the marine mammal side of it. I am not sure if it's in the tissue. I know it affects the neurons of the actual animal itself, but you can test for. Yeah, that's a great question. I'm not sure what tissue, if it's in the tissue or how to process those samples. I just do it in the water, so raw water, basically. Yeah. Yes.
Do you know if domoic acid is biomagnified?
It depends. It can be. There's a relation. There are a lot of papers that demonstrate increasing temperatures amplify domoic acid, and if you know, we eat fish that are contaminated with domoic acid, we can get domoic acid poisoning, so it can climb. Yeah, so don't eat sport fish when there is a flume, because I write those reports, so just I send them out. I'm the one that goes out every Wednesday and writes those reports for South Monterey Bay.
Are you measuring the DNA of the diatoms?
Yeah, for domoic acid, not for the domoic acid, but I am measuring the DNA for when I do qPCR, yes.
So when you do that, is there like an ambient level of that particular type of DNA in the water?
Yes, there is. So you always run a blank. So naturally, pseudonymia does exist in the water column in background concentrations, but within the last few decades, it has increased with nutrient inputs, rising temperatures, things like that.
What would be their main nutrients?
Nitrogen, silica, phosphorus, those are the main three that usually support these big blooms. Silica limitation has been shown to increase domoic acid production, so there's a lot of different nuances to it. Different macronutrients and micronutrients also impact the abundance and how much toxin is produced.
Is there any buffering to the domoic acid possible?
That's a great question. Man, you're asking good ones.
What was the question? I couldn't hear the question.
Is there any buffering to the...
Buffering, oh.
Yeah. I don't know at the moment.
Why do they make domoic acid? I mean, what does that do for the diatom?
Yes, that is an excellent question, and that is a PhD question that someone should
Future study.
We don't know if it's allelopathy, we don't know if it's a defense mechanism, we don't know, it's kind of up in the air in the research right now, but I'm sure it's being investigated by somebody out there.
I want to give our last two panelists a chance to respond to what you guys are most excited about. It's 4 o'clock right now, so.
Yeah, so for my minor in geology, the first class that I ended up taking, it was called Coastal Systems. And the first issue that they talk about is essentially because of sea level rise and because of the intensity of waves, it is starting to degrade some of the sea cliffs. And so any houses that are on the top of the sea cliff, it will get completely destroyed. And so that's actually what I'm studying right now, just because my beach, or main beach in Santa Cruz, there is West Cliff, and so there are a bunch of houses that are around it. And so the fact that I learned that, and that was just something that I've been wanting to study, and now that I'm doing it, it's just, I don't know, it just gets me really excited.
Right now, I am running a pilot experiment for my thesis project, and I'm interested in how corals are impacted by changing ocean conditions because of the temperature. So for my project, I'm taking coral samples that were collected in the Galapagos and exposed to different heat stress conditions and preserved, and I'm taking those and I'm looking at the gene regulation and gene expression level that is happening in those corals as a response to the heat stress. So looking at different genes and proteins that are upregulated or downregulated or changed, and kind of give us a story about how the organism is responding to the stressor, and then from that information we can kind of gather how corals might be responding in a way that is making them more resilient to climate change and warming oceans, or more susceptible to bleaching. So that's kind of what my project is on, and we just did an RNA sequencing run for the first time of my samples, and I got all the data back, so it's a lot, but it's also exciting and first part of my project.
Sweet, well that's all the time we have. Feel free to ask any of these guys any more questions afterwards if they don't have something to run off to right now. And can you all join me in giving them a round of applause? Thanks, you guys. Thanks, everybody. That was it, yeah.
Oh, cool. I got one more. Is anyone doing any research on viruses in the marine environment? There are like lots of viruses in the ocean. Lots of interactions between the phytoplankton and viruses.
Yeah, I don't know of anyone
It's like
here that's doing that.
Yeah, I do know a little bit, but I don't do the research in it. But typically, viruses kind of act as like population control when it comes to like anything living in the